That's a shark there.
And so, if you're just joining us, we are 127 kilometers north of the Azore Islands, north of the northwestern island, the island of Corvo. We are at a latitude of 40.4528 north, longitude 29.9093 west. We've just reached our maximum depth of about 1,230 meters.
1236, I guess, because we're still about 4 meters above bottom. 1235.
And we are on the. Let me see if I can spatially orient this. We are on a normal fault on the western side of the Mid-Atlantic Ridge. And we're in a small volcano. We're on the eastern side of the western-facing slope of that volcano. That's on the western side of the normal fault. That's on the upper end of the top of the footwall of that normal fault.
It's all easy for you to say.
Yeah. Well, if you think about a normal fault, there's something called a footwall and a head wall, and the footwall is basically the one that sits on the underside, right? But since we're on top of it and it's exposed, we're on the top of it.
Gotcha.
So it's the foot on the top.
It's all about the angles.
Yes. And this volcano has been kind of bisected. The easternmost 20% of it has basically been cut in half by this normal fault.
Yeah, I really like seeing that in the mapping. You can really clearly see it in the plan view.
Yeah, and I was when we were looking at the slope of this feature, I think I mentioned that, for some of these constructional submarine volcanoes, we expect to see slopes between 15 and 25 degrees. And on this side that we landed on this inner side of the volcanic cone or crater, the slope was much higher. It started being 35, 40 degrees, which was different than all the other faces of this volcano, which to me makes it seem like we should expect to see lots of talus and rubble, because that talus and rubble, as it falls off that volcano, it actually makes the slope a little steeper as it starts to approach what we call the angle of repose. And so when we land here and I start to see all of this rubble,
you called it pretty well.
I'm validated in my...
Validated for sure. I'm really impressed with what I see. I've already seen, we saw a shark when we came down, and I can see several sessile organisms on the rocks looking up the slope. So that's pretty impressive. Despite this mangled word that I just typed into the chat room, we're calibrating the camera right now, doing a white balance. You saw the color at the edge there before. We have 4 trained videographers, engineers on board. Take great care in the cameras, in the lighting, in the color balance, and this results in some of the unbelievable video that we have and the image products, many of which are available on the website. If you're seeing some of these things you really like and want a new desktop for your computer, they're all available for download.
Yeah, if you want to take down some of the James Webb space stuff and put up some NOAA ocean exploration, GFOE stuff.
Some of the otherworldly pillows that we saw yesterday would be a fine backdrop, especially if they had a coral on it.
Yeah.
Yep.
I shared
both worlds.
I shared the Moitira Vents short with some of my administrative staff at the Hawaiian Volcano Observatory, because they're like, "Oh, where are you? What are you doing?" And I shared that to them, and they were just blown away with both the content and the composition. So they really loved it.
So now they feel a little bit better about signing those approval papers.
Yeah,
for the
exactly.
travel.
Right.
Excellent.
Appro-
He is doing something out there.
Approving that budget that makes me go work for someone else.
Terrific.
So.
I just forwarded you an email that we received from one of the scientists on shore. Let's see. That was Daphne Cuvelier. And let's see, Daphne looks like she's at the University of the Azores, and she put together a mosaic of one of the, let's see which one, the bulky fumarians. And so she took a bunch of the imagery and sort of pasted it together, and she sent us a low-resolution version, but you can see the magnificence of this structure.
Yeah, it's a beautiful chimney.
Yep. So thank you for that, Daphne. And we have just collected our second water sample, and we use these. The water samples are brought back to the surface, and we filter them, and then the filters can be taken back into the lab and DNA extracted from the water. And this is another way that we now use in our modern area of molecular biology to do biodiversity surveys. Basically, all these organisms that are in the water are shedding some of their skin cells into the water, and there's DNA associated with those cells. And so we can process that DNA and get some understanding of what is the diversity of organisms, even if we can't necessarily identify them all. But with more of the sorts of video that we're taking, the samples that are being taken, we can then relate what's in the eDNA sample to what we have seen on the bottom. And we have a much better catalog for future camera-only based studies. So these eDNA samples can be quite valuable.
Yeah, and that's always really amazing to me because you think about probably the lack of biological organisms that we're actually observing. What are the ones we are missing, and so what else is out there, or what is microscopic that we just aren't sampling.
We know for certain that there are many of the fish that are avoiding the ROV. And then, as you say, we've talked a few times about the in-fauna, the burrowing fauna, or just stuff that's too small and tucked into the corals and so on. And, of course, bacteria, which really rule the world, are extremely diverse. And all of those will be picked up in the eDNA. All right, so it looks like the calibration of the camera is complete. I had noted in the chat room that you were validated, and I really liked the view out of my ROV window this morning, and Mike Vecchione is suggesting I don't open that window.
I see something.
Copy that, Nav. Thank you. Understood. So you're satisfied with the camera and we're ready to start looking? All right, cool. So let's just to the right of the lasers, there's a rather large rock there. Let's just have a look at what's on there, and then we'll go over to the left of that three-branched coral. Thank you. Oh, yeah. Sorry, yes, I don't see the fish, so... Oh, there we go. Yep, go for it. Absolutely. The fish are mobile. Yep. There's a second fish. The one in the water column is a midwater fish. That's cyclothone. Let that go. There's lots of them. I am going to predict this is going to be a codling. Yeah, so to my untrained eyes, this looks like
we are ready.
a codling. I'm seeing that
10 meters, 060
really long
decimal 2 knots.
Roger, we have a range
dorsal
of
fin
10 meters bearing 060 at a speed of 0.2 knots.
Good copy.
Really nice view. It's got a little peduncle before it gets to the shorter caudal fin, long dorsal fin, long anal fin, and then these modified pectoral fins with what I interpret to be sensory structures on them. Limonema, I believe is the genus, and at least the one that I'm looking at here in the guide, short-beard codling, perhaps, but something in that genus. Thank you, pilot. That's a great view. Seeing several colonies perched on some of these rocks here. We're going to have a look just to the left at the first rock we came in on. I'm not sure if that's the shark still hanging around back there in the distance. I'm not sure if that's... We saw a shark when we first got here, but let's just check to see if that's what it's... Yep, still there. Just kind of hanging around in the background. There we go. Well, that's a good view. Thank you for that. Les Watling thinks that the codling we were just looking at was a juvenile, young individual. We don't see too many of those, he says. There's an Oreo swimming by. I expect we'll see lots of those during the dive. We don't need to go on that right now.
ROV nav bridge.
No bridge.
Move is complete.
Copy that, thank you.
Yeah, the sponge? Yep, absolutely. Yeah, so we first got down here. There's a number of things on the rocks. So we'll just have a quick look at several of these so we get some sense of what's down here. I see a stuck crinoid just to the right of that sponge. We were seeing several of those the other day where we had the snail for the hydroid growing on the stalk. It looks like the same thing may be going on right there. But this is kind of a large platy sponge. I suspect it's a demo sponge, yellow in color. You can see those two red dots. Our laser's 10 centimeters across. So this is probably at least half a meter from side to side. It looks like there's all kinds of things growing on that sponge. I see at least a hermit crab on the top of it. It even looks like there's a coral either growing off of it or behind it. And I see the arms of a brittle star. And so now this is interesting. I'm seeing mostly white to this large sort of plate sponge, but then there are little clumps of yellow here and there. And I don't know if that's the same sponge or a second sponge overgrowing it. There's our little hermit crab using his gastropod shell. Yeah, so there you can see some more of that yellow texture. Understood. Yep, no problem. There was a second type of sponge down below. So the ship's already moving, so we're just getting quick looks at things. And there's lots of coral up here. I see, I think, an iridogorgia on the far left. Maybe a black coral. When you can pilot and you go over one, try to get a look at these white thin branch things. I understand that you're moving, so it'll be quick. Okay, thank you. Just so we can get a sense of what kind of coral we're talking about. It's an octocoral, but you know there are more than 3,000 species of octocoral, so we'd like to get a good look and try to nail it down a little bit more. My sense is that at first I thought these were going to be bamboo corals, and now I'm wondering if they're going to be the anthothylids like we collected the first day. Yeah, I kind of think that's an anthothylid, family Anthothelidae, and I'm basing that on the fact that the main axis of the skeleton almost looks wiggly. The tissue is kind of thick, and you can see these little mounds, and then you see the polyps themselves have a different texture or transparency to them. You can see through them, but you can't see through the main tissue, and that's a hallmark of both the plexaurids and the anthothelids. So based on the collection that we made earlier in this expedition, I think this is Amphithelia Day. Thank you, pilot. That's good. Sure, yeah. Yeah, a little lava sponge, thank you. I think we were seeing something like this. Was it yesterday or the day before? It must have been the day before because we were so deep yesterday. I wouldn't expect that the species that we were seeing yesterday at below 3,000 meters would be the same as the ones that we're seeing today.
Are you talking about the sponge in view right now?
Yeah.
I thought we did see something like that yesterday.
We saw something very much like it, but I'd be surprised. It was kind of smoother, thicker, lighter in color, but maybe. There's also a very interesting, where the left laser dot is now, that encrusting thing I think is another sponge. And I peeled several very small ones that were less than a centimeter off of rocks yesterday. It's quite amazing to me. The diversity of the sponges when you get right down to the really small size stuff. And I don't know if those are sponges that stay small or they were so deep that there's just limited food. So you notice this one doesn't have that sieve plate at the top, and there we get a good view of how it's attached to the rock. It doesn't have a fine holdfast, but I don't see a lot of thread-like silica. I forget what the name is for that sort of tangled web of silica fibers that attaches to the base versus a nice clean holdfast. I think, at first I said a sponge, but now I'm thinking this is a tunicate. Yeah. Oh, that's a really good view. And I'm saying it's a tunicate because you see within it all these white spots, which I think would be the guts, basically. So if this is a compound tunicate, and I'm pretty sure that it is, check that. Yeah, Asako Matsumoto is agreeing. So a tunicate is a chordate. It's one of the chordates, and they're filter-feeding organisms. And this is a colony. You can see several holes in the colony. And let's see, I'm not sure what kind of compound tunicate this is, but in some of them, each individual will have two openings, one for bringing water in, they filter it, they get the particles out, they digest it. The white spot would be basically where the stomach and other organs are, and then the other opening would be for excurrent. But in some of them, what they call compound tunicates, several different zooids will share a single excurrent, and they'll have arranged around it in a ring several incurrents. Well, that's cool. So that was a tunicate. There's that shark still hanging out there. Yep. Kind of looks like this gulper shark that I'm seeing. This fish guide. Yeah. What is this indeed? Age-old question. I think that might be a... Oh, wait, there's something at the base. Boy, I wonder if this is a pseudomorph or if that's a snail at the base. I'll bet it's a pseudomorph. It's one of the best examples I've seen, and I see Mike Vecchione's in the chat room, and if he agrees, then I'll explain what a pseudomorph is. Yeah. Well, it's either a snail that's hanging down there at the end that's producing an awful lot of mucus, or, yeah, okay, so Mike Vecchione does say it's a pseudomorph. So a pseudomorph basically is an ejection of ink mixed up with mucus that a squid will release in order to distract you. And it's doing a really good job because it means the squid is somewhere else around us. Really nice. That's a nice one because usually the ones that I see have just passed through the thruster, and so they're getting torn apart. And there you can see that that mucus does hold that ink together long enough to make it look like some kind of organism swimming in the water column. And Mike suggests that that white dot that's hanging down below it is some kind of marine snow that got bound up in the mucus. So during the morning here, as we're moving up this slope, we're going to be moving a little bit more quickly perhaps than we have on some of the other dives, and that's because we really want to get to the top of this rim to see the community up there. This isn't some of that brown stuff, right?
No.
Okay.
You know, I was still keeping my eyes out for it. And so one of the things that we've been requested to look for is from Ricardo Ramalho at the University of Lisbon, who's interested in these explosive underwater volcanoes, which this might be pretty distinctive of, given this kind of crater-like feature at the summit of it. You know, we saw these constructional elongate pillow volcanoes on the mid-ocean ridge axes, like on our axial volcanic ridge dive last week, and that had a really constructive top. There was no kind of crater or pit at the top of it. And so the fact that this feature, this volcano we're diving on now, kind of has this large, maybe several hundred meter diameter crater at the top of it, that could be formed through explosive volcanism. So one of the things that we're going to be looking for is what we call hyloclastite deposits. And hyloclastite deposits are mixtures of fragmented basalt and glass that is quenched quickly, and it tends to alter very quickly and oxidize. So you usually see this kind of darker...
Rope? Sorry, Pilot, is that net or rope, that black, or is that just...
I think it looks almost encrusted.
Okay.
Yeah. And so what we're looking for is kind of these dark red, or not dark, dark brown, lighter red, weathered sedimented features that'll be much more different than kind of the white calcareous sediment that we've seen before, and it'll look like it'll have a mixed in broken basalt pieces in it, little pieces of glass and volcanic pillows that you can imagine kind of get entrained when this process occurs and this eruption happens. And so one of the reasons that's interesting is we were talking about volcanic glass the other day and how it allows for subsequent geochemical analysis. So Ricardo Ramalho at the University of Lisbon is interested in looking at the glass grains in that hyloclastite deposit and seeing is this more indicative of mid-ocean ridge basalt processes that we're seeing on the ridge, or is it more indicative of maybe hotspot volcanism that we're looking at in the Azores? And they can look at the glass and they can look at what we call trace incompatible elements. So we're thinking like rubidium, cesium, that tend to be preferential in ocean island basalts, those hotspot volcanic products, opposed to the mid-ocean ridge basalts. So end of the day, keep your eyes peeled for brown dirt with black pebbles in it.
Brown dirt, black pebbles, copy. We're seeing on the right there is a bird's nest sponge, Pharonema carpenteri, I believe. We did collect one of those the other day. Just below it was a cup coral, and then we're zooming in here on a plexaurid coral, I believe. Oh, I'm fooled. It's a black coral. Okay. So, yellow-colored tissue over a black skeleton. I have no idea what genus this is. It's not a morph I've seen before, so I will stand by and hopefully somebody else can provide me an identification. But the reason I know it's a black coral is because I'm looking at the polyps, and the first thing I noted is that the tentacles of those polyps don't have the little side branches that are characteristic of the octocoral. So then the next thing I look at is, well, can I get an idea of how many tentacles are associated with each mouth? And I can see that it's fewer than 8. And, you know, that's hard maybe, but I understand the way this thing is put together, so I can see that it's clearly a hexacoral. Maybe not so clearly, but it is a hexacoral. It is a black coral for sure. And that's a good view. Thank you. If you did pull out just in front of it, there was something reddish on the floor, not on the rock, to the right, I think. Yeah, a little bit lower than that. There it is. There it is. It's a rock pen. I knew it. Just between the lasers. Yes, the peduncle. So here's a sea pen that's modified, and there you can see it really well. It's modified to be more like a suction cup, and it's holding on to a little piece of dead coral. And this is a classic anthotyllum rock pen. Beautiful.
And so is this the one that started on the rock, went to the sediment, and then came back to the rock? Or?
So octocorals in general, here you can see very nicely, 8 tentacles, that's an octocoral. So the ancestor to the octocorals was probably living on a rock, and most of the diversity of octocorals we see today live attached to rock.
Yep.
Sea pens are a subgroup that have that inflated base that allows them to live in a sediment, and you are right. This one clade, this one evolutionary lineage of sea pens has now returned to the rock. But what's interesting is the way they attach to the rock, this suction cup, is very different from the way all the other octocorals do it, which essentially cement themselves to a rock.
Now that peduncle, that's filled with water, presumably,
Yep.
and can it push the water out of that peduncle, or is it always kind of inflated with water for this particular?
It's a really good question. If you can get a quick zoom to the right of the lasers, there's another 3-branched. Yep, it's either a bubblegum or a swiftia. That's what I want to find out. Thank you. So that's a really good question. I haven't observed it, but in general, yes, sea pens can release the water from the colony to adjust their size, and I think especially the ones that are able to withdraw into the burrow. So I think this is probably Swiftia and not a bubblegum coral. So Swiftia would be a type of plexaurid, and one of the things you can tell is the polyps alternate. So it looks like it's zigzaggy. It's like an old phone cord that you've stretched out. And right now, all the polyps have their tentacles withdrawn into that little mound. And part of the tissue is removed from the top. You see there's a yellow anemone just below that hermit crab. Well, he's turned his back on you, so I don't think he's waving anymore. And then there's a crinoid on the branch on the right. Nice video. Carlos down in Horta at the ECC is saying that on the various drop camera and ROVs that they do, they don't see many anthotyllum in their images, but he acknowledges they tend to go shallower and primarily to harder substrates. So if we find sometime in this dive a somewhat larger rock pen, that would be a good sample to make. So far there's only one species known from the Atlantic, and it was collected in the Caribbean. So that would be novel out here. What is moving under the rock in the back? Sorry, Les, I didn't notice that. Here's some interesting structures. Look at these, a little chimney-like features, or columnar features.
Oh. More mucus.
They look like old tree trunks or giant charcoal briquettes.
Yeah, they very well could be kind of little manganese-encrusted chimney structures.
Another pseudomorph just drifted by. Yeah. Yep, that's what we were just talking about.
If you have time, you could poke it, but I don't want to take too much time. You know, it would be interesting in terms of if it's friable and we can get a sample, because that's one way we can identify whether it's a chimney structure or not.
Otherwise, around here, you know, we've been seeing so many pillows recently that this looks much more, I don't want to say sheet flow, because I'm not trained in that field, and I know you have a very specific meaning for it, but that's what the surface looks like to me.
Yeah, it's hard to tell just because it's so heavily sedimented.
Okay. Love tab upcoming, and here you're watching lower left. It didn't move very much. I wouldn't call it very flyable.
No. Yeah, you know, it might be interesting if we have time at some point, if we see more, or this, you know, trying to grab that outer surface, that far away portion of the circumference to see if it breaks off. But I know we have priorities.
Sorry, I was just typing in the, yes, please. I was just typing in the chat. I think that's is that a Synapha or is it too small? That might be too small, unless we have a theme of babies this morning. Small fish. I'm not sure if it's holding something or its mouth is just, its jaw is sort of moving up and down. Yeah. It has a ventral mouth. This is not a fish I recognize. This is new. Yeah. This is very different. So great video, great catch, pilot. Look at that. And even the dorsal fin looks really different. It looks like a series of very small spines and not much tissue between them. Cool. Mike Vecchione says the snub-nosed spiny eel, which every time we say we see a fish we don't know, that's what he says it is. So let's see. These look like rat tails with really long, flattened noses. Grenadiers. Or are they small chimeras? Rat tails. Thanks, Mike. Well, these are also fish that we have not yet seen on the expedition. I mean, we've seen the grenadiers, the rat tails, but not these ones with the really sharp nose. Mike Vecchione is suggesting the genus Coelurynchus. At least the ID guide that I have in that genus, we don't have anything that has a snout that's quite so pointed. So that's interesting. Good stuff. See, there's a fair amount of rubble on the surface right here. Center of the screen right now, if you could just zoom, probably right where the lasers are, or something very light and fuzzy. Yep, that's the right. There's a series, yeah. Well, so the one going off up out of screen to the left is a live black coral. Oh, actually, this is all black coral. I see. So there's 3 long stems, but they all connect at the base. We were seeing this on one of the earlier dives. So that's a different kind of black coral. And then growing at the tip of the black coral, so when you're ready, we can go back in, pilot. Copy that. There's, I believe, an octocoral growing on the black coral, and that's what the lighter white pink is with the much larger polyps. There's a scale worm swimming up, photobombing here on the left side. Yeah, great. So this is probably, you've heard me say this before, stoloniferous octocoral. It means it's the type of coral that grows as a ribbon on other corals and substrates. It's not producing its own axial skeleton, and it's growing on this yellow-colored black coral that that branches just at the base. Great, thank you. We can go on from there. Let's see, where did I see this? Manuela Ramos down in Horta is suggesting that fish, the grenadier we saw with the really long nose, is Coelurynchus labiatus, perhaps, and perhaps a juvenile form. There's some barnacles here on this black coral. Barnacles are crustaceans, and so they're one of the very few crustaceans that's sessile, that is, that kind of makes the, I don't know if I should say decision, but makes the transition from a larval stage that is swimming through the water column to permanently gluing itself to a substrate and undergoing this metamorphosis where it just uses its back legs to catch food. Good view. Several more sponges here. I didn't say it, but I forget what it was we were zoomed in on just a short time ago. I believe I also saw a very small stylasteran hydrocoral colony. A couple of more different sponges here. One of them quite frilly, reminds me of something in the family Farreidae. Let's zoom on the sponge in the center that's sort of beigey yellow in color. Thank you. Yep. So it's interesting. This one actually looks like one I think we sampled the other day, where it looks upon first view as sort of frilly, but when you get closer, it seems actually a series of hardened tubes. So I think this is a glass sponge, the one that's here on the right that we're seeing, with all these little tube-like structures that are kind of poking out from the inside, and there's actually hollow. You can kind of see at the top there's a pretty wide opening, and so you kind of like stick your hand down on the inside of this thing. There we go, or zooming in on that. There you can see the opening. Very nice. And very often there's a whole series of associates that live on the inside and in these protrusions, these little tubes that you see.
And texturally, if I was holding this in my hands, what would it feel like?
So the ones that I have held range from quite brittle and resistant to the one that we collected the other day, actually, if I recall, is quite soft. Now that it's dry, when you pick it up, very, very soft. Yeah,
it looks superficially kind of almost gelatinous and soft, but I don't know if that's a...
not gelatinous. Yeah. It has a fair amount of resistance, but I would say it's almost cloth-like when it's dried, except for the spicules, of course, which are silica. They're glass, and so those are kind of spiny. And then, Pilot, if we can just look over briefly to the left when... just a little farther left, the other sponge, yeah. So this is a completely different sponge that you see growing here with all of these different oscula on it, and then a couple of the large chimney-like oscula. I'm going to guess that that one is in the group Demospongio, or the more yellowish-hued one is in the glass sponges. Actually, you know what? I'm changing my mind. I don't think it's a glass sponge based on what I just described as it being cloth-like.
Yeah.
Yeah, so they're probably both Demosponges. But if you want to have some very accurate descriptions of the sponges, tune in to EX2206. That's our next, the Voyage of the Ridge 3, I suppose is how I should refer to it, when Joanna Javier will be the biology science lead, and she's a sponge expert, and so she'll be able to tell us a lot more authoritative information on the biology of these sponges.
Well,
Thank you, Pilot.
Since we're leaving the Mid-Atlantic Ridge and we're approaching the Azores, which is where 2205, the third expedition starts, and then they're going back to the ridge, I like to refer to them as Voyage to the Ridge, Return to the Ridge.
Oh, that's good.
Yeah.
Voyage to the Ridge 3, Return to the Ridge after Voyage to the Ridge 2.
Yeah.
That's good.
It rolls right off the tongue.
Yep. Some more of these anthotheelid, the yellow branching octocoral colonies below us.
Now this is hard to tell. This almost looks kind of sheety to me, but it's almost a little disingenuous because I know we're kind of on this rubbly pile, and maybe it's just the way it's been preferentially sedimented and then swept off. it's also hard to tell just because there's a ferromanganese incrustation on it. So there's probably a few millimeters of ferromanganese, and so one of the things I would be try to look for is kind of a break in a contact where you see really nice, maybe a cross section of a pillow, and then underneath or above that you see kind of that horizontal stratification of that pillow sheet.
I see.
Of that sheet flow.
I was going to say, it looks very different from the pillows I've seen yesterday, but actually from this higher view, I can kind of imagine that some of these have this pillow-like structure, but they've been kind of buried and covered over, as you say, with incrustation.
And so that's one of the things that's always the hardest to get used to is, we're used to thinking two-dimensionally and three-dimensionally, and when we look at something like this, we really got to think four-dimensionally. We got to think about what does it look like in 3 dimensions when it was in place, and then what does it look like in 3 dimensions as it's progressed through time and to where we're seeing it now.
Pilot, if you are heading towards that boulder, your eye is the same as mine. I would just like to have a look at the density. It's just an interesting example of how you can get local recruitment. And the question is why? Why is that boulder a lot better than all these others? Why are there so many colonies right there? Yeah, exactly.
It's a great boulder.
But what do they see that I'm not seeing? I really like, this is almost like a deep sea maze that's been set up here, or sort of a park with the way all these features are sticking up from the bottom, or maybe I should say the features that haven't yet been buried in the sediments.
So, do you think this was a small group of colonizers that was floating through the water and all landed on the rock around the same time? Or
Right, so that's exactly the question. So as I picture it, there are larvae. These are small balls of cells that have cilia that are capable of swimming or crawling. Mm-hmm. And I don't see any reason why a group of larvae would travel together.
Yeah.
Now, initially, maybe if they all hatch in a similar place, mm-hmm. maybe they're entrained in some body of water. The size that they are, we have this. They're living in a world that's very viscous. They experience water very different than we do. So maybe they are sort of held together and they all settle at the same time. The fact that they're all about the same size suggests to me that it's not that one got here and started producing offspring that then settled. Mm. But it could just be something about, they're being delivered from somewhere else where there's a common flow, and this is the first high spot that they get, and they settle.
And there's not a possibility of one of them landing there and then releasing some type of chemical that says, hey, everybody.
Yeah, that is possible as well. I would just think that they would be a little bit closer together.
Yeah.
Or maybe one settling on the other. Mm. That's not typical with the octocorals. I do see some octocorals do seem to have a preference for landing, for example, on old Lophelia, mm-hmm. calcium carbonate. I saw something else on the rock, though. What did I see? I think it was just as you were going in. Yeah, I don't see it now. Don't worry about it. Thank you. So I think those were a bunch of the Anthothelidae again. So I do believe all of those corals we see are of the same
Now that rock
species.
Is not very sedimented on that kind of leftward-facing face that the other ones around it are. So
right, and it's more angular, right?
Yeah.
And so it's a little bit taller.
Yeah.
It's more angular. Sediment's not accumulating, and so if the larva can detect some of those properties, then that would be a good place to settle. Alternatively, a larva can't determine those properties. The ones that have landed on these flattened surfaces have been buried before they can grow to that size. They grow very slowly. So you have to consider both the settlement process and what we call the recruitment processes, which is the successful growth of the larva after it arrives in a place. And that all depends on how much food there is and whether it's being buried. This is good. No, so we have maybe this other Swiftia, the coral on the right, the pink one, and then the sponge. This looks a little bit like the amphitheater sponge that we sampled the other day, just a smaller version of it. And it's interesting, we've now said for a number of things, this looks like a baby or a juvenile or small. And maybe that means we have relatively recent recruitment of a lot of different organisms to this area, or this part of the... would you call this a caldera?
No.
No? Okay.
No. So a caldera is a caldera is one of the things I study during my day job. I studied the Long Valley caldera in the eastern Sierras of California, which had a really, really large eruption about 750,000 years ago. So Long Valley is similar to Yellowstone in terms of the size and the volcanic processes that occur. And then there was a caldera-forming eruption in Hawaii on Kilauea in 2018. And so when we think of a caldera, we actually think of a really large body of magma that feeds eruptions on the surface, and then that magma is evacuated through an eruption that's further away underground, what we call downrift, through a rift zone, and it removes all of the magma beneath that summit area, and then the magmastatic pressure that's holding up the crust above it is no longer able to support the weight of the crust, and the crust falls down, sinks down. So it's almost like a sinkhole. Now what this could be is it could be an explosive crater through an explosive volcanic process, or another feature we have is something called a pit crater, and that's where we have a kind of similar to a caldera, except instead of just that material sinking down because removal of magma, it's almost an active process where the magma beneath it is moving and it's slowly eroding the inside version. It's almost a bottom-up process. I don't expect that those caldera processes don't occur here, and you don't expect those pit crater processes to occur here because we don't have anywhere where that magma is going. So a feature like this is usually thought to be explosive. Now,
so let me just say, this looks a little bit like the euplectellid bolosoma. The genus name would be bolosoma. It's a glass sponge in the family Euplectellidae. Thank you. Yeah, go ahead.
Well, and the other thing which is interesting, and I am not a carbonate geologist, so I can't speak with this to any super level of intelligence. But, when we're looking at those cross sections of those exposures right now, you got to think, remember, we're on 2.5 million year old crust. We could have easily several meters to tens of meters of possible calcareous deposition. Yeah. And so, a lot of those could just be kind of encrusted and concreted shells. And that kind of gives them that rough texture. And so
we saw something earlier. We didn't get a zoom on it, unfortunately, but it looked to me like it almost could have been old coral reef. Tumbled over and cut open. And so that would back up this idea that there's a lot of carbonate here. I understand from what you're talking about, where you're talking about sinking planktonic shells have been consolidated together, but still carbonate.
Yeah, exactly. Still carbonate, which still means I don't know much about it.
Yeah, that's the same fish that we saw earlier, so I think we're okay with that one. Let's go back to that sponge. Thank you. Yeah, one of these, big vase or bugle. Let's see if I can get this one ID'd. Yeah, so this looks like the one that I think has a much harder texture. It has this sort of tall vase or cup or bugle shape with these protrusions from the inside. And this looks to me a lot like a sponge that I remember collecting in the Aleutians. So it'd be interesting to find out if it is soft and flexible, or if it was sort of crunchy and hard. Hertwiggia, thank you. Hertwiggia flaccifera. All right, we have an idea on this sponge. When video's happy, we're happy. And I believe, Manuela, maybe you can correct me on this. I believe Hertwiggia is a demo sponge. Is that correct? In which case it would be relatively soft. There's a pseudomorph again. There's a squid somewhere around us. Yep. So this is mucus with some squid ink, and the mucus is sticky, so it binds up some of the marine snow that's floating around here.
And we call it a pseudomorph because it looks like a squid?
it looks like something.
Yeah.
So the morph is just referring to that it appears to have some morphology out there. The pseudo is meaning that it's fake, and so it's meant to be a distraction. You know, get your head to turn towards that, where the thing that made it swims away. Thank you, Manuela. So Manuela is telling me that that is a hexactinellid glass sponge. The that yellow one that we were just looking at, Hertwigia flaccifera. I'm not sure how to pronounce the species name. I'm not sure if it's flaccifera, flaccifera.
So when I want to go to lunch, I put my jacket over my chair, I put my hat on top of my jacket, and you think I'm still here because that's my pseudomorph.
Exactly.
Yeah.
That's exactly what it is. Let's see what's going on here. This looks like a, oh, I see. It's like a demosponge plate that's growing sideways off of this, I don't know if you would call this a composite rock or whatever it is, but yeah, nice. And you can see these sponges also have to contend with all of this sedimentation that's occurring, and so it gets on the sponge surface. So that's a challenge. I'm not sure I understand all the mechanisms by which sponges keep the sediments off. Part of it may be mucus secretion along with the water flow. And maybe that's a benefit of attracting organisms to walk over your surface as they clean off the sediments. So many questions to ask, so many studies to do. Manuela suggests that sponge may have been a Desmocella, possibly near the species Grimaldi. What do we got here? That is... It kind of looks like... I want to say it's an amphipod carrying a worm or an ostracod with a long tail, which is weird. A cumation? Yeah, you think?
Yeah.
I mean, I know cumations have the long tail, but okay. That would be cool. If Les Watling is here, he would love to see a cumation.
That's our data science manager, Arvind Shantaram, chiming in. We rarely get to hear from him, but...
Yeah, I like to chime in on the macrofauna size class animals.
There you go.
Well, Arvind, if that's a cumation, that would be the first one I've ever seen on one of these dives, so that would be kind of cool.
We're calling it. Put it in the dive log.
I see a cup coral down there just to the right of the right laser. There, just passed over it. Actually, Pilot, if you could look in your lower right corner in the sediments, there's, yep, it might be a Panatula. And this is a good opportunity to show the more typical sea pens that anchor themselves in the soft sediment. And what you, exactly. So this would have that inflated peduncle, but it's now anchored so you won't see it. You shouldn't see it. Maybe you'll just see a bit of a curvature at the top. But you can see these fleshy lobes that come off the primary or axial polyp that have the secondary feeding polyps. Oh, actually you can see the inflated peduncle there. And it kind of looks like it's perched on top, but I'm pretty certain that that actually protrudes deeper into the sediment. So you're probably seeing the top half or the top third. Thank you. Good view.
Peduncle's a fun word.
Peduncle is a fun word.
Mm-hmm. Word of the day, everyone out there. Peduncle.
Peduncle. In fact, this whole feature we're diving on is kind of like an inverted rock pen peduncle.
There you go.
So we could have called it Peduncle Ridge. Yeah. We're getting a word from Asako Matsumoto, from Tina through Asako, so from Tina Sako, that the yellow branched bamboo coral that, oh, look at that. Is that a gulper eel? It's certainly gulping.
Oh, wow.
That's a good view. Or a synaphobranchid eel.
A gulping eel. You guys are as imaginative as geologists.
Thanks. Now, that's just me, right? The non-fish person.
Oh, that's not his actual name.
Yeah, synaphobranchid, Mike Vecchione agrees. Very long and slender. I didn't see much of a dorsal fin there. Thank you. So Tina was saying that fan-like yellow black coral that I couldn't identify is in the family Aphanopathidae, she thinks. And then we're having some debate in the room about whether that was a cumation or a mysid. Tara's got my back here and says to you that there is such a thing as a gulper eel. So.
Well, that's fine, but you gotta remember I'm very gullible when it comes to biology. So if you say there's a gulping eel or a screeching eel, sorry, Princess Bride fans out there.
Screech eel. See some more cup corals, some whip-like black corals, the orange ones. One coming up ahead here. So we've seen the orange black coral whips and the yellow black coral whips. The one that's ahead of us on the right, I think is the yellow one. It looks like the base has been taken over by a variety of other organisms. They tend to get the name fouling organism. And a fouling organism simply means typically a sessile organism that will settle on another live organism and kind of take advantage of the free real estate, if you will. And all they're simply trying to do is get up off the bottom into the more accelerated current. There's a sideroid urchin on the sediments just on the left. Copy that. Yeah, really large, thick spines, I think, are characteristic of this group of urchins, the sideroids. There's a barnacle growing on this urchin. It's probably growing on one of the spines. And let's see, I'm trying to say, it's a little hard to see on this one, but it does have tube feet, and they're aligned in 5 rows, I guess, evenly spaced around the margin of this globular test. There's also a little scale worm tucked down there. It's kind of bluish purple, just on the lower side on the left. And not seeing anything else. This one doesn't look too active right now, but the mouth is underneath. It has these teeth-like structures that it used to grind. Oh, actually, there's a whole bunch of zoanthids growing on the spine coming out the lower left at about 7 o'clock. So that's another kind of cnidarian polyp. And so I just talked before about the fouling organisms. So you could refer to the zoanthids here, the barnacle, as fouling organisms settling on the spines of this urchin. Great. Thank you. No, F-O-U-L. Yeah. Foul in the sense that it's, you know, messing up the surface of the animal, which would, no. Oh, here's a Metallogorgia. How cool is this? I was not expecting to see that. This is pretty shallow. Let's get a good view of this. I think this is pretty shallow. So this is probably at the upper end of the depth range for this coral. So this is an octocoral. You see it has this long stalk. And what I will call the adult phase of this colony tends to get, you know, close to 1 meter tall. Some of them, a few of them are a little bit taller than that, but they don't seem to get much taller than that. And you see there's no branches on the stalk, but when they're growing, there are branches, and they shed them, and we're not sure why. So all the branches are remaining at the top, and inside the center of those branches, you see that sort of orangey red thing. That's a brittle star. Ophiocreus oedipus, I believe, is the name. And we've told this story many times, but it's such a fascinating thing. You only ever see that species on this species of octocoral. And so there's a very close relationship between the associate and its host that's clearly evolved together. and it raises all kinds of fascinating questions about how the larva of that brittle star finds this coral. And I think I can recall only once in all the times that we've seen this in the collections that we made have we found more than one brittle star on a colony. And that raises the question of, well, how does the brittle star reproduce? And I don't have a good answer for that. I mean, there are separate males and females. So you mean somehow the sperm of one individual has to get to the eggs of the other. And there are plenty of invertebrates that undergo what they call broadcast spawning. So they'll shed their gametes, their sperm and egg, into the water column, and the eggs will be fertilized into the water column and turn into a larva. But there has to be some reasonable distance between the individuals for those gametes to find each other in the space. So that's really interesting question there. And you can see how the arms unravel and can extend over all parts of the colony. And we think that the brittle star may have a role in keeping fouling organisms off of the colony. You notice there's nothing else sitting on this colony, even all the way down the branch, all the way down the stalk, excuse me. And probably part of that also is chemicals that are being produced by the Metallogorgia itself. There's probably something noxious in that tissue that organisms don't like to settle on it. The only time we ever see anything on the stalk is if tissue has been stripped off the stalk, and then you see all kinds of organisms will attach. And so that's a little bit of clue, a little bit of a data that is suggesting that there's something about the tissue that is a deterrent to other organisms settling on it. So that's a really nice observation. As I say, these are more common around 1,700, 1,800, down to about 2,000 meters. Right now we're at 1,205 meters. Good stuff. They're very photogenic, those corals. I used to call it the pink parasol. Oh, there's a large fish upper right. It's not a wreck fish, I don't think. No, it's definitely not. Oh, it's a roughy. Okay. That's a looks like a big old one. Orange roughy, I believe. Kind of looks grumpy this morning, this one. And so there have been fisheries for orange roughy in various parts of the world. And they're a bit of a problem. They're relatively slow-growing fish. They live to an old age. And if I recall my orange roughy papers, they don't reproduce until after a couple of decades of life, I think. Mike Vecchione is guessing that, this could be more than 100 years old, this fish. Yeah, just amazing. And so that's problematic with a species that you'd want as a commercial fishery, because if you fish the large ones and it takes, 40 to 50 years for them to start reproducing the healthy population again, you can very quickly wipe out that population. Great view. He's taking us on a guided view of some of the sessile fauna. I see a stalked crinoid behind it, a sea lily. Some black corals, anthothelids. Mike Vecchione says that they aged one of the orange roughy out here that was 120 years old. Wow, I had no idea that they got that old. Yeah, I thought they were like 40, 50 years old. But
no, it's amazing the ages of some of these organisms when we let them live into their geriatric years.
Yep. This gets back to something we've discussed a couple of times about how cold it is down here. Right now, the temperature in the water is 6.3 degrees Celsius. So the D2ROV has a number of sensors on it that tell us the temperature, the salinity, the oxygen concentration, even a measure of turbidity, which says how much stuff is in the water column and how easily light can penetrate the water column. So we can always reflect those environmental variables to the things that we're seeing. And where you're in very cold water, metabolism tends to be lower, you're using less energy, And I do believe that that equates with longer lifespans. There's less metabolic and oxidative stress on some of the chemicals and the cellular components, mitochondria, for example. So this, I guess, is this a sponge in the center with all these? This is like maybe a polymastia sponge with a whole bunch of like chimney-like structures on it. Yeah, absolutely, that's what that is. This is an interesting one. I don't recall the ones that I've seen before having quite so many knobby protuberances. Those little chimneys are the excurrent openings. So this is kind of a standard sponge in the sense that it's undergoing suspension feeding. It has cells on the inside that have little flagella that draw a current into the sponge basically by creating negative pressure, suction. And so that water is pushed out through the main openings. Yeah, so thanks to Upashana Ganguly, one of my grad students at UL Lafayette, has added the idea of polymastia to our annotation list on the video. So I was just checking my spelling, make sure I got the name right. But these polymastia sponges, they look like little hedgehogs, you know, from a distance.
Yeah, looks like a little hedgehog on its back with its feet in the air.
Yeah.
What a cute little belly.
This one.
Yeah.
7 big tubes coming out of it.
Are we in a position to collect?
Yeah.
One of our colleagues on shore, Manuela, says that she agrees it looks like polymastia, but never seen one like this. And I agree, I've never seen one that has quite this branching to the protuberances, not those chimneys. That's a very typical sort of oscicle for these sorts of things. But all those structures around it. There's a little octocoral growing on it, speaking of fouling organisms. And what's that in the background? That's an amphipod. Mike Vecchione checks his notes, and he has a correction on that fish aid. He apologizes. It was 139 years old. It's getting older every time we hear about it.
Watch, did you want to sample this? Or?
Yes, please.
Okay.
Sample.
It come out video.
Seems like a biologist fishing story, right? It was this big.
I'm sure it's around A-frame.
That's what I was starting to
Yeah.
think, but
What
apparently that's data that's written down.
0 samples.
Mike, can you remind me how they do the aging? Is that based on layers in the otoliths, I believe? Those are these little concretions that are in the, what I would call the ears. All right, so we're standing by. We're going to prepare to sample this sponge. Pilot, I think you had the lasers on it already, but is that about just over 10 centimeters? Okay, so do you think you could just try to scoop it from the base or grab it from the base so you're getting most of it?
Yeah.
Thank you.
Yep.
I do not see it. Where is it? Oh, there you go.
Right in front of me, yeah. You can zoom in partial on mini Zeus in addition to Zeus if you got it. Yeah. Oh, that's good. Yeah.
Oh, that's a healthy sized sponge.
Okay. Lower grip force.
See the spongy texture of this thing.
Just trying to gently squeeze it without doing too much damage to it. There it goes.
Very nice. Very nice.
Zoom out. I might hit the starboard side, Fernando, so I might have to come up, so just come out careful. Watch that starboard wing. Okay.
And it's in the port outboard bio box. Very nice collection. That loofah was a little bit bigger than I thought it was.
Yeah, the jaw's definitely like closed all the way on it, so it might have lost its structure, but it's all there.
Yep, it's a sponge. Should rebound.
Now, I remember hearing about otoliths, and this is a little bit of an interesting tangent, and I do a little art detective work as a hobby every now and then, and I bought a painting a couple years ago that I think is actually 300 or 400 years old, and one of the ways I was interested in kind of determining it was doing age dating. So I did some isotopic dating from the molecular, the isotopic geochronology lab at Arizona State, and then I was looking into dating the lead radium isotope concentration. So that's another way you can date things that are really young in the last few hundred years by looking at the relative concentrations of lead and radium or thorium and radium. But the sample sizes are so small, the people who are doing that work are the carbonate people and the fish people who are dating the otoliths through radiogenic decay of thorium and radium and lead and radium. Really interesting.
We should just note that otoliths are calcium carbonate structures, and we've been talking about a lot of different calcium carbonate structures through this expedition. That's what's in the tissue of corals, for example, and the spiny skin of all these echinoderms. So you also find them in fish. Their structures located just behind the brain of fishes, and their calcium carbonate is deposited in layers such that when you look at them, you can actually see these rings. And you were just talking about using the isotopes to age them. I believe, I'm not sure if they're annular or subannular, but the bottom line is with some experience, you can get some accurate ages on these fish.
Yeah,
and Mike Leone has been providing some more information on that large orange roughy that we saw. And he said that, they previously aged from out here on the Mid-Atlantic Ridge on orange roughy at 139 years old. And he says that he believes they start reproducing at 30 years of age. And that in one study, 95% of the stocks that were examined, that is the populations of the orange roughy that they were looking at, the breeding age was over 73 years old. So that is just
Sean, we can get moving 090 degrees.
Copy that. 090, 10 meters.
Yeah.
Copy. Coming up.
I mean, just imagine if, humans didn't start reproducing until they were in their 30s or 40s, and we're still reproducing or most of the individuals were reproducing in their 70s.
Having just turned 40, I'm not going to go there. But it is an interesting comment in terms of fishery sustainability. You know, if it takes a well-aged population of fish in order to maintain that stock.
We're just going to keep going up the hill here.
Les Watling notes that the ICES Council, that's the International Council for the Exploration of the Sea, has a postcard for their anniversary with a photo of an orange roughy on it, and the caption with it is something along the lines of 125 years old and still gorgeous. Little cup coral down there, that orange thing just next to the lasers, and one of these, I think, we'll have to find out from Tina Sacco. Yeah, can we get another view of these sticks that are in the lower right? We now have our expert having a close look, so she can give us an ID on this. Thank you. So again, it's interesting. These are not whips. These are colonies that are branching right at the base, and they seem to have 2 or 3 branches to it. It's definitely a black coral. I'm not sure if it's in the in the family Antipathidae or Aphanopathidae. There you can see a couple of other branches have broken off, so it looks like there's at least 4 branches, 2 that are still alive, 2 pointing towards us that are dead, and I can't tell if the one that's sort of moving off over the face of the rock to the right is part of this or not. But that's a really good view because you can see the underlying skeleton, then you can see how the tissue is draped over it. I even saw a couple of polyps that were retracting their tentacles there. But as we go up, you see all the others are extended. And I'm seeing even in the tentacles, you can see these little dots. Those are the nematocyst bundles, the so-called stinging cells of this cnidarian. And this is something that you see in the hexacorals. You can very clearly usually see the nematocyst bundles in the tentacles, whereas the octocorals don't have the same kinds of cnidae. They don't seem to have as many of the the nematocysts, and so you don't tend to see those clusters, those dots in the tentacles. Thank you. That's a really nice view, and hopefully, there, we already got an identification. Antipathes viminalis. So that would be in the family Antipathidae.
Thanks, video.
And Ashton was asking me about this the other day, the origin of this name. The Antipatheria, which is the order that includes all of these black corals, is Latin for against disease, and it goes back, I'm pretty sure, to the time of Aristotle, ancient Greeks, and their belief that the powder from the skeleton was useful in curing some diseases. There's been no scientific basis for that in studies more, in the last century. There's the shark again coming through. I don't know if it's the same individual that we've been looking at. And my best guess earlier was some kind of gulper shark.
Take it. Oh, he's changed his mind. He said gulper shark. I've never heard of that before.
Gulper shark, gulper eel.
Yep. Well, Mike Vecchione thinks it's a cat shark. Apresturus.
Watch where you're going, buddy. Swim right into a rock.
Okay.
I know. It's almost like there's a big ROV messing with it.
Might be a little bit of swing left Sirius, but ship stopped 1-2 minutes ago, so.
Copy.
Okay.
Looks like we're about
just one more comment about what I was saying about the Antipatheria. It gets a little bit confusing to the novice. So we have the order Antipatheria, the family Antipathidae, and the genus Antipathes. And one of the things you'll notice on each of those words is that although they have the same beginning, they all have different endings. And their endings, there's a
180 meters.
there's a formal vocabulary in taxonomy and systematics. So it's kind of a shorthand. So if somebody says to me Antipathidae, it ends in IDAE. I know they're talking about the family level, even if I don't know the family. And that would be true also of the order, superorder, and so on.
Okay.
Again, seeing lots of suspension feeders here, so these sessile organisms. Sessile means that the animals that you're seeing, and these are all animals, not plants, have attached themselves to the seafloor in various ways, some kind of cement typically that they have secreted, and they are growing up off of these raised rocks and boulders and pillows and so on, in order to get up into the water column so that they can get their feeding structures into where the current is moving
the steepest part right now.
a little bit more quickly to deliver more food.
I'd say this is about it. If we trust HiPAC, we should be starting to get out of it.
Okay.
But yeah.
Well, there are plenty of colonies here inside this crater.
Fernando, you want
And
10 more meters
sponges.
to the east?
Yeah.
If we could have... Sorry, pilot, if you're in a position, all the way, just going off the screen, lower right. Off the screen now.
It's the white branching.
Yep, that fan.
Partial zoom video?
Further right, just on the edge of the screen.
Oh, okay.
Yep.
White branching.
Hold on, video.
Thank you. So this colony, the texture of the colony looked a little bit different to me, so I wanted to just get a look at it. It's what the lasers just passed over. It's that left side. The polyps are a lot smaller, and I'm not sure if this is just something that's overgrowing, if this is a prinoid or a hydroid.
Okay, partial zoom video. Let's get the lasers on.
No, it's an octocoral.
All right. Lasers coming off. Then, rising. Hold on.
Oh, maybe this is the dendrobrachia again. Well, that would be cool.
Okay.
So this is something that we were seeing on one of the earlier dives. I don't remember if it was, I think it was dive 5. There was a lot of debate about what we were seeing. And some suggested dendrobrachia. I thought at the time that the skeleton was too thick for that. If we have any more zoom, that would be great. There's an interesting gastropod also down at the lower base there.
A little bit closer.
Thank you.
Okay, that's good. Video, let's come in.
Yeah, that's terrific. Great. Great.
Tilting up slightly.
So I wonder if all those white dots along the skeleton are meaningful. I'm seeing it both along the main axis and the side branches. And if, actually, so the view that I see of the branch that's kind of sticking out towards us going to the right, and it seems to have some raised bumps, almost looks like there's some grooves. And that's a hallmark of dendrobrachia. The skeleton, instead of being smooth and round, has ridges in it. So I think I agree with everybody who is saying this may be dendrobrachia. That's what it is. And one of the interesting things about that is, if I recall, when this genus was first described, it was described as a skeleton, and it was described as a black coral, because black corals have this sort of thorny skeleton, so the ridges were interpreted in that way. and only later was it transferred to the octocorals. Great. Thank you. Great view. So these last couple of dives where we've seen it, this is the first time I've ever seen this genus in situ. Yeah, Tina's agreeing with me. She's also seeing those longitudinal grooves in the skeleton underneath the tissue. And that's pretty distinctive for that genus.
Pilots, are you ready for a heading change? A-frame, 20 degrees starboard.
Copy.
Pilot?
Yeah, go ahead.
Upper right, there's a slender, unbranched whip just coming into view.
How far right?
It's coming towards the center now. It's almost directly in the center above the lasers now.
Above the lasers,
Yep.
the really tall one.
Yep.
Copy.
Thank you. So this is either going to be a primnoid whip or a sponge or a crinoid that's lost its head.
Let me know if I move too far, co-pilot.
Okay. Look like you have
I
the ability to get there.
suspect it's
Copy.
going to be...
Got a little bit of swing left in that last move.
No, I'm not sure. I think it's going to be the primnoid. I just noticed a Stauropathes, very small Stauropathes black coral. I'm just saying that for the folks on shore.
Okay, let's rotate to the base. All right. Let's come in video.
All right, good. So as we're zooming in on this, what you're going to see is that the polyps are sort of pointing down, and there's 3 or 4 of them all arising from the same general area, kind of like think of them as being oriented in a ring around that main axis. Looks like maybe it's 1, 2, 3, 4 or 5 actually in these whorls. And that's what sort of gives it from a distance this really smooth, sort of thick appearance to that whip. And so why I knew from a distance this wasn't going to be a bamboo coral. The other thing is, if you were able to see the polyps closely enough, you'd see a series of scale-like structures going up the body wall of each of those polyps. Thank you. That's a good view. It's interesting. What I think I'm seeing, and this is for the folks on shore who know the primnoids pretty well, is that the scale at the lower part of the polyp has a pronounced needle at the top of that scale. So that's kind of interesting. Yeah, good stuff.
Okay, when you're good, video.
Great view.
Pilots, we want to get you a move in.
Yeah.
Yeah, we're ready.
10 meters due east, still good?
Yeah.
Copy.
Okay, taking off. And then I'm gonna.
So we
center
continue
your
to move
camera.
up slope. We got on the bottom. Let's see. Wow, it's already been almost an hour and a half. That's amazing. Time just flies. We were at 1,235 meters on the. Just a quick zoom right above where the lasers would be. That orange bushy colony.
This one right here?
Yep, that's the one.
Okay.
That's going to be an Acanella. I already know what it is, but just so we can
Partial zone
document what it is. This is a
Hold right
bamboo
there.
coral, Acanella. Was very common when we were diving at our dive 5. Not too much shallower than where we are right now. And I've heard from our Azorian colleagues that
Slowly
it's very
approaching.
common in these depths around 1,000 meters up here. Thank you. That's terrific.
Okay.
So
ready.
yeah, we got
Let's
onto the
come
bottom
up.
at 1,235 meters inside this volcanic feature, and we're climbing
up.
up the wall towards the rim. And we are currently at 1,183 meters, and we think the top of the rim is at approximately 1,000. Actually, it was about 1104, I think. Is that right? Or 1110?
1100.
1100? Yeah, so we're about 80 meters below that rim. And it'll be very interesting to see how things change. Yeah, we'll definitely take a look at this large branching coral. Thank you. It'll be interesting to see. We're already seeing, I think, in my opinion, much more sessile organisms than we expected. But I expect the density to get even higher. So this is a pretty spectacular little rock here. This is going to be filled with different things. I see a black coral feather on the lower right at the top. I see two Chrysogorgia kind of bottle brush shaped colonies. There's a large globular sponge in there. There's the three bright white anthithelids on the left, but then there's this major colony that we're going in to look at. And we'll find out if this is Plexauridae, which would be the sort of closest to the characteristic shallow water sea fans, I think, that we see down here. Or is it a bamboo coral? Probably a plexaurid. But there's also another kind of coral that's kind of growing off towards us that's white from that mass. It's been here for a long time.
Copy.
Yeah,
Good
good view.
contrast.
So
Awesome.
this is, let's see.
Looks good, doesn't it?
Let me just hesitate a moment there and get a better view, because I was typing in the chat room, so I didn't see as we were coming in.
Go ahead.
I don't want to say that we're looking at one thing when it's another, because as I look at the sclerites here, I want to say this is a bamboo, but when we were coming in, I had convinced myself it was a plexaurid. And what I don't see are the little mounds or calyces that the plexaurid polyp should withdraw into.
What else are you looking for?
Well, ideally, if it's a bamboo, you should
right
see
there.
nodes through the tissue, but I don't. Oh, there you go. I think Ashton found one.
Oh, yeah. That's
Yeah.
right.
Yeah.
I'm an expert. Don't you know it?
Just left center. Let's keep doing what you're doing. Just go. Let's go down this colony.
Keep coming down. Copy.
It's yeah. There's another one. I think another node right there, or maybe there.
Right.
And then go to the right, please.
And remind me how those nodes guide your interpretation of what this is.
Only
Trying
the
to pass
bamboo
slowly.
corals have nodes. That's not entirely true.
Yep.
That's a a generalization. In the deep
Copy.
sea, there we go, perfect. Yeah, absolutely. And this is a nodal brancher. I can even see these two major lower branches are coming out from these black spots. So that black line that you see there, these are protein. And those are the nodes, and the white space in between is calcium carbonate. We call those the internodes. And so only in the deep sea here, this family, the Keratoisididae, has that kind of structure. The colony behind it, the lighter white, is another kind of bamboo coral. I think it's a different species. That one I'm confident is Jason
How are we
Isis.
doing
This
with
forward
that
one, I'm not
co-pilot?
confident what it is. So
I'm
that's
not sure.
fantastic.
I think we just started swinging. Maybe got a little
Great
left. It's a
stuff.
short move.
Great stuff.
Copy.
We got
That's
for
one
timing
that says J3 clade. I think the one in the background there is absolutely J3 clade, and the one in the foreground quite possibly as well. But very prominent spines, I noticed. All of these polyps are kind of curled up and they're hiding their tentacles. And when we were looking at the ones up at the top, that's what made me hesitate.
Right.
Oh, very cool. There's the Chrysogorgiid. There's our sponge, our hermit crab. Oh, actually, the yellow colony down low on the left, that's an Acanthogorgia. And we have anthithelids. So right here in this one small
yellow one
little
on the
rock.
left bottom?
Yeah, lower
Okay.
left, yep. We have 1, 2, 3, 4,
Copy.
5, at least 6 different octocoral species, plus the sponges and all the things living on it. And we have cup corals.
Ooh.
Yeah, so that yellow one back there, that's got an anemone on it. That's in the family Acanthogorgiidae, probably the genus Acanthogorgia. And so I'm thinking back to the dive. What did we call that dive? Dive 5? The one you named? Zenith. Thank you. Yeah, the one we named Zenith. We'd seen some Acanthogorgia there as well. We had some requests to get a collection. I've had a request to collect the Dendrobrachia. There's excitement about the bamboo coral. At least the bamboo coral, if we wanted it identified, it would need to be collected. We can't get all of these things. I'm just sort of relaying to you kind of the excitement and the desires of science on shore, because there's a lot of things that we see here that in order to actually put a name on, we'd have to have a collection. We can't collect them all, but I just want to share with the public the information that we're doing our best to put names on these things, and some of them we know better than others, but that's why we take the collections. And there's that Bathypathes, I think. We got the ID. Yeah, Bathypathes is probably pseudo-alternata as a species name. So that was a beautiful rock.
What did you say?
Wait,
what? No. After that.
Oh, that was a beautiful rock.
Yeah. All right, I just want to...
Didn't even know what I was saying.
I know.
I mean, you know why it was beautiful, in my opinion.
It was
covered in biology.
Okay. I was going to say that beautiful basaltic texture with a ferromanganese crust that just shouts, "Hey, look at me! I'm doing ocean chemistry and precipitating out minerals." But maybe not.
Is this
yeah, that's
partial
interesting.
zone?
Yeah,
just like that texture.
Yeah, it's weird. It almost looks transparent. Huh. I would almost want to call this a tunicate from a distance. I mean, the size of it should be a sponge, but the texture and transparency looks more like a tunicate.
Here, sorry. Okay, right there. If you have more zoom, let me actually... There you go. So you can zoom in.
Huh. Oh, I see. It is a tunicate. It's one, I think it's one very large solitary tunicate. And that would be the X-current opening that we're looking at. We're zooming on right now, upper left.
You ready for it?
You can see there's a hole there.
I think it's 10 meters.
And this would be the tunic itself. And the tunic sometimes can incorporate spicules and sand grains and stuff from the environment. And if we move up this to the right, all the way at the tip, there should be another opening, a much larger one, and that would be the in-current opening. And then inside here, there's a pharynx. This thing generates a current, pulls it through the pharynx. There's a basket, lots of mucus, removes the particulates, and then the outflow of water is sent through this this other opening so that it doesn't have to process all the water through its guts and can concentrate the
That's a
food.
10-meter move.
Yeah.
Very cool.
Copy.
I don't think I've ever seen a large solitary tunicate like this down here. And then just in the foreground, we notice, there's a Lophelia, that stony coral. I expect we're going to be seeing lots more of that today. So that's the first observation of that. Excellent. Thank you. Good eye pilot. That's really cool. Do we have the lasers on just to get a sense of how big that thing is?
Thanks for a reminder.
So the base is probably 7 to 8 centimeters across. Wow. I mean, if that is a solitary tunicate, it's huge.
We need those Elmo golds.
The what? Oh, yeah. On the rotator?
Copy.
Yeah, you like the golds better than the Zip Like You?
Second.
How smooth is Minisus?
Oh, it's great. I mean, look. Nice candy. He did midwater.
Yeah, that's perfect. That's what we need.
And it's fast, too. You like the gold ones, don't you?
I mean, it would be a lot better for the zones, definitely.
Is it easier to program the gold than the, like the Rosette one, that Zip Like You? Is that different? Or same command set?
We use the same program.
I think the golds are smoother, though.
They have like an extra mode or something, but...
Well, they have a velocity and position mode.
Yeah, something like that, yeah.
I don't think the Simplicuan has that.
Oh, great. So Les Watling says that that's the gumboot tunicate. It's also seen in the porcupine sea bite. The porcupine sea bite is off the west coast of Ireland, and there's a tremendous amount of deep sea work that's gone on there for several decades. It's a very important place in deep sea biology because of all the work that's been done by UK scientists. We know quite a lot about the seafloor there, the abyssal basin, and more recently, I think they've done a lot of studies on the canyons that cut into that bite.
Let me center it. Yep, there you go. Go ahead.
We have another fish here. Let's see, what are we looking at? The underside of something.
I thought those were bushy eyebrows for a second.
Can't tell. I don't think it's an Oreo.
Let me know if I'm moving too much to the side.
I'm just a pseudomorph.
You're good for now. You're right in the middle.
Copy.
Is this like a very pale roughy? This is like a gray roughy or a white roughy.
I think it's one of the rare upside down roughies.
Yep. And that's what the texture looks like. It's just lacks its pigment. Very interesting. Yeah, so Mike Vecchione also says roughy. Yeah, so maybe somebody can let me know. Is it common to see them so pallid?
Yeah.
Interesting. Yeah, I don't know if that means it's young or it's old.
Good.
And just before you leave and finish with the video, if you just put the lasers on it so we can get a sense of the size.
Copy. Let
Thank
me
you.
know when you're clear, video. Right.
Another one of these antipathies, black coral sticking out.
Copy.
So there's the lasers.
Okay.
So there are a couple of other species of roughy that are not so red, so I suppose there's a potential that that could be a different species. Yep, the boarfish up there.
Okay, then what do we have here?
Oh, so in the foreground there's a smaller rock in front of the rock pile that has a fan that's sort of pointing towards that rock pile. And I think that's the one that we're looking for to collect.
Copy.
So now, yeah, below the lasers.
So is the ship moving?
I think they just completed. Oh, great. Perfect.
Copy. All right.
Perfect.
All right. That's still too far out, right? Okay, so I guess from here, let's just take a look. Zoom in video. Hold on, let me just stop shaking so much.
Or if this is once it's
There you go.
brittle or if it's
Let's go in video.
rigid.
So watch lead.
Yeah, a little bit tighter. Now I'm not so sure this is the same one. Oh yeah, there's the ridges. Yep, that's the one.
This is the one?
Yep.
Okay, so let's come out video.
Thank you. Terrific.
Let me see. All right. That's a good distance right there, right?
Yeah.
Copy. Okay, center.
Some information in the chat room here from Ashley Maranzino says that fish also tend to change color depending if they are sleeping or if they're in poor health, which is kind of interesting. And Les Watling says that the New Zealand fish biologist said that roughy tend to dive to the substrate to avoid trawls. They think the behavior came from avoiding whale
Just
predation.
pushing out ahead. bring that set point back just a little bit. Should be good. Could probably push down a little harder. I was doing 40, 50, but.
Alright, that's 40 right there.
There you go.
Okay, so gauging auto lock. I mean J lock. Tilting down.
Could put this in a jar, clip and jar it, or bio box.
Okay. So watch lead.
Yes.
Let's come in video. How much of the specimen should I cut? How far up the main stem?
Let's say,
Or should I actually cut it
I would
up
just get
at the top or just one of the sides?
It's not very big. I'd get the whole thing.
I think it would be better if you cut that main axis. So what I'm looking at is if I go from the bottom on the right side, there's a long branch, then 1, 2, 3 small branches, and then 2 longer branches. Just get just above that longer branch if you can.
Come in video.
Almost in the center of your screen now.
That's full zoom. Okay, so in between those two in the middle and the one on top, going to the left, like just cut it right there.
Cut it right, yeah, exactly.
Like-
So you see that longer branch going off to the right? You can be just above that. And it's approximate, right? So we're talking approximately the top third. There's no specific
All right.
spot. Thank you.
I think once you get your jaws there, just probably get the whole thing.
And then
Hydraulics are out. I'm thinking,
let's try the starboard,
okay.
maybe outer.
Okay. Prepared.
Copy. I'm just going live. Okay. Let's zoom out on the main camera video, please. Are we zoomed out? Okay, there you go. I see the claw right there. Thank you.
So yeah, Nav, keep an eye on the Sirius camera in front of you. Thanks. Delta 17.
Okay, let's come in on main camera, the Zeus. A little bit more. That's good right there. And then I guess let's come in on mini Zeus, but I don't think I will be able to see much from there. Okay, that's good.
And pilot, given its size, it's okay if you go a little bit lower than what I said.
Copy. Grip force 4.
So I was on, yeah, we're good. I'd just get the whole thing. It's pretty small, Fernando.
Copy.
Just gotta get close to the rock without touching it.
Alright, then let's zoom out a little bit video. Right there.
Nice.
Watch lead 2, survey 1.
Go ahead, survey 1.
What are we calling this species?
Dendrobrachia.
Roger that.
Oh,
I have to twist it. There you go.
okay, let's come out.
It has a protein-based skeleton, so you see that
Grip lock on.
it doesn't snap like a calcium carbonate skeleton.
You want to get some video of it, or you want to just do it?
Sure. Okay, let's go.
So you can kind of see the ridges along the skeleton. That skeleton isn't smooth. It also has little spines on it, so you can see all the little bumps that are sort of poking out through the tissue itself. That's great. That's definitely Dendrobrachia. So we know the genus. We're not certain of the species.
All right, you can start pushing the drawer and slowly out. Takes a little while, huh?
I'm just being careful.
Yep. Yeah, I don't think you're gonna touch anything.
I think I'm touching.
Okay. Do you want me to?
Yeah.
Okay. Hold on 1 second. Oops. Okay. Lifting off.
Roger.
Okay. Have good bottom lock, solid. Gauging out of depth. Gauging out of Y. Auto heading. Oh. Oh. Okay. You can come out whenever you feel good.
Can you back me up a little bit on that rock? Or up. Go up off the ground a little bit.
Alright.
Yeah, there we go. Alright. I'm watching the jaws. Looks like you're above the moving parts. Alright, I'm all the way open.
Copy.
There you go. You said you wanted starboard.
Yep.
Roger.
Alright. Alright. Oh, let's try it right here.
I think the jaws are contacting the box there. Just careful. Yep, see that? There you go. That's probably good. Should fall in.
Copy.
Not a lot of room to open the jaws there.
Yep.
Yeah, I'd probably come out of the box a little bit and drop it in. Otherwise the jaws get hung up in there.
Alright.
You get contacted on that side of the box. Get the wrist right a little bit. There you go. And then up a little bit. Yeah, I'll drop it from there.
How come it's not? Okay. There you go. Now it should open.
There it goes. There it goes. It's falling. There's still part of it on the jaw, but most of it's in. Once you get above the box further, yeah.
Okay.
What happened?
That's okay. You get most of it.
All right. Let's just.
So, very nicely done, pilot. So the pilot was had to guide this sample that is feather-like, and so it can drift in the current into the bio box. We did successfully get the major piece into the bio box, but now there's another little part that's kind of stuck on the claw, so we may be seeing that for a while. But we got the major part of the collection. Nicely done.
Wow, it's stuck there.
You can swing the arm out and just open and close, maybe. Get away from everything and just. There you go.
Okay. Thank you.
So, pilot, I'm not sure about how to do this, but right in front of that coral, I've been asked to collect the one that was sitting right in front of it.
Okay. We can come back down.
Okay. I wasn't sure if it would help to tell you that before you stowed the arm. And what I'm going to propose, once you look at it, is that we just take a very small piece and suction it. So clip it and then suction it off the claw. And that.
Okay, so I'm gonna rotate down,
Yeah, if you bring up jar camera on that monitor.
and then I'm not sure exactly where I
I think
was.
It's
that's
a
just
little
to
bit
draw
back
one.
where you are now. It's just under the sled, I think.
It's right here. Small white branching. See it?
Just a little bit
It
farther.
was on the same rock that you just collected from, I believe.
Yeah, on that rock right there. The one that just came into view, the white one.
Yeah. He's thinking clip it. I got jar 1 prepped.
Copy.
Roger. Suction light. Enabled. I think it's perfect. As long as you put me in a position where I can draw out just like a little bit to get the suction nozzle in view, and we're good.
Okay. That's good right there. Okay, I'm still live. And then how far up do you want this cut?
So we only need whatever, just a small piece. So whatever it is that you are able to cut and then be able to suction.
Okay.
It doesn't need to be large. Essentially what we're doing is to get a piece to verify, is this the same species that we see at 500 meters depth? So we don't need much.
Copy. Alright, let's come in video on the main one. Alright, right there, that's good.
No, we need it. Thanks.
Okay, so
I'm keeping an eye on it.
let's come in with Minisus to see how much. Make sure that I actually get something.
Sorry, yeah.
Okay, so
Apparently you can also come straight down on it, maybe.
right there, I think right there I'm gonna get a few parts, right?
Not a lot, but a little. I'd try coming down on it.
Almost creepy rock right there.
Yep. There you go. Definitely cut some branches off. Nice.
Oh, I didn't get it with the.
Yeah, twist it. It might break. Or it might pull off the. You make it the whole thing.
No, come on.
Yeah, it's not a calcium carbonate skeleton, so twisting it is tough. There we go.
Looks like it slid out.
Okay, there you go. Let's come on video.
Tough bugger.
Alright, do you want me to
Alright, so just
take
watch your
off.
elbow here.
Copy.
And then I think I can draw it a little bit right here. I'm just going to draw it out a little bit, okay?
Alright, go ahead.
Easy out. That can work for you?
Yeah, I think so.
How much section do you want? There's 800.
I think that should be fine. Let me just. Okay, so I'm gonna engage, rotate the jaw. There you go. And then actually get it a little bit more straight. Okay. Let's go down.
Got 1,000.
Copy.
These is a pretty good view too, if you want to zoom that one.
Right there, that should be right in front of it. I can see it.
Okay.
Alright, open it slowly.
That was brilliant. Thank you. Really nicely done.
Alright, perfect.
Into the canister. All right, gang, we got our two collections there that I saw were of the most interest in the chat room. Thank you to Pilot. That was excellent. And when you're ready, we can resume upwards.
Okay, I think I got all the chunks in. I'm going to retract the drawer.
Copy. Okay, arms looking good right there.
Looks great. Indexing to
Hydraulics are off on the arm.
Sure, 2.
So while Pilot is getting ready to move us on again, I'm going to share with you some information that's been provided in the chat room by Ken Sulak, our fish biologist extraordinaire, recently retired from the U.S. Geological Survey. So he says that the macrurid, that rat tail or the grenadier that we saw earlier that had the long pointed snout,
That's good.
he says that's Trachyrhynchus scabrous, which is a common North Atlantic species, which is interesting. I had said at the time it's not one I recognized, and most of the work that I've done is in the western North Atlantic. And one of the things about the whole ASPIRE program is the connectivity between the western and the eastern Atlantic, and there's a lot of suggestion that here in the mid-Atlantic ridge we tend to see more of the eastern Atlantic fauna than the western Atlantic. So it's really interesting to hear that from Ken, that that's a species that's common in North Atlantic. But Ken had never seen that imaged underwater before, so that was really exciting that we got that. He also says that Trachyrhynchus, the genus that houses that particular fish that we saw, is the genus that gave rat tails the name grenadiers because of that long pointed snout that resembles the tall pointed hat of the British grenadier soldiers. Those are the soldiers with rifles that are able to lob grenades. So very cool.
Fracture Zone video.
Seeing Okeanos here, the feather-like black coral that's right in the center. That's Bathypathes we identified earlier as pseudo-alternata for the species. The sideroid urchin, a couple of sponges. Ken also says that the small spiny eel that we saw earlier is Polyacanthinotis meriti, and that name meriti is named for Nigel Merritt, and it was Ken Sulak who described that fish and provided the name, and it was in recognition of Nigel's contributions to the knowledge of Northeast Atlantic deepwater fishes. It's terrific information, Ken. Thanks for sharing that. Bird's nest sponge, Pheronema carpenteri, there on the left, and I believe this looks like a bamboo coral skeleton that is partly overgrown. It's not branching. And then we have some of the antipathes, these much thinner skeletons. They're kind of yellow. They look like whips, but they're actually branched at the base. This fuzzy one that just came into view upper right, that's new.
Copy.
So let's have a look at that. So I wonder, this is either going to be Thuarella, which is a kind of primnoid, or it's some skeleton that's been completely overgrown by hydroids. So get a closer look here to verify. And getting these closer looks is what helps us with other tools that we use, like drop cameras, where we don't have the luxury of getting close-ups and really clear images. And so having this ROV here with this fantastic cameras and lighting allows us to do a lot of ground truthing that we can then take back to other data sources and identify things. Sure is looking like a Thuarella to me as we zoom in.
Let's try to get it from here. Let me turn on the lasers. Get them down.
There's the lasers. So maybe 4 to 5 centimeters across at the holdfast.
Okay, ready? Let me know.
I'm deploying some of your lower string arms by that.
Go ahead.
That's okay.
Great, yeah, there we go. So this is definitely an octocoral. This is in the family Primnoidae, which is, I have said a couple of times, this is a very diverse common deep sea family. You see it has all these side branches with really small polyps, and you can see the polyps have this sort of rounded disc at the top. And this is in the genus Thuarella. It looks a lot to me like the species Thuarella grassify that we've sampled in canyons of the western North Atlantic. I don't know for certain that it's that species, but it's definitely that genus. And it would be interesting if it is, because I've just told you this information about how the fauna on the Mid-Atlantic Ridge resemble more the eastern Atlantic, but Thuarella grassify was originally described from the western Atlantic. Great stuff. And that's a crinoid, which is a kind of echinoderm related to the sea stars and the brittle stars that likes to clamber up on top of other organisms to get those arms, those suspension feeding arms, and its 2 feet up into the water column.
Copy.
Very nice view. Science is happy when video's happy.
Copy.
Another cup coral there in the lower right. Several Acanella.
All right, let me know.
This looks like another large fan in the upper left. It's a little hard to tell because we're edge on to the colony.
Thank you.
But maybe as we go up. So pilot, as we're moving forward here, you're going to see that on the left side is a very large fan. See if you can go past it. I don't know on what side, but just turn to see the wide plane of it.
Okay, let me approach it. Okay, so let's get it from this side.
And if our viewers turn into camera 2, you'll see the camera view from Sirius, which is looking down on the ROV-D2. So you get a good sense of how the pilot has to maneuver the ROV in order for us to get these really nice images. And he also has to be very cognizant of where he is within that frame and where the tether is.
Okay, fine.
It's a very complicated dance, all these vehicles together.
No, I wonder if I should get another one in.
So I'm kind of thinking this is a corallium. This may be our first corallium of the cruise, or at least in the family Coralliidae. This would be one of the precious pink corals. I mean, there's a possibility it's a Paragorgiid, but it kind of looks to me like a corallium.
All right. me settle down a little bit, video.
Can you get in any closer?
Yeah, but I would have to like.
I'm getting confirmation from
I mean, like if I get
the
closer,
ECC
I'm gonna
in
be
Horta.
looking down even more.
Manuela
So
Ramos
that's
is
okay.
typing that.
Yeah.
This is probably
So
Hemicorallium
it's a little bit
tricolor.
closer.
That's great. So this will be the first
I don't
example.
know
It's
how much
also
light you have right there,
conveniently
but,
has a sea
I'm
spider
gonna
on
tilt
it.
down.
There's been a lot of talk about nightmares on the ship with
All right, go ahead with the zoom.
sea spiders and caterpillars. So here we have a twofer, our first precious coral, Hemicorallium bicolor, and I imagine the bicolor is for the
Hold right
red
there,
polyps
hold right
against the white axis of the
Over
skeleton.
there.
And then we have this pycnogonid sea spider that's on it. And I've mentioned before that the pycnogonid sea spiders are specialist predators of polyps. And so if we're lucky,
You can come in.
or if the coral's unlucky, we'll see the proboscis actually in one of these polyps. And can't quite see it because it's on the far side of us, so it's hidden from some legs. This phylum, the Pycnogonida, used to be called the phylum Pantopoda, which translates as all leg, and I think you can see why it gets this name, all leg. This is probably the family Colossendidae.
And of course, I come back right to a giant sea spider.
I did say something about nightmare. I hoped you'd heard me in the dry lab and would not come
I
in.
don't see anything
So.
here.
Yeah.
Great view. And so we've just added to our diversity of corals.
Not sure why it's vibrating so much, Rowan. Not
This is a
applying that
known
much, but
species.
uptown.
Maybe there's something caught one of the.
Hemicorallium, I mean. Note from shore pilots. Thanks to the pilot and camera person working together nicely to capture close-ups of even moving fishes today. Credit due to the skill shown.
Actually, probably the moving fishes are much easier than the...
Yeah, you don't have to stay stable. Maybe if it's possible, before you leave, if you could turn to the left, I see a snail on the main axis.
All right, video.
And there's a
Are we
second
going
one
to
that's down on the rock, so let's look at this one that's a little
Or do you
closer.
want to go to the snail?
The snail?
Copy.
I think he's talking that one to the left edge.
Is it the one up here or the one on the rock?
No, the one up here, I think
on the
I think the
coral.
one up here is easier to see because I
the
can see
texture.
its antennae. Or, excuse me, its sensory tentacles. We don't call them antennae.
Oh.
Yeah, we want to be careful not to
All right, so
hit this thing.
top or bottom?
Left. The textured one. White
Yeah, the
ball
top one on the...
Okay.
on
Yep.
that branch.
Thank you.
Centered right there. Go.
All right.
There we go.
Okay, hold on 1 second, video. All right, there you go. Let's come in.
There we go. So you can see the snail, the texture of the shell, and those two structures that are sticking out the front. Those are the sensory tentacles of this snail. I don't see a siphon on this snail, which suggests to me that perhaps it's simply grazing over the surface rather than predatory.
That is not because of auto heading, right?
Somebody else maybe
I think it's
who has
overt,
more
like a,
experience with the mollusca can identify
chunks
that.
in
That's
the
great, pilot. Thank you. We're done.
balance of the prop.
Copy.
Go full screen.
So I want to give
coral.
some more context to people who are tuning into our streaming
lasers.
video and sort of pity the poor pilot. So I'm sitting
Right,
at
so
an
tilt it.
audio
Hold on,
system.
lost
I have a
rotator.
headset on, and
There
in
it
front
is.
of me is a panel that has 1, 2, 3, 4, 5, 6, 7, 8,
Right,
9,
gonna
10,
turn off
11,
lasers.
12, 13 different switches,
Tilt
all
this
of which
slightly
will
up.
connect to somebody different, either in the control room or on the ship, who can be
Let me
speaking
know
to
when
me.
you're clear.
The pilot
Video
always has
clear?
to be listening to
Copy.
the navigator and the co-pilot, but he's also listening to video, and he's listening to me, and he may have the Okeanos line on, so if any other scientist tunes in, we'll be listening. So there are a multitude of voices in his ear, so sometimes when he's asking for a little clarity on what to look at, it's difficult because I may be saying something different than somebody else. So it's a really challenging, as it is for the scientists in the back row here, because we're also listening to all these different voices as we also read the chat room. So it's exciting.
Got a close-up looking on stream 3, looking over the pilot's shoulder.
Yeah, you might be able to see both all the different monitors that he has to tend with, as well as all the audio, and then of course the controls, the computer controls for this vessel. This is, yeah. Oh, you're wrong. It's a bamboo coral, and the thing is, I think I know what it is. I suspect this is going to be an internodal brancher, and it's probably, this could be Ecnomysis. If it is an internodal brancher, and if the polyps kind of look like little mounded volcanoes, and they do.
I'm going to give you part.
And
I'm just trying to rotate up.
see. Ashton's here. He's the expert in finding the nodes.
All right. Oh, look at that, streamers.
Oh, it's full of the ctenophores again. Thank you, Caitlin. So Caitlin just, I don't know why I didn't see this, but there's all these.
What was that
I'm so focused on the
side,
nodes, I didn't notice all these streaming tentacles. If we could focus on the background right. There we go. Beautiful. So all of those lines that you see are coming from these little tiny ctenophores, and I can see some there. Perfect. We're seeing some at the top. They're flattened. So these are benthic ctenophores, platytended ctenophores, and they have these tentacle sheaths. So there's little raised nubbins on them, and they have these long retractable tentacles. Oh, beautiful. On cue, retracted some of the tentacles into the tentacle sheaths. Awesome. And they're basically fishing. And those tentacles are studded with structures called coloblasts. And these are sticky secretions, so when they catch something like some kind of small zooplankton, they can glue it. And so you see them all along the bamboo coral here. So just like a fisherman, you know, you want to position yourself in a good place to throw out your line. All these guys have either swum or crawled up this bamboo coral and are fishing higher up in the water column. What a fantastic view. So we collected, I don't even remember what dive, day 1 or day 3. A bamboo coral, and it was covered in these ctenophores. I hadn't realized, but more than 2 dozen. So here we see the same thing, but we didn't get as good view as this. Wow, that's fantastic. Thank you. What a great view.
Let's go directly left. You can see
meanwhile, the bamboo coral, I think,
a little bit down,
maybe an Ecnomysis. Actually, Les thinks it may be in the J1 clade. That's interesting.
This one or the
Ecnomysis is in the D2 clade. And what's going on right here?
There's something else.
There is. I think it's a little anemone, maybe, that's growing there. Yeah, it's a little ring anemone, and it's withdrawn its tentacles and sort of contracted the bell. There's 2 more on that branch to the left.
Oh, wow. I'm pretty far away.
And we'll get a zoom.
So many ctenophores. Great stuff. I'm wondering if this may be the same bamboo that we collected previously, because that one also had really long spines. You can see all these polyps that are kind of closed up. They look like little volcanoes, but it's like they have these very long fingernails extending from them. Those are the needle-like sclerites. And the piece that we collected, those sclerites are so long that they're almost poking out of the bag that they're in now. And I definitely got one stuck in my finger the other day, and that's how sharp they are.
You're full wide.
They're not called needle-like sclerites for nothing.
Yep. All right.
You want to run lasers across it?
They're on, but I don't even see them. Okay, there they are. Let me just go to the base.
There you go. Video's clear.
Copy. All right.
Looks like it's getting a little steeper, Fernando.
Lifting off. What was that, co-pilot?
Looks like it's getting a little steeper.
Copy.
Judging by this, but we'll see what it looks like once we get up there.
All right. So you have me at 45 degrees. That's good. Let's keep going.
Roger.
Rotator. It's level right now.
We see that thing on the left there?
The spira. Some sort of gorgia. Spira gorgia? Vertigorgia? What are those called?
A Ritogorgia.
A Rita?
Something like that?
Vertigorgia?
Maybe.
Arita gorgeous.
Yeah, beautiful Arita Gorgia. Arita Gorgia. Yeah.
It's like a truffula tree.
Everyone is laughing at my pronunciation. And so we see it again perched on the kind of a large outcrop of exposed rock. Now, a lot of the rock that you're seeing around us is basaltic talus.
Scott's not on.
And then it's kind of intermixed with a lot of this. You can see it kind of on the lower portion. It's slightly sedimented. It looks like a bunch of partially concreted shells and marine debris.
Maybe a couple percent, maybe 10%.
I think it's too far away.
Yeah, it's way up anyway.
Oh, okay.
Okay, let me turn to the left.
Yeah, video, can you just shed more light on it?
You got it.
Oh, you're just looking way up at it?
Yeah.
That's what's going on.
And you see that beautiful spiral structure of that center branch. And then hanging off of that main branch are all these subsequent branches with all these individual polyps.
Finally, we got an interview.
Shooting up high.
What is that?
And can you zoom in on the lithopathies,
I'm
Barbie?
on OKX.
You are?
What is that?
It's
Watch
just
sleep?
super soft.
Oh.
Oh, that's why. Got it. It's on my end. Rolling.
Watch sleep?
Watch these. I don't know if you're depressing pilot, but you've gone off of OKEX.
OKEX check. Okay. I didn't click off of it, so goes to the machine. Here's our fearless leader, Scott. But if you could zoom in, please, a little bit more.
Get those polyps.
Still that vibration.
Fernando, why are you vibrating?
Oh, there you go. I had a little bit too much thrust, but
I know. I think there's something stuck in one of the verts. You know what I mean? When you float off and you have time, just go clean them out.
No, I think this time it was me.
The corals in the family Chrysogorgiidae are among the most beautiful and kind of mesmerizing. And Iridogorgia is one of those. And I'm seeing now, as I've just come back in, these polyps look to be somewhat elongated and swollen along the base. And that's another good indication that many of the polyps are in a reproductive state.
Let's go
octocorals in general,
rotating up.
Circular opening.
what we're seeing, these little dots that are in the center of this 8 tentacles, that's the mouth, and the mouth leads into the gastrovascular space, which takes up most of the polyp body that you see here. And that space is subdivided by 8 sheets. Think of infoldings of the wall of that cavity, each one between one of the pairs of tentacles. And on the bases of those infolded sheets is where the gametes are produced, so the sperm and the eggs. And so when there's a lot of feeding and energy is available to produce those sperm and eggs, the bases of these polyps can get swollen prior to them releasing those sperm and eggs for fertilization.
I started off with the spiral and I went away from it.
Hold on, I'm not sure why I'm moving.
So this is interesting. Les Watling is who's done a lot of work with Chrysogorgiids in general and Iridogorgia in particular, is noting that the axis has these verrucae, so these little sort of bumps associated with it. And that may be a characteristic of the species Iridogorgia portolezi. And I think that may have been the original species described in this genus and is known
Is there
from
any
the
special
eastern
extra
North
shots you want,
Atlantic.
Caitlin, while
In the western North Atlantic,
this?
we have Iridogorgia splendens and Iridogorgia magnispiralis
Yeah.
at the least. And those are two
That's my
species that Les
standard
Watling
repertoire.
described from collections that we made on the New England seamounts. And so those seem to be more characteristic of the Western Atlantic
Just
and
gonna get the
the
full
Pacific. And this is
of
a
the top.
theme that we've seen
Copy.
in a few
Holding that
species
shot.
where we have similarities in the biogeography and the distribution of species in the Western Atlantic extending into the Pacific, and then some differentiation from the
Just
Eastern
get
Atlantic.
that top headroom and sort of pan down as I zoom out till we get the base full screen.
Go ahead.
And when you're finished with the beautiful image here, if you get the lasers on the top of that colony, that would be helpful.
Look at the lasers after the shot.
Yeah, I don't think, Les, that this is a magnispiralis. The coils don't seem wide enough.
The bottom is definitely too hot for the shot, but okay. If you want to tilt up the
Pilot, you
lasers.
think you're comfortable enough to just take a snipping and suction and slurp, excuse me?
Oh, this one?
Yep. Just all we need is one little branch, but I mean, I hate to
Zooming in for lasers.
disturb it.
I mean, how can I put
Yeah.
lasers on it?
Oh, the lasers. That's tough.
Yeah, it's hard to tell where they are.
Yeah.
All right,
They're
let's
there.
come
They're
out.
there. There's the lasers. That's good.
All right, there you go. Let's come out video.
Full wide.
Okay. All right.
Hey, Fernando.
Hey, Chris.
Hey, Chris. Welcome to the chair. You can adjust HD2
That might be
for
too
sample
close,
mode.
right? Navigator.
Yeah.
Is that too close for sampling? Like for the arm? Like it's way up here. Like you can barely see it in the manips.
Chris, can you adjust HD2 down for sample mode?
Okay.
Yeah, Roland. Let me,
So I'll just
let me
put
fix that.
Geological and rotate to the right. Okay. Yeah. Okay. All right. Okay, so that's a good distance. All right, sounds good. Okay, so that's 45 down. Let's get Geological engaged right there. Nope, I didn't engage it. It's 50. A little bit down. Okay.
I think you'd be able to get out there pretty easily. It's not too far out in front of your toes.
Yep.
Pilot, so I have some updated feedback from shore.
Go ahead.
We actually need one or two of the coils, and so you can't slurp it. It would have to be a clip and then into the bio box.
Copy.
And just as a warning, this would be a floater.
All right, so we'll go on port. On the port side, I think both are empty. We have
So port inner is empty. You have to go port inner or starboard outer for bio boxes.
copy. I'll try to go port inner.
Yeah, I believe we have the
Come
sponge
back
and
off,
port
Fernando,
outer.
back up. Draw out. And then stow it.
Yeah.
Yeah, you probably have to do something like that.
Yep.
Okay. So we have hydraulics.
Hydraulics are up.
Copy. Arms coming live. All right.
I'll follow you over.
Yeah. Oh, that's a rhodellid siphonophore. Siphonophore. Kind of photobombing this. One of the dandelion animals.
actually. Alright, like I said, it will look good like that.
Any zooms you want for position?
Couldn't. Yeah. Keep going in. That's good right there. And then mini Suze, maybe you can zoom in a little bit, but I don't think I'll get much from it. Okay. And then watch lead. The cutters are at the bottom.
Yeah, I see how difficult it is to get up in there, so that's fine.
So I mean, I can. So you said you needed 2 of the.
Yep. Yeah. I mean, if you can get up, so there's 2 corals, that'd be great because then we could leave the feeding part still there, and it's very flexible, so they should flex through your.
Oh, so you mean like all the way?
Just keep going up. Keep
there?
going. Yep.
Okay.
Yep. Exactly. See if you can get above one of those spirals so that we leave enough there.
All right. So just one of the spirals.
Yep.
I'm gonna take.
I think if you do what you did before, come down underneath and then pull up through it.
All right.
Exactly. Easy for me to say.
Yeah, I think that's as far as I can go with.
Yeah, that's good right there.
No, I don't think it's gonna cut it, right, Sean? Or you think?
I think you have to go. I think it's just outside the blade.
Yeah. Okay, right there, I think.
Right there looks good.
Yeah, right. Okay.
Yep.
Gonna close.
Nice.
Nice.
All right, let's come out video. That was most of it, though. Okay. So, do you want to zoom in on it, video?
No, we could just put it in the box.
Copy.
We got exception.
I personally don't need the zoom. We know it is.
Okay. So, coming out of Geological.
Do you want to port enter, Fernando?
Yes.
Understood.
Okay. Rise a little bit more. Okay. So that's good. You should be clear right there to open. Let me just engage out of that. And auto XY.
All right. Let you settle.
Yep.
You happy with that?
Yeah, I think we're settled.
Okay. Sure,
So if
coming
you
up.
wanna go ahead. Okay.
I can rotate you down on your main if you want, Fernando.
No, I'm not looking at the main. I'm looking at miniatures right now. Okay, so all the way down.
Remember, science said it's a floater.
Yep. Alright, so I'm gonna try to, well, it's pretty far down right now.
Yeah, you're probably touching the bottom of the box, I bet.
Okay, let's lift up a little bit. Then I'm gonna try to open the jaws right there. Slowly. Okay. Okay, let's going. Okay, there you go.
Nice work.
All right, close it.
Scratching.
All right.
Nice work, ROV team.
Okay, that's good for the arm. Stow position, Sean. Okay, hydraulics are off on the arm.
Hydraulics secure, valve pack secure.
What was that again? It was.
Alright, out of depth, next fly, heading.
Bars.
Hey, pilot. Ship's all stopped, so you're off stationary.
Good copy. Oh.
Yeah, pilot. We're just moving up slope, roughly east. Should be through the steepest part of this ascent, but still good slope ahead. So, yeah, just doing short moves.
How far are we to waypoint 2, Sean?
Hey, video, are we full wide?
Yes, sir, you are.
Waypoint 2
Got it.
is
100.
150 meters away. Yeah.
I'm looking at you 3-0, Lars.
Copy. Should be stationary at this time.
Yeah, you want to get a move.
Yeah, I'd say so.
It's about 10 meters, moving 090. Does that work?
Sure.
I'll put it in.
Seems like there's a little current pushing me on up this feature. Very slightly.
I should feel that pretty quickly, I would think.
Yeah, only at 1,000 meters today.
Yep.
So I'm reading some of the chat from the shore-based scientists, and Les Watling is thanking us for grabbing that Iridogorgia. He said he could solve a mystery of more than 100 years. And as background, what is left of the type of the Iridogorgia is just a few polyps. And that was collected back in the 1800s, and the branches and the axes that was collecting during the 1800s has all been lost. So probably a really great need for that sample. And so we are currently about 127, 130 kilometers north of the Azore Islands, north of the northwestern island, the island of Corvo, where we're about 12 kilometers, I think that's correct, 12 kilometers to 20 kilometers west of the main axis of the Mid-Atlantic Ridge. We're on the upper footwall of a normal fault. And we're diving on a submarine volcano. We're on the inside of that volcanic crater, and we're heading the inside east wall. And when we looked at the 3D bathymetry, or the shape of the sea floor that we mapped with our sonar system, on this volcano, and that's likely because it was we get, and so when these volcanoes form, especially through submarine extrusive volcanism that form these pillows that we saw nice examples of yesterday, they tend to form very characteristic slopes based off of how thick the magma is and how quickly it cools. And those slopes tend to be 15 to maybe 23 degrees. And so that was pretty indicative of all the slopes on this volcano, on the north, on the south, and on the west, and on the inner surface, except on this face that we're diving on now, the interior eastern face, which was a much higher slope feature, going up to 35 degrees, and we hypothesized that was because it was full of rubble. And that's very indicative of what we're seeing right now. We see lots of rubble blocks, lots of angular fragments of rocks that have likely tumbled down. And if I had to wax and wane geophilosophically about why this side is rubbly, it's because if you think of this volcano as almost a little donut on the seafloor, and there's a normal fault that formed near the mid-axis ridge about 2.2 million years ago. When this fault occurred, it bisected, it kind of cut in half, or actually cut about 20% of the eastern side of this volcano off, and that section dropped down, and that was a lot of stress on this volcano. It was it would have been associated with a series of earthquakes, and I'm guessing that this side, just being on the other side of that fault, also collapsed inward to a certain extent. And so you see all this rubble of that volcano.
If I was a guessing man, I think they'd want to look at the white sponge right center, and the purple stalk that is just off-screen top next to them.
And most of this rubble is also coated with a thin, probably maybe a few millimeter at most, maybe 1, 2, 3 millimeter coating of ferromanganese, iron manganese. and that gives it also that dark color. But it's kind of difficult to see sometimes because the natural volcanic rock, the basalt, is also very dark in color. So color can always be a little disorienting. And then in between all of this rubble, you see the lighter color material, which is some sediment, but you also see some very, for lack of a better word, gnarly, kind of gnarled, rough, highly fractured and kind of porous material that's very dark, and that's probably concretions of
you're coming in on the
calcareous organisms, pteropod shells that have kind of consolidated into almost a carbonate rind.
Copy.
And so the goal is to get to the top of this volcanic feature,
Coming up a little.
investigating the biology of
the
the abundance and the diversity. And then as we get to the top,
It's a nice floating shot, Lars. Is that pretty neutral for you?
Sorry, say that again, Chris.
I just asked if that was pretty neutral for you.
Neutral.
Z.
Kind of thinking a little bit. Trying to practice floating.
Understood.
So you want to look at that stalk.
and so one of the things that's interesting
Just
to
left of
me
center.
as someone who studies volcanoes is how this feature formed. It's not very similar to the elongate composite volcanoes that we see on the ridge in the axial valley of the Mid-Atlantic Ridge, which tend to be much more constructional and have a very well-defined peak at the top. This has a clear depression in the middle, and there's a few ways that can form. One of the ways is that it was actually a constructional peak, a high point, and then there was magma beneath it in the crust that was removed, and that the upper portion then collapsed into that void space. Similarly, that magma could have been moving through beneath the volcano, and it actually actively eroded the upper crust,
Slowly
which it then
go to
collapsed
the
into.
top of it?
And so those are two types of processes that I don't expect to occur in this type of setting. This volcano is a really isolated feature in 3 dimensions. It doesn't have what we call rift zones. It doesn't have elongate zones of magma transport that we are more associate with places like the Azores or Hawaii. This is a much more isolated, single-sourced feature. And so another way that these depressions in the top can form is actually through explosive submarine eruptions. A really large explosive submarine eruption would also form this kind of this crevice. And so one of the things that we're looking for is something called hyloclastite. And hyloclastite is really just a fancy word for a mixture of fragmented rock, fragmented basaltic glass that we looked at the other day. And it tends to weather to a reddish product because it's iron and magnesium rich, and so that oxidizes out. And then you get these little fragments of black basaltic glass in it sometimes. And so we'll look for those. I haven't seen any yet, but I know that there's some scientists who are interested in getting samples of that if we do see it.
Straight right, just a little bitty stalk.
Hopefully when we get to the top of this, we'll also get some more primary fresh-looking pillow basalts like we saw yesterday. And then if we get one of those, we can get a really nice sample, and we can do the same type of glass analysis that we would do on these hyloclastites, but on these pillows. The only issue is then we don't know whether it was associated with an explosive eruption. These pillow eruptions tend to be much more what we call effusive, so not explosive, just kind of flowing. And right now it's lonely in the back row because two of our people are out to lunch. So our science data manager is out to lunch, our co-lead, Scott Francis, out to lunch. I'm sitting here with my video editor, Anna, whose sister has a birthday today, who turns 30. Do you want to get online and give a shout out?
Let's see if it'll work.
Fogs up. 50%.
There you go. Yeah, that does help.
Happy birthday, Lara. I hope you're having fun on Orcas Island. And your cup is currently in Sirius right now being shrunken. So I hope you enjoy it when you see it. And it has the merman riding the seahorse on it.
Yeah, so it's always nice to say hi to family from the ship.
All right, let's keep making our way up.
And so one of the classic science activities that we always do, especially for outreach, and they make great little examples of the pressure that we have here. So we're at a depth of about 1,150 meters below sea level. And so that's equivalent to about 115 atmospheres. So we're under 115 times the pressure that you feel on the surface above water. So, it's an incredible amount of pressure. And one of the ways we demonstrate that for outreach is styrofoam cups. So we take styrofoam cups, or cup of noodles are a popular one. Styrofoam mannequin heads, I've seen those before. We can put them in kind of a mesh bag, we can put them on the ROV, and we can take them down. And all of that pressure shrinks and compresses all of the pore space in the styrofoam out. And you can shrink a styrofoam coffee cup down to about 10% of its original size.
150 meters,
and that's a really great tool to demonstrate to school children the amount of pressure.
Probably 40 meters altitude.
Or 30-year-olds. But anyways, it's a good example of leveraging the asset we have for something that's not directly science-related, but really helps communicate the environment that we work in.
It's pretty dead on right now. Yeah, excellent.
You tell a kid, oh, it's 110 times the atmospheric pressure. They don't necessarily, identify with that. But you show them a cup of noodles cup that looks like it was made for, a raccoon, they get that sense of pressure.
15 degrees slope, looks like. So you get to waypoint 2.
And so this crust slope we're looking at on the right, that's all above us, and then kind of dips down to the lower right, and it's kind of overhung a little bit. This to me does not look volcanic. That looks like a concretion of probably calcareous organisms and shells that's then been encrusted with manganese and then slightly sedimented. And then you can see the difference between what the laser's on right now, which is that nice basalt crust, and then what's on to the left and the right of it, which is much more gnarly. Yeah, so there's a great example.
I could bring your port lower and board if you want.
Or CH.
Stow
If you
that a
don't
little
mind,
bit.
yeah.
Understood.
Thank you.
And
You
you
can
can
do
see
that on
how
starboard as well. I think I might.
The texture on the left almost looks like it's knobbly. It looks like maybe rough and some rounded stones stuck together. So that's what I expect to be, that's kind of that
dark, like the
that
overhang?
calcium carbonate.
The cave behind it.
Yeah, that's that calcium carbonate material from organisms living in the water column or on the surface, or corals on the seafloor, pteropod shells, foraminifera, little phytoplankton that build their shells or their tests.
Sorry, what did you say, video?
I was going
out of calcium carbonate.
Okay.
And as they die, they land on the seafloor, and they slowly get consolidated together. The calcite, the calcium carbonate in those shells and tests and corals gets dissolved and remineralized, and it formally forms a limestone-like structure. Depending on the age of it and how well it's concreted, we might add a term to it. We might call it fossiliferous limestone, a limestone made from fossils.
Okay, continuing on.
It's a cool shot of Sirius from the port rail cam.
Oh, yeah.
And so I will stop talking about words like fossiliferous limestone as Scott Franz walks back in. Yeah.
You guys want another one?
Yeah, I think we should probably get another one in. You okay with that, Lars?
We got all sorts of fun words. Agglutinate.
Something bigger.
That's a good one.
We can go with 15.
We use that one with the forearms.
Oh, yeah.
Agglutinated forearms, for sure. That's a good one.
15.
All right, enjoy your lunch.
Thank you, thank you.
If Sean was in here, I'm assuming it would be, but
I was watching it during my lunch, Roland.
Okay, so
unbranched.
It was not open.
Whip, purple, yep.
thank you.
Thanks.
Yeah, I thought it was very unusual because we don't usually see them that tall and unbranched with that family.
Okay, you guys ready
So
for that move?
I
15
wonder
meters?
if it wasn't
Ready. East.
a plexaurid after all. Sure looked like it, but yeah, I was eating my lunch in my room, so I
like one of
couldn't
those
take my
lava
eyes off
pillows.
Look at the
footage.
brittle
What did they
spot.
say here in the chat room?
On
Anybody
the
have
right
any guesses
swirly
to what
thing?
that was?
Copy.
Okay, I am reading through it now. She identified a crinoid. Les Watling noted he didn't know of any whip plexaurids either.
Go ahead and start coming in.
Oh, this is interesting. This looks like one of the basket stars, actually, that you would normally find on a coral, with its arms all wrapped around a coral. Maybe not. It's got quite a lot of spination, so maybe that's not what it is. The arms aren't quite as smooth. But it's a brittle star. Not exactly certain what kind it is. It's certainly not one that I've seen lying around on rocks all that much. I wonder if it's one of the fishing type, one of the ones that clamber up on corals and then hang their arms, their very long arms, off onto the water column to capture stuff that's drifting
Getting a
by.
couple, unexpected.
Yeah, you just dove right there as soon as you creeped in.
It's quite an interesting sponge to lift also.
What'd you say about being heavy, Lars? You think you are a little heavy?
Yeah, just the Z-bias, it seems quite a lot for what we're doing.
Understood. I will note that.
Just
Maybe
getting
weight
some
log.
weird oscillations, but I think it's just the current around this little feature. It's kind of weird.
Can we go tight inside the hole of the sponge?
This thing
if you're
tucked
stable?
down in the rock, that may be another compound or colonial tunicate. We were imaging one earlier today that at first I thought was a sponge, but then when we got in close, the texture suggested that it was a tunicate. And I believe that may be the same thing here. Yeah, I think I'm seeing more closely. You notice that there's a whole bunch of cases, and this is actually a very gelatinous one. So each individual zooid kind of has its own little house or case or capsule and has its own filtering apparatus. And then they're all joined together in this common matrix. And tunicates can form a whole lot of different kind of matrices, but in this case, the overall one seems rather gelatinous.
Coming down a little. Holding.
Yeah, if you could focus on the upper side a little bit, you might be able to see more clearly the atrial openings.
Copy. Upper side of the
of the surface of this whole gelatinous thing. It's like a whole series of holes.
Coming up.
Each of those is a zooid, and they should have a couple of different openings. One for the current to be drawn in. Actually, that's nice. As I'm looking on the left side there, you notice there's striations through it. That's probably the internal pharynx that it's using to filter once that water flow is brought in. It's got a mucus lining that pharynx. Very nice. You can see how it's kind of attached to the rock down there with its gooey strands. So we collected one the other day that I described as kind of like a big parasol.
I think your sonar
and then when we got it up on deck, it did have some main stalks, but it also had a lot of other attachment points, and it could be something similar to this. It was also very gelatinous.
Unless I'm just saying right over top of it.
Video's clear.
Yeah, I got
copy pilot square.
Right.
It's a nice shot, Lars.
Yeah, that's Watling notes that he's also never seen an ophiuroid or brittle star with its arms kind of curled up on the rock like that.
Understood.
It's kind of interesting.
Lars, I'm going to let you go here just for a second.
Yeah, I wonder if it got dislodged from some coral.
Might be.
I see now Chris is in there.
I think you should come up, Lars.
Copy. Coming up.
Yeah, Chris, does this look like one of the ones that, you know, I would call the fishing ophiuroid to sort of climb up on corals and hang their arms off into the water column?
Copy.
You suggested the family Ophiacanthidae, and I think that's because of the spiny nature of the arms.
Coming up with you.
Understood. See some of these yellow whip-like structures are the black corals. We imaged some of those before, and I think that we might be passing over our first Leopathes, unless you saw one while I was at lunch. That's the orange-red one in the upper left. Oh, actually, so in this, let's see, to the left of the lasers, but below center of screen coming off the front of that rock, there's a pinkish poof. Now it's in the almost lower left corner.
Stand by. Maybe let's get a 5-meter move back if possible.
Oh, and I see a rock pen up there.
Right on that little spot.
Over to the left.
Yep.
Almost directly to the left of the lasers now. What I'm trying to see is if this is a juvenile Metallogorgia or if it's something else.
Affirmative, yeah.
Okay.
Their left laser just crossed the stock. Interesting, yeah, so this is a Metallogorgia. So this is an earlier phase. It's not the adult phase. What's odd is that it has such a long stalk above it with no branches at the top. That doesn't make much sense. But here you see the branches in the center, and there's your ophiuroid, all curled up, your Ophiocretus oedipus, and the typical sets of branches. So our experience, anyway, with this species is that as it ages and gets taller, the branches kind of move up the stalk. I mean, these branches aren't literally moving, but new branches would grow up above, and these lower ones would be shed. What I find a bit of a mystery is why there's so much of the stalk above it and no branches up there yet. That's a little bit odd. Thank you.
Keep the stalk or the poof in the bottom of the screen.
So did you actually have to tweak anything?
I changed frequencies. I'm not really sure.
Yeah, there you go. Oh, it looks like there's maybe two polyps up there. If we can get a zoom at the very tip of that stalk.
You know.
Yeah, it wasn't picking anything up.
It was pretty tight.
There we go. So I wonder if those polyps are sort of the precursors for the branches that we would predict are going to arise. So this one's in its teenage years, I guess.
To do some more
very cool.
Reading.
Very cool.
Yeah.
And then just behind it, that yellow thing on the right, that's a bryozoan.
That's a good catch.
There's a yellow bryozoan colony.
Yeah, no.
Very nice. So pilots, I noted when we were coming in here, there is a rock pen, and we finally have to collect one of these rock pens. I keep seeing them. We haven't collected one in the Central Atlantic yet. This is the farthest east that they're known. So I think it's small enough that it can just be slurped.
Understood.
Okay, where was that?
It's on the seafloor to the right. It's just on the shell hash. I'll point it out when we see it.
Copilot, I think we'll keep it at this frequency for now.
Yeah.
Actually, it wasn't on the. I think it was like just before that sort of stair stepping. How come I don't see it now?
Yep.
Maybe if you look up just a little bit more to the right. Oh, there it is. It's coming in the top of the screen. I think that sort of reddish brown.
Also a sea star.
Maybe we could put the lasers on so I can get a sense of the size, but I think it's small enough that the whole colony can be suctioned.
Scott, on the left, you can also see there's a sea star climbing up that black coral. This is in your private channel.
Yeah, thank you. So video is just pointing out to me that the Leopathes black coral had a sea star on the branches, and I can see that it is on the part of the branch that has no tissue. So I wonder if that sea star is feeding on the black coral.
You want to do a quick snap on this rock pen?
Wait, where did it? Oh, yeah, that's it. Yeah, it's sort of drifting towards us now. It's bending over. You can see nicely here how it's suction cup to the rock. What do you think, pilot? Does it look small enough to you to just go into the hose?
I believe we can make it work.
All right. It should dislodge very easily once you start to suction it.
Copy that.
Can we center it a little bit more for one more zoom?
Sure. Stand by. Holding. It's opening.
And let's turn on the extra lights.
Understood. Fog's coming up.
Let's take it up another 10%.
75.
Let's give it more back scatter than light for clear.
Yeah, I think it's a little far away. Light's clear.
Yep.
All right, so this is a confirmed collection.
This is a confirmed collection copy.
Copy that. Ship is stationary. We move back 5 meters.
Yeah, I think I'm stopped, so we should be good.
This might be a tricky spot to sneak into.
I'm gonna.
The jog's coming up.
Copy that. So ideally, I'd want to set up on the right-hand side, but that's
You don't have DVL.
Copy.
Upashnik Anguli is offering some observations in the chat room. She says that we are seeing two morphological variants of rock pen in the North Atlantic. The one we saw a few days back had a curved axis, but the one we saw earlier today, and this one had a straight axis and a more rounded distribution of polyps.
So
I think she means that the polyps are all around that main axis instead of coming off on just one side.
I'll keep an eye on you. Looks like you're still slowly drifting down.
Okay, if you want to bring the door out.
Door's coming out.
So we'll just grab the nozzle, pull it out a little, and then I'll go ahead and...
You want all the way?
Fire. That should be good there.
Okay.
Okay, it looks like we can... Ramps coming on.
Looks like you're clear to the right, so that drift is totally fine, pilot.
Good copy, thank you. If you could come out just a little bit more.
Coming out just a hair. Just tell me when.
That's good.
I'll stop.
Yeah, it looks like you're coming in.
Okay, if you want to go ahead and bring the door in.
Sure, coming in.
Thank you.
Fully in.
Okay. I think I drifted over to starboard a little. It's back to port, if I remember.
Yeah, I think it's the upper left of the screen, just underneath that yellow filament.
Copy.
I can give you a better view of Mars.
Yeah, nice.
To the left of the big rock.
Left of Big Rock.
Right in front of your, yeah, just keep going left. That rock right next to your claw, it's straight left of
It's
that.
just underneath the yellow filament to the left.
Copy. Oh, I see it now. Okay. Arms coming live.
I'll check your tape. You still got room?
Copy. Stand by.
Reset.
Did I lose the grip on that?
Yeah, looks like it slid out.
Copy. I don't see the... Okay, there we go. We might be good to bring the door out here. Just nice and careful. You have that one rock on the
Understood.
port side.
I'll give it a go. Stand by.
You could try to wing in too, and then extend the drawer.
Understood.
Yep. Help.
Okay.
I'm not sure why you lost.
I'm not quite sure either. I grip force at 8.
Yeah, when you're out that far, it's just more tension. That's the only thing I can think.
Copy.
Coming in slow.
Understood.
I was taking notes when we decided on the subject. What are you going for?
There's a little rock pen out there.
I see it. Okay.
Yeah, I can barely see it.
All right.
It's that red thing.
The perfect combination.
All right, Lars, coming out with the drawer.
Good copy.
I think as long as you get it partially out, I think I would go for it. You know, you don't have to extend all the way.
Yep.
Nice.
You think that's good enough? I'm afraid to bump you there.
Yeah, just do it really slow. We'll see if I start losing my toe.
Coming in really slow. Still coming out.
Yeah, I can get it there.
Okay. We'll stop.
Copy, thank you.
I'm worried that's a little too far from there, but I think just grab it here and see what you can do.
You worried the subject is a little too far?
Yeah.
Copy. Actually, that's the extent of my
I can come out just a hair more if you want.
Just a hair if you don't mind.
Yep, stand by. I'm going to bring the camera back up.
Yep.
All right, coming out easy.
Okay.
Should be good, right?
All right, we'll hold that.
I think before you take that out, look at your starboard rail cam, and you're kind of at an angle back.
Yeah.
You know.
Yeah, I'm trying to get the nozzle to lean down into it.
Okay.
If possible. All right. Okay. Go ahead and bring the door back in, please.
Coming back in easy. All right, you got the black tape there.
Copy.
Can you see the subject from Mini Zeus? See how far away it is?
Yeah, stand by one.
You want me to try and slide closer?
That's kind of what I was thinking, but it's up to you. I can't really see.
Not gonna lie, I can't see it at all.
Is right there.
Oh, yeah.
Yeah, you can try it from there, yeah. Seems like you have the reach, I just don't. Yeah, it's out there a little ways.
You got view on tape. Yellow tape. You got plenty of room, Lars.
Copy.
Yellow's not even in view.
Yeah, I need to get closer.
It's too far out, yeah.
Good. Stay by one.
Coming back.
I think for things like that, you need to grab it square too, because if you grab it at that angle, it doesn't have as much teeth on it.
Right. Let's come out. We'll bring if you want to bring that drawer, sorry, the wing all the way back out.
Understood.
We'll grab it again.
Wing and outboard.
Surprised with grip force 8.
Yeah. I mean, you may as well go up to 9.
Yeah. All right, go ahead and bring the drawer all the way out.
Okay, drawer's coming out.
Good.
Nice and square. Looks good.
Okay, go ahead and bring the drawer in, please.
I'm good with that.
Stand by. Okay, go ahead and bring it in.
Drawer coming in.
Alright. Let's find this again. Get this thing in and then move on.
Yep, you got time.
Yeah, it's a tough one. Having to do all manual pretty
Just
much.
below you now.
Copy, thank you. Alright, I see it.
Temp probe is pointing right at it.
Yeah, we've got eyes on the video. Thank you.
Still got auto depth. Yeah.
Yeah, thank you.
Set point. Messing with you.
Yep.
There you go.
Nice.
Okay.
Ah, you can put it in a little forward.
Yeah.
That's still a good spot right there, I think.
Yeah, you're just right in front of it.
Okay. Coming on. Good zoom in on main HD, please.
I'm going to rotate you up. You're good.
Yes, please. Just a little.
Coming up just a hair.
Thank you. And then zoom in on Mini as well.
Got the cam up. Looks like you're on jar 2.
Jar 2. Confirm. That's open, right?
If you want to.
Yep.
Yep. Gotcha, Lars. Sorry.
We get the jar light on.
Okay. I think I'm ready.
All right. Suction coming on at your mark.
Go ahead.
Suction on.
Co-pilot, can we get the jar light on?
Stand by 1.
ROV suction versus rock pen suction. ROV
Nice.
suction wins. And there it is in the canister.
Yeah,
Looks great.
Suction.
Full wide on the main Zeus and Mini. We lose the home.
We
No,
did.
That's all right.
I hit the home by accident.
Okay, it's
That's
offset.
all right.
That's fine. All right. Come out of joy, up and back.
It's not a swimmer, so we'll handle that later.
Yep. Understood.
All right, sorry about that watch lead. Shaking off a few cobwebs. All right. You want to start coming out with the drawer?
Not sure what you're apologizing to me for, but that was a great collection.
Yeah, I agree with watch lead. I wouldn't have changed anything that's I love that kind of sample, totally under control the whole time. Made good decisions. There's no rushing. We got the sample.
So I believe that's the. Well, we don't actually know what species that is.
Stop.
There's only one species known from the North Atlantic. Happens to be Calibelemnon francii. That might be the same, but this is going to be the farthest east that we know of any.
The left one was sealed the last time he brought it out. I don't know why it didn't that time.
Pilot, as you stow, just for awareness, it's been made very clear to me that we would really like to look at the black coral that was over on the left, so before you make any attempt to move away from the area.
Good copy. Will do.
Thank you.
That's right.
Hydraulics are secured.
Hydraulics off. Not packed off.
Thanks.
Sirius is still where we left it.
Okay, heading back over. Should be able to find it.
Yeah, we haven't gone anywhere.
So, Chris, I have the one in the first jar as also not a swimmer.
Understood.
So I think you just home it on your own at your own leisure.
So it's that thing, I think, that's coming into view right now below the toe.
Copy that.
And in the center of it, there's a sea star apparently feeding. So I think I have two different scientists who give me input. One wants to see the sea star, and I think the other wants to see just the coral itself.
Copy.
Thank you.
So we'll need to sort of rotate a little bit right in order to see that sea star.
Oh, that's interesting. Les Watling is noting, he's paying attention to the salinity and the sensor data that we're getting. And he says the salinity here is pretty high, so there must be some influence of Mediterranean overflow water. And the temperature is over 6 degrees, which is kind of interesting at this depth. And so a couple of times, I think, Ashton, we've talked about water masses and mixing of water masses. And oceanographers can identify different, I'll call them parcels of water. The temperature and the salinity dictate the density of the water. And if the density is different enough, those water masses don't mix. And so you can actually track to see where did that water come from. So Mediterranean overflow water is water that is coming out of the Mediterranean. It's very highly saline because the temperature there is so high, you get a lot of evaporation, you concentrate the salt. And as it moves out into the North Atlantic, it has a higher density because of that salt content, and so it sinks, and then it continues to spread. So Les is suggesting we're seeing some of the influence of that water all the way out here at the Mid-Atlantic Ridge.
Confirm that's jar
Thanks,
1.
Les. That was interesting.
Oh, yep.
Okay, coming to 3.
Nice.
Vipassana Ganguly, whose dissertation is studying rock pens, says that those were a few tense minutes. It rather felt like a penalty shootout of a good football match. But she's got a happy face on now. Oh, that's no sea star. Those are barnacles. Sorry, Chris. You can stand down. But we'll see what the interest is in the black coral itself. Still, you do see some tissue that has been removed from this section of the black coral, and that's specifically where the barnacles are growing. And so I think what's happening there is you have the skeleton exposed, and so when the barnacle larvae are swimming by, it's not deterred by whatever chemicals that the black coral is emitting through its tissue, or these guys produce a lot of mucus, so that mucus probably contains some biochemicals that are noxious. And so the bare skeleton then becomes a good settling location for the barnacles. And here you can see barnacles of many different ages based on their size. All the way on the right there on that branch, you see a very small white one. That's great. Thank you.
Maybe pan left and show the coral if they need more interest in it. Science, is there more you wanted to see on the coral?
No, we're good. Thank you.
Copy that. Pilot's clear.
Video's clear.
So this doesn't help us now, but I just realized I think if we use the Titan, it's out so much farther, that would have helped us like see if that port box would have hit that rock a little better.
Yeah.
But super helpful now for me to tell you after we did it.
It's good to know for next time.
I'll just put it right there for next time. We'll be ready to go.
I think we kind of, me included, obviously, forget about that camera. It's nice for that kind of thing, you know.
So it only helps if we go to port. I'll remember.
And for the tape, too, like if you wanted to use Mini Zeus, like zoomed in or something.
Yeah. No, I didn't even think about it.
Neither did I.
I think the colony over there on the left is Dendrobrachia. We collected one of those earlier in the dive. Kind of excited about that. Now we've been seeing a number of them. So that'll be interesting from the perspective of we have a collection of something that we can consider a dominant member of the fauna, as well as ascertaining exactly what species it is.
Glad folks have picked up on it.
Oh, pilot, if we can still zoom on the sponge we were just passing over. Sorry, we just got a note from a sponge biologist to have a closer look.
Go ahead and start coming in a video. Anything in particular, watch lead, or?
I'm not sure. I'll have to be guided by shore here.
Copy that.
Partial.
Bivalves in the center of it. Top.
Copy.
We can see, I think that's the attachment point to the rock is actually quite long. It's not one little point, but is actually that whole curved part near the top, I believe.
Slight tilt up, feel the screen.
Yeah, this is Joanna Xavier in the chat room. She'll be your biology lead on the next expedition. She says she's a bit baffled
Chris,
by the
when
sponge.
you take over, just be mindful as you come into the future.
Yep.
A little bit closer, you get a
If it's okay with folks, I'd like to try to move up the slope a little bit. We really would like to get up to the rim here. And let's see, we're still at least 40 meters below the lip of that rim.
Good copy.
Chris, did you have to fight high back this morning and move the waypoint 3 back down?
It crashed, so I had to close survey. I was unresponsive, and then everything was not saved.
Understood, but the plan still is go south, right?
Correct, yes. Sure is.
Lars, I was trying to figure out what was going on with the current when you were stationary, and I still don't know. I think the rail cams are pretty good for if it's not being spun up by thrusters, for seeing like general movement, and the tether's, you know, a really good indicator too. But I couldn't figure out why you were drifting port so much. So there might just be a weird like boundary layer in this caldera kind of thing.
ROV, do you have an estimate to the next waypoint, given our current moves and speeds, assuming no sample stops?
Time estimate?
Yeah.
Yeah, let me get that for you.
Thank you.
Looked like a colony of the Ecnomysis bamboo coral there that just went off the left side of the screen. I see some fish, sponges, black corals, whip-like black corals, the Stichopathes. I think I see a Parantipathes, a sort of bottle brush one. That's at the top right of the screen.
Oh, and I don't think that jar
Maybe the first one I'm noticing
I
today.
fired up. I don't think that brightness goes anywhere. So There's where it is all the way
Joanna
off.
says that big white
There's
sponge
where it
we
is all
passed
the way
over is
up.
probably the genus Calyx, C-A-L-Y-X.
It gives it color when you kick
Well,
the
actually, we
lights
have
on.
25 minutes if we do a 0.2 move consistently, which I don't think has happened yet in 8 dives.
Yeah. And how many moves is it?
I'm sorry?
How many moves?
Are you guys clipping that? Kid?
Well, it depends, but we would probably do moves of 15 or 20 meters. So 7 maybe.
Okay.
8.
Okay. So if we do 8 consistent moves, we got about a half hour.
8 consistent
Correct.
high-speed moves is what I heard, faster than we normally go.
Well, not
This would
faster
be-
than the normal bear, just faster than the observant bear.
Sure.
This would be a better push.
This is a fantastic scene that we're coming up to here, so we certainly don't want to go at point 2 right this moment.
Okay.
I don't necessarily want to zoom in on everything. I just want to set the stage for some of the abundance at the top of this feature that we're seeing. It's quite nice.
Like a shell.
2 tubes, llama tubes, I
Interesting.
guess. You want to snap and see it as you go by?
Yeah, sure, if you want.
Blue view looks pretty cool right now.
Oh, I got you. Yeah.
Yeah, unfortunately for our colleague at the University of Lisbon who requested possible hyaloclastite deposit outcrops and samples of, I think if they were present, which I haven't seen any evidence of yet, I think they're just going to be too heavily encrusted to really identify. Or
you're full wide.
Oh, yeah, you like the little rock with a hole in it?
Yeah.
Yeah.
It's like a mini version of what we're in.
Looks like the Death Star.
Yeah, it does.
Some say it is the Death Star.
Yeah.
You know what I bet's inside that rock?
What?
More rock.
It's a good bet.
Okay. I can see pretty far
Another
out from it. Looks like we're at
Metallogorgia,
the top of that.
several Acanella, a few Lophelia colonies, and note the way these are all arranged on the top of these rocks in front of us, but on that flatter slope behind it, you don't see nearly as much. It has to do with the sort of raised topography getting into the water flow, and then the topography itself, the ruggedness of the seafloor, creates its own currents and turbulence, so that's a good spot to be for feeding.
Now, given I haven't seen any
I think you
evidence
can
of those
see
explosive
visually
products, I
up
think
and
what we
over
will try and
that little stuff
hope
right.
to grab eventually is at least one
So we
geologic
have
sample
the
that's
big
kind of
fish
like
in
the
the
ones
background.
we grabbed yesterday. So coming from a nice
Going to a
pulled out
pillar crop, and hopefully they'll have enough unweathered glass for him to do some.
Look just below it, though, pilot, on the sediment surface.
Copy.
I think that's another fish.
You want to get a 10?
No, is that a fish or is that a rock? I guess that's a rock. All right, rock
It's a
with
fish.
a sponge. It's a rock disguised as a fish.
Looks like it's got an eye and a fin.
gill, a
Yeah, I
porculum,
think I'm
and an
up over
eye.
the top of this. We can do
No,
another 10.
look at me instead.
Yeah.
I'm a real fish.
Yeah.
10 meters. You want to just do 100?
Yeah.
Okay.
I think start cheating towards the south.
Okay. On the way.
Good copy.
I'm going to come around just a little bit to starboard, Lars. My heading. Yeah, I think a 110 lines me up.
Pretty well with that.
Yeah.
It's 12, almost 25 meters out. I think it might be that. It's only, well, yeah, that might line up.
15 meters. Yeah, maybe.
Just a reminder on shore, whoever it is that just dialed
You
in
wanna get your
is
lowers
not
back
muted
up?
on the phone.
Yeah, if you
Thank
don't
you.
mind, that'd be great. Thank you.
Lars coming out.
Sorry, Scott, it's us from the control center in Oslo, and this Skype call always comes with the microphone on, and we keep forgetting, sorry.
No problem, understood.
Chris, do you care if I briefly switch
Thank
your sonar back to the other frequency?
Looks like another Thuarella,
I'll change it
the
right back.
Primnoid colony we saw earlier that kind of looks like a bottle brush or a test tube cleaner
lowers
from a
it back out,
distance
Lars.
going off the rock there. And several Okeanos colonies. These are the bamboo corals that are kind of golden orange in color. Have a
Scott,
bright white
with those
main axis that you can see peeking out, just attaching to the substrate.
Scott, what is it with those
Getting a
orange
good
ones that
look out
like
into the
bowling
distance
pins?
here. Right now we're about 1.2 meters above the bottom, and our actual depth is 1,135 meters.
I got you the 2
Scott,
5 bars.
what's with those orange ones that look like bowling pins?
I can come down a hair.
Bowling
a little bit more.
pins.
That's all right.
See them around there.
Look good.
The one with the white at the base. Seen like 4 or 5 of them.
So, for example, the one that's just going towards the lower right corner?
Yeah.
Yeah. Yeah, maybe, pilot, if you're in a position, we can get a close-up view. The one that's coming into the center,
sure,
down low, looks like it's bulbous at the bottom and then gets thin and then wide at the top. And there's, I'm guessing, I wonder if that's part of its skeleton. I'm wondering if this is something that's grown, tipped over, and grown again, but we'll see when we zoom in on it here. So this is one of the Okeanos bamboo corals. Not sure if it's growing on an old dead skeleton. Oh, I see. That's a hydrocoral just in front of it. So you have the Okeanos, which is one of the few octocorals that has the ability to grow in sediments, separate from all the sea pens, which of course have the inflated peduncle. This doesn't have an inflated peduncle. Instead, it's got a hard skeleton that grows as kind of like root-like structures into the sediment. This one had either a shrimp or a galathid crab in there, but that bright white thing in front is a hydrocoral, stylasterine hydrocoral, possibly the genus Crypthelia. All right, good view. And I think for the Crypthelia, it has to be on something hard, so there must be, that's either a thin veneer of sediment right there, or there's some rocks or pebbles or cobbles just below the sediment that the larva attach to.
We could probably get another 10-meter move in if we'd like.
10 meters, you guys seeing some flat bottom there?
Quite a bit.
I guess that would make sense on the average since we kind of came up that cliff.
Joanna has said something interesting here about Foraminima, so that's the bird's nest sponge, and there's one of them coming into view, the top center of the screen. You see that it's quite white, and if you look at its base, you see that it's kind of grayish in color, and it's growing on the sediment, and she notes that it's interesting to see them here on both soft and hard substrate. For a long time, this kind of sponge was thought to occur only on on soft substrate, excuse me. It was thought to occur only on soft substrate. So it's interesting to see it on both hard substrate and soft substrate. And that gives it the ability to live in more habitats.
Well. Chris, it's coming from port to starboard.
Understood.
Pilot, as you're passing by, the two stick-like corals in the center, either one of them, they seem to have a little branch coming off of them.
Copy that.
Just want to get a close look at that. So the question that we have to face, note that one that's on the right that has a whole lot of branches. So the question is, are the two that are in the center, are these just younger versions of that much larger one? And you could go further back in time and imagine that, well, it probably grew as a whip for a very long period of time. And so it's very challenging for us to identify whips from a distance because they can take on a lot of different morphologies if they only start to branch later in life. And I'm not sure if my student Mary Durie is watching this, but she's doing her master's thesis on a coral that looks a lot like that one on the right and trying to understand exactly this process. How do you differentiate in all of the video that we're taking something that has a growth form like that when it's at an earlier stage of its growth?
Go ahead and start coming in.
Good view of that bird's nest sponge growing on this rock. And that's what Joanna was pointing out. They used to be thought to be just on the soft sediments. So one clue that we can use is what's the density of the sclerites, what's the density of the polyps, what is the general texture of the polyps, what do the sclerites look like. Here, this is very convenient. The tentacles are curled over the mouth, and so you can see these intertentacular needles are very long. And trying to see what's going on in the body wall, and I'm having a little bit of problem, and I can't see through the tissue as well. So that's telling me that there's probably lots of minute scale-like sclerites that are reflecting the light and not making it transparent. So that's a good view. Let's just pan over to the one on the left. It's a little further behind, and see if the sclerites look basically the same. To me, from a distance, they look thinner and taller.
Okay, you get a little closer for more light.
And on that one, it's a bit more transparent. We might actually be able to see if the branch is coming out from a node or an internode.
Good copy.
And it looks to be coming out from an internode. I don't actually see any nodes. There's got to be nodes in there somewhere. Les Watling thinks they have the look of the F1 clade. Unless, is that one that has the very long internodes?
Okay, go ahead, start coming in.
Oh, and Asako's pointing out that there's also in the background an Iridogorgia, probably similar to the one we collected before.
Max,
Are we in anywhere we can take a quick, easy sample? Rock sample?
I mean, it's probably about done.
Yes, we should be able to grab something.
All right, you know, we keep passing over kind of this this crust that's really gnarly looking.
Copy. Once we're done with this, we can go ahead and pull out and find you one.
And this doesn't have to be in place. It's not like that basalt sample we took the other day. I just want to kind of baseline with that carbonate. So, you know, there's kind of a pile of it, middle screen right of the long coral.
Could we see if there's a pile of it over here?
Yeah. And
just look to the top right.
Anywhere where there's a nice grabbable fragment of that really gnarly stuff.
Yeah, if there's, because I want to look, the third thing I want to do now is look on this coral.
Yep.
Just to compare it to those two.
Yeah, so there's a piece of it right in front of us.
Yeah, great.
Yeah.
And we can get the rock first.
Yeah.
And it doesn't matter if it's just sitting on the bottom?
No, for this stuff, I'm kind of just more interested in what the surface is. So, you know, that one that's kind of center screen right now is perfect.
Understood. And do you want me to do a grip test on it, or just get it in the box?
Let's just get it in the box. It might actually be not too hard, so maybe a gentle touch.
Copy. And then you wanted to look at the coral, so maybe I can set up for...
Yeah, the same sort of thing that we were doing, but it can be after the rock. There's no hurry.
Understood.
Whichever works best for you.
After the biogenic rock.
Oh yeah, you think that's biogenic?
I don't think that's basalt. I think that's probably a portion of calcareous concretions, and maybe. Yeah, it'll be interesting to see. It'll be interesting to see when we get aboard if it looks similar to what else I'm seeing on crusted everywhere else. So I'm hoping that's it.
You could spin me
so I'm actually hoping that once we bring it aboard, it's not a basalt.
Right. So on first glance or first test, would the weight of it tell you anything?
Open?
It might be a little heavier, yeah. Basalt's a little denser than what we might expect, the density of a limestone. You know, actually, you know, when they'll grab it, I'll have them bring it up, and we'll take a nice, good look at it, probably before we sample it, just to make sure it is what I think it is. And that'll give me some baseline idea of what's kind of forming this gnarly surface that's covering everything else.
In that case, I'm not going to ask you now to give me a name for it. I'll let you pick it up first, and then give me a name.
Let me look at it before I...
Okay, if you could bring in the port rock box, please, or port wing.
Understood.
Arm's coming on, arm's coming out.
It's in.
Copy that. sight is all off today.
Yeah, let's give it a nice, let's get a nice look at it. See if we can see what any of that surface texture is.
Copy that.
Turn on the lights.
Yeah, please.
Fogs at 50.
Yeah, that's a good level.
I see some texture I like.
Yeah, can we zoom in a little bit? Try and see if we can identify any of the individual structure. Yeah, that's good. Okay, yeah, that is perfect. That's exactly what I'm looking for.
Copy that.
All right, let's.
So is that, you think that?
Let's bag it and tag it.
Pyroclastic sort of thing, or do you think
No,
that's
no,
the
I think
consolidated?
I'm going to call that coated carbonate concretion.
I'm going to call that sponge garden.
There you go.
At least 4 sponges in there.
Well, yeah, I wanted.
Unless that's a tunicate, you know, it's got that interesting margin. I'll bet that's the tunicate.
Do you see that really hairy thing on the left that just rotated out of view?
That was a sponge.
Can we just like knock that off?
I'll knock it off in the lab.
There you go. Well, you know, I didn't think you'd be busy enough, so I figured I'd grab the rock with the most stuff on it.
Thank you.
The ball at the bottom.
Very nice.
No, and so you know, this is not something I would go after because I'm interested in kind of igneous volcanic products. But you know, I'll be honest. You know, my understanding or my knowledge base of kind of deep sea sedimentary processes is kind of weak. So, you know, to me, superficially, this looks like, you know, a bunch of conglomerated and partially remineralized carbonate material.
So one really cool thing about this view we're getting of the sponge right now, I think it's just attached by just a little bit of a stalk, which is kind of cool.
Look at those little dots.
Yeah, I don't know what those are.
Those little red eggs.
Yeah.
Yeah, that's perfect. We'll box it, and there's a little pteropod shell, right? Yep. And so that's what I think it is. I think it's a lot of pteropod shells that have been kind of remineralized and concreted together, and then coated in ferromanganese.
Oh, cool. So, yeah, Joanna thinks that might be a juvenile rosellid sponge. So, you know, the other day we were talking about those things on the stalk with the bulb at the top.
Yeah.
What do they look like? Well, that might be what it looks like when it's young.
Now, do you think we could grow that?
Oh, you know what? No, never mind.
All right, so we all good? Logged, sampled?
Let's in. No, I got to push this button.
So let's
going. What do you want to call it?
Nice. Coated
Samplars.
Carbonate.
My depth perception are just all off today.
Concretion.
Oh, wait. I was at like the very extent of
You can't think of any more C's to add to that?
No. And I think it's coated in a ferromanganese, but I don't want to necessarily assume off the bat. So it's coated in something.
Understood.
Okay.
Thank you, ROV. That was awesome.
Of course.
And I can probably guess that's going to be a smelly rock.
Because of the biology or because of the concretionist?
I think a little bit of both.
Okay.
Yeah.
It's got some great sponges, and I really think that was a tunicate on there as well.
Secure.
Hydraulic secure. Belt back off.
Hey, pilot, before you move, you see that sea star that was left on the floor?
Yes.
Chris Marr would love to have a zoom on that.
Copy that.
So I'll give you your $20 later for collecting that rock.
Oh, you're welcome.
Thanks.
I also have a fish in the background. Don't know if it's an Oreo or not. Relatively large.
I am not seeing it.
Does it have a creamy filled center?
Oh, okay. Yeah, kind of hovering above us.
That's not an Oreo. We should look at that.
Copy.
Oh, yeah,
Should we grab that just while it's moving, or?
Yeah, I'm switching cameras to see if I can tell. Yeah, go ahead.
It's right.
Oh, right there. I see it. That's the orange roughy again. We got that earlier.
Understood.
The very pale one. Thank you. We had a long discussion about the age of orange roughies and when they start reproducing. And Mike Vecchione told us that they aged one using otoliths, these calcium carbonate concretions in the head. They aged it at 139 years old for this fish.
He's still good.
And there was another study that showed that in this population, the average age of the breeding fish was 73 years.
I'll pass off. Thank you for reminding me.
Of course.
Okay, video if you want to come in.
I think this is Goniastridae, I think is what Chris will tell us. With these really nice large plates along the outer margin of the sea star.
When we come out.
Chris Ma, guess what? He would like to suction it. Let's see, I'm looking around the room and I see absolutely no surprised faces at that request.
No, I just have this mental image of a sea star like holding on for dear life to the front of the suction device as it's trying to be sucked in.
Ship is stationary.
Its little feet.
I'll bring up the jar cam.
Tara Luke seconds that request.
All right, pilot, you ramped up. Stand by.
And just a reminder, 7 moves to get to the top.
That's right. And how many of those 7 have we done, Ashton?
Oh, no, I was counting. It was 8, but I think we did 1.
Oh, I thought we actually went backwards.
Oh, maybe.
You bring up my video when you get a chance, please.
Okay, thanks, Chris.
15 whole meters.
I mean, to be perfectly serious, I mean, this is great. We've got a lot of interest here. We've got a lot of different kinds of interests. And, you know, it's one dive. It's hard to satisfy everybody in the room.
Hydraulics are ramped up.
We try to do a little bit of everything.
Bring the door out here.
Okay.
Going to start rotating out or bringing it out.
Understood.
So Chris Ma relays that the last 2 types of sea stars like this, and Chris, can you confirm for me, is this a Goniastrid, so I can put it in the database log? Chris says that the last 2 that we collected on the 2 previous expeditions were both new species, and one of them was a new genus. So that shows you how little is known about this group and why these collections are so valuable.
Understood. You can come out. Is that full out?
I can come out a little more.
Hey, Scott.
Hey, Chris.
Thanks
Thank
so
you.
much.
Yeah, of course.
And thank you, of course, to GFOE, without whose expertise none of these collections would be possible. Goniastrids are among the most diverse of the sea stars that are alive today, and even in the fossil record, there's quite a few of them. And I honestly thought that in the Atlantic we weren't going to see that much diversity, but I am astonished how many of these things we're seeing that I don't go in the book. And so, you know, and at this depth, especially, every time we see something like this that doesn't quite fit into one of the known groups, I'm quite surprised. So I mean, I have no idea what it's doing, but I also thank Ashton for collecting that rock, because if he hadn't collected that rock, we wouldn't have seen this. And
well, I only collected that rock because I knew that star was behind it.
Well, I attribute all of this good fortune to my geology training as a scientist, and all that time I had to spend hanging out with hard rock geologists. And for your gracious rock collection, I offer you this joke, and you'll probably get it, but where do you put a peridotite clock?
I don't know. Where do you put a peridotite clock?
On the mantle. Get it?
Oh, boy, we're pulling out the nerd jokes now.
I get it, and I'm going to have to explain everyone. All right, so when we think of the top of the oceanic crust, we think of these pillow basalts, and underneath these pillow basalts, we have these basaltic dikes that are feeding those pillow basalts, and beneath those basaltic dikes, we have the lower crust, which is gabbro, and then beneath that gabbro, we have the upper mantle. The upper mantle is primarily made of a mineral called peridotite. So where do you put a peridotite clock? On the mantle.
Well, now that joke is much funnier. Thank you.
Even though you had to explain it, yes.
Oh, because he had to.
Some of us were already laughing.
There you go.
Chris. You're pushing on the ground.
Understood.
Queued up.
Thank you, Roland. Well, I must credit my colleagues at the University of Illinois in the geology department for providing me with that jewel, which I really don't think I would have come up on what's on my side.
I'll make sure I pull that one out later on a later dive chat.
Be able to bring your forearm down. You know what I mean?
So please spread it around like wildfire.
No, right?
Yeah, you could still scrape, but what that would do is bring your arm out of the way a little bit. Might help you see it better.
Will the star actually go into the suction sampler?
Yeah, I think so. Yep.
And then
we're just playing with that
kind of move your jaw
standard problem of 3D, 2D, a 3D space with a 2D screen, and just understanding how far it is.
Right to left.
It's small enough.
Well, it's actually kind of 3D, 2D, 2D, right? Because the starfish is kind of 2D, right? It's on a 2D surface. It's not really prone above the ground. So.
It depends how much you're willing to average out that third plane.
Yeah.
That third dimension.
Oh, a fair bit.
Okay, it'll fit.
Sorry, that's...
One of the interesting things about the general physics of the body on these is you notice how all of the arms, all of the sides, the plates on the peripheral side of the arms, how they kind of join over the middle, and they kind of create an almost flat triangular surface on each arm. And so it's almost like a snowshoe. It spreads out the surface area of the animal. It's never been sort of, it's all kind of intuitive hypothesizing based on body shape, but it's thought that this is how these animals sort of adapt to the soft sediment here.
Okay, and then that allows them to kind of stay on top of the sediment as opposed to sinking in.
Just stuck in the turn, so the more you bring the hose in,
and there's other there's many genera of goniastrids that practice this, or is thought to practice the same strategy.
If you don't mind.
I mean, it's never been like shown empirically, but it does seem to jive with a lot of the habitat.
So I don't know, Chris, if you still have the delay that you had earlier, but we sucked up the star, and it seems to be in the tube somewhere. And so we're kind of doing a waggle of the tube and varying the suction to see if we can force it down the throat of the tube. It's in there. It's just somewhere.
Yeah. Well, I'm at the museum, so I'm seeing the low latency stream, and I saw the collection.
Okay.
But as you say,
I don't think
know if
have we seen it go into the collector? No.
Yeah, I can't see.
Okay, holding.
Understood.
Now maybe this is the time you suck up a small rock.
I'm not going anywhere. It'll be fine.
Yeah.
What's that?
I said maybe this is the time you suck up a small rock and kind of.
We're at full suction.
Yeah,
clear the pipe.
I can only apologize on behalf of my request for the collection, but
no, I think it's
There you go.
We'll see
There it is.
Yep, perfect.
It's in there now.
It's in there now.
In there?
Yeah.
Bonus.
Okay.
RPM.
Well, thank you. Was it Lars doing the collection?
There you go.
Who's up there?
I think I can't tell if it's Chris.
Chris Wright
Nice work, Chris.
was the pilot for
Yeah,
that.
Chris Ma.
Chris Wright.
Okay.
Chris Wright is, I think, at pilot, right? Yep.
Yeah, sorry, Chris. We have Chris has
okay.
nav,
Well.
Chris has science on shore, and Chris
There it
is
is.
pilot at the moment. So a lot of Chris's.
There's a lot of Chris's.
A lot of Chris's.
Chris
Yeah.
Ma, thank you. Very
Yeah.
much appreciated.
We just had a
Chris
rotation of
collection on
the
dive.
we just had a rotation of the canisters, Chris, and verified that it is in there, so it's in good shape.
Oh, lovely. Okay. Yes. Well, I see that
Bring in the
my
door as
naming
well.
Platonic Caster GFOEI is well warranted. So
There you
thank
go.
you. I'll talk to you guys later. Appreciate it.
See you, Chris. Thanks for calling in.
Bye-bye.
All right.
Nice.
Nice sample, pilot.
Thank you. Hydraulics secure.
Little back deck check. Still there?
It's been like 3 months since I sat co-pilot.
Oh, we could change that.
So, pilot, the final component here was to look at that coral that's in the very upper right of the screen right now,
Understood. This big one here.
the big one. And if we get when you're ready, get a close-up, we want to look at the polyps and at least a couple of branch points, please. And so just as a reminder to everybody, what we were doing before we did those couple of collections is there were 3
pilot, you also have
bamboo
the,
coral colonies here in a row with different stages
Fish
of
over to
branching.
your
And the question
starboard.
is, are they
Looks
all
a
the
little
same
bit different.
species or different species?
Understood. I'll get
And
this
it's
for
really
these
hard
guys
to
first,
tell
and then we'll-
Understood.
from a distance
take a peek at
because
that.
there's a lot of variability in how and when during their life they can grow these branches. And actually, I should be careful there. There may not be as much flexibility as we think for some of these species in when they branch. And so the hypothesis with this sort of thing is that it grows for a long time unbranched, which it must because it's only growing from the tip. And you can see here, looks like perhaps close to a meter. Oh, there's a squat lobster kind of swimming up onto the coral. Maybe he's going to grab on. Oh, kept going.
All right, Rowan, you can take that.
Can you see it? Yeah, so we're looking for other clues to see, well, what else can we look at to see that if it's unbranched, how do we separate them? So here there's something a little odd going on at this node. It's like there's a double node with a very short internode between it. That's very interesting. And there must be some ctenophores on the colony to the left because I see these 2 or 3 strings wafting through the screen. Those would be the fishing strings that are being extended from a ctenophore. Each ctenophore has 2 of these tentacles.
Find a branch.
Yeah. So if we could just keep going left a little bit more. Actually, a lot of these nodes look kind of wide.
Like center screen.
It's a good view. Still pretty milky. You can't see the skeleton inside there all that clearly. Is this one as spiny as the last one? Right there might be a good place to zoom in if you go back down a little because one polyp was closed.
And video just came off my toe there.
Right there in that bend.
Wide.
And then I promise we can move on. Yeah, Les Watling thinks this one is quite different, and I tend to agree.
All right, Rowan, we can come back in on that.
The tissue's a little bit more transparent, and the prominent sclerates I don't think are there. Yeah, you see that one polyp in the center? It looks like it's kind of closed up on the upper side of that middle branch. That's what I'm looking at.
Built up, coming up.
Next there.
And can you tilt down just a little bit, or maybe you're moving down? Yeah, right there. Yeah, so this one definitely doesn't have as prominent the needles. Okay, thank you. Let's resume our journey.
Understood. Are the videos ready?
120 meters to waypoint 2.
Crossed it.
All right, Pellet, looking down at you, 5-5.
Lasers on. Push mode. Understood.
Clear.
Nav, watch lead 2.
Hey, watch lead 2.
Hey, can I confirm that we have on the order of about 2 hours of bottom time remaining?
Yeah, let me double check.
Thank you.
What do you got me on, Lars?
5-5.
Did that fish off serial ROV right go away?
Yes.
Okay.
Yep.
Looks like another small hemicorallium down here on the right side.
Okay, following you up, Pilot. Right there is 4-5.
Understood.
Pilot, feel free to zoom in on this fan-like colony on the right side of the screen to the left of that barrel sponge.
I'm taking partial of the video. Still creeping in on.
This is kind of looking to me like a Candidella primnoid. It's most certainly in the family Primnoidae. And what I'm looking for right now is swellings in the branches. That's the characteristic I use because that species, Candidella, I'm forgetting the species name right now, houses polychaete worms. And you can often see where they are because of swellings in the branches. And I do think I see some of those swellings. A couple of amphipods there.
Is that the rotator that's jittery? Or is that thrusters?
Hermit crab
Might be something in my
on the
thrusters.
seafloor.
What's that?
Might be something in my thrusters.
Copy that.
There's that nice Farinema with all the silica spicules extending from the base, anchoring it to the substrate.
All right, come full line. Yeah, I'm not sure.
A couple of
rotator and it was still
Okeanos, actually 4 of the Okeanos bamboo corals growing in the sediment, or shallow sediment, maybe it's rooted to the rock. There's a lot of rock poking through.
Well,
Imbricata, thank you, Manuela.
cleared.
Candidella imbricata, that's what I was trying to remember.
Okay.
So I'm pretty sure that's what that colony
Something cleared.
was.
Yeah. Yep.
Yes.
Okay, should be all good, Pilot.
Thank you. Yeah, I was saying the jitter.
Then we get on this flatter area, there's a lot fewer colonies. There's a Chrysogorgia that doesn't seem to mind growing here, and there's lots of Acanella. They seem to be quite happy on this flatter surface. And it looks like the Chrysogorgia colonies are on the rocks that are just poking above, or perhaps they're just slightly buried.
Copy bio.
But we've lost a lot of the other suspension feeders.
All right, I think we can go ahead and get another move if you're ready, Pilot.
I agree with that.
Still feel good at 100? Sorry, 110?
Yeah, 110's good for me.
Good copy.
110, you want a 20 meter? What do you guys want?
Like so.
Yeah, we can do 20.
Let's do it.
Les Watling thinks that may have been a Norella.
Nixon tracks.
Maybe there at 4-0.
Understood. 4-0.
I haven't seen any squid.
No. Neither have I.
getting a little out there.
Understood. Yeah. Is that a new fish for us this time?
I don't know that it is, but if you're going that way anyway, please feel free to zoom on it.
Take a look at it. Grab a partial there video.
I think it's the genus Lepidion. And if so, we did see some earlier and did get some comments about it.
Sorry, I was going for the
No worse.
cookie Oreo.
Very nice view of this. Yeah, Mike Vecchione agrees.
Yeah, sure.
Lepidion. So we've seen this at least a few times today.
Video's clear.
All right, let him go. Come back.
Ken Sulak said earlier that those long pelvic feelers and the long chin barbel in this species are both sensory.
It's better to raise a vehicle.
And the long barbel suggests that this would be Lepidion guntheri. The tall black first dorsal ray is possibly not sensory, but it is used as a species recognition character.
That was really good. But,
thank you.
Keep that in mind, where if you think you know where it's going, try and use the thrusters.
Oh, what he's saying is it might be used as a character for one fish to recognize the other as being in the same species. So he says, well, if there's no sun at this depth, then why would you have a visual species recognition character? So in this case, the species range is well above that critical light extinction depth, meaning this species of fish that we're seeing probably can be found shallower where there is sufficient light, that the coloration and size of that fin would make a difference in attracting mates. Very interesting. Thank you, Ken.
Go ahead, Bridge.
Lots of Okeanos and Chrysogorgia along this transect. Not sure. This is probably a large bamboo coral that's just starting to show up in the distance on the upper surface of this rock. I'm basing that kind of on the shape of the overall colony and the apparent thickness of the branches. It's not really the branches that are that thick, but it's the polyps from the distance give it the appearance of thick branches. There's a nice Leopathes black coral growing off both sides of this rock as well, on the lower sides of the slope. And there may be another hemicorallium, the pink precious octocoral, just in front of this much larger colony on the rock face. The bird's nest sponge. And I think there's also a colony of Acanthogorgia. If we hadn't made so many collections already, I might want it, but right now I'm more interested in getting to the rim. See the way that's flexing there? I'm now going to have to change my mind about it being a bamboo coral. I believe this is probably a prinoid, maybe a plexaurid.
Be advised, you're right at the corner.
Understood.
And I am moving forward.
Understood.
You're over here, Lars.
Pilot, just checking with you. Will you give me a close-up of that coral? I'm not sure if you're positioning for a better view.
Yep. Just sliding off here, but yeah, we can.
Okay, yeah, I'd like to get a zoom on the polyps if you have a moment.
Stand by one video.
Thank you.
Standing by.
Do you want me to swing my head?
Yeah, if you can come over, just keep me in view.
Got that. Coming over.
It's this big one on the left. Watch lead.
Yep, big one on the left.
All right, rolling.
Came over 20 degrees.
Understood. All right, rolling. Snap down.
Now I know I can't see it well enough. There we go. Oh, okay. So this is the, we collected this. Wait, didn't we collect an inverse one? Didn't we collect one that was yellow with blue polyps? This one now is purple with yellow polyps. So this is a plexaurid for sure. And it could be a Paramerissia in the genus Paramerissia. This is not completely unusual morphology, and I've certainly seen Paramerissia of this color. So I'm really glad we came in and got a close look at this. Very clearly see the 8 pennate tentacles, and in this type of coral with the thickness of the tissue, those polyps can completely retract into that purple matrix. That purple matrix is called the cenonchyme, and it also is packed with sclerites, but it also has tubes running through it that interconnect all those polyps to one another, and it's essentially part of the digestive system, the gastrovascular system. Thank you, pilot.
All right, video whenever you're clear.
Video's clear.
All right, come full line. All right, coming back over to you, Lars.
Good copy. Yeah, came 20 degrees to port.
Understood.
Some wire corals. That's the black corals that grows these very thin whips. Do a lot of coiling.
This is a tip.
Sideroid
You're
urchin.
settling uphill right now.
Yeah, I'm turning
A lot more platey steps here.
If you kind of come back as you
Yeah, and I was kind of interested on that feature that coral was on. It was just an odd shape, for lack of a better word. You know, it almost looked like a garlic clove. Like, you imagine a really large garlic clove. It had kind of a bulbous base, and then it kind of tapered off towards the top. It didn't look like, you know, a large piece of basaltic rubble that had come down.
Okay, I'm going to start bringing my head back.
And again, you know, these
something sort of. I don't know if that's a breakage plane, an erosion plane.
No, that looks like a little fracture plane.
Cool.
Take partial view.
That's nice.
But these overlapping benches of concretions are kind of odd. You know, part of that could just be because as we saw in some of the areas, maybe on our first or second dive, we saw that the bottom currents kind of made these almost large wave-like patterns in the pteropod shells, right? And so this could be kind of a larger scale version of that, where the prevailing bottom currents kind of make the marine detritus and pteropod shells or whatever's forming this calcium concretion into preferential little waving standing patterns, and then those get lithified in that kind of form, and then they just get reinforced. But I'm not 100% sure because
Sure.
Yeah.
That's so interesting to think about because it, again, makes you think about the immensity of time that's created some of these structures for all of those shells that we've seen deposited on the seafloor to become aggregated and cemented together. Really nice view, side-on view of lots of these tubular sort of they're tubular bushes. Those are Chrysogorgia are the paler ones, and the brighter orange ones are the bamboo coral Acanella.
Totally tubular.
Tubular. This is all I see now. Roland, our video engineer, said this before, and I don't know why I was so dim to catch it, but he was referring to them looking like bowling pins, and that's absolutely the case. You can see that really nicely on that one on the far right. That's kind of orange. So that's the Acanella, that's the bamboo coral. So if you have a bunch of those Acanella lined up, you just grab one of those, I don't know, a Geodia sponge or something to use as the bowling ball. Can we zoom to the right of the lasers? There's a little fan there, red in color, and I'm wondering if that's a Swiftia. Below the lasers now. Yep, that's the one. And I think the stark white colonies, just the lasers on one of them right now. And there's several others here. I think those are also hydrocorals. Although the one in the far distance on the right, that may be an Antilopsamia.
You're going to take partial there.
Okay.
Yep. Yeah. Yep, I'm still going to stick with Swiftia on this one. In fact, I can see one of the polyps sticking out is kind of yellow in color, so absolutely. That is Swiftia. I've seen that before. So that's good.
Full wide. Thank you.
That may have been enough for him. All right.
Probably go ahead and get another move.
Let's do it.
15. 110 again.
15 meters.
You good with that, pilot?
Yeah.
Copy that.
Sounds good. Thank you.
Over on the right-hand side, there might have been a larger sea star. It looked like it might be feeding.
Understood.
But it was a little ways off, so I might be mistaken.
You committed me now, Lars.
Copy.
We're getting close to lunchtime on the East Coast, so why don't we give a summary of what's going on here. If you're just turning on the internet streaming video here, you're watching live streaming video from the deep ocean in the middle of the Atlantic Ocean.
That's yellow is what I saw.
This is the Voyage to the Ridge exploration team on board the NOAA ship Okeanos Explorer.
Move us in.
It also may have been employed to
My name is Scott Frantz. I'm a deep sea biologist from the University of Louisiana at Lafayette. And I'm the biology lead here, and I'm joined by Dr. Ashton Flinders, who's with USGS geologist. He's busy. Oh, there we go.
I gotta jump in before you call me a geologist. So
sorry, volcanologist. My bad.
Before you call me a volcanologist, too.
My bad again.
I guess I'm a geophysicist. I guess you could say I'm a geophysical volcanologist. So I study volcanoes through geophysical means. So using earthquakes, variations in the gravitational field. If it can be done with a computer in a volcano, I'll do it. But yeah, I'm with the Hawaiian Volcano Observatory in Hilo, Hawaii, as part of the U.S. Geological Survey.
I think Instagram tells me you're a geologist.
I know. Well, you know, I as I've been saying, I'm not a geologist, but I play one on TV.
Internet don't lie.
Yeah.
So we're all out here on the NOAA ship Okeanos Explorer exploring the Mid-Atlantic Ridge, which is a rift zone. It's basically the birthplace of our planet's oceanic crust. The plates are moving apart and being replaced with new crust down here. And the feature that we're diving on today is about 60 nautical miles or so west of the center of that spreading center. And we're actually in this little volcanic feature up on one of the ridges, and we started the day off in the center of the crater of this feature at 1,235 meters depth, and we've been climbing the wall all day, and we're moving up towards the lip, which is at about 1,104 meters or so, still about 20 meters above us, more or less. And I think we've been really pleasantly surprised that we've seen a whole lot of diversity and abundance of sessile fauna. Sessile fauna means those animals that attach themselves to the seafloor. We're passing over another rock pen right now. I'm just going to note that. It's great to see. We collected one earlier. So sessile fauna, these are the animals that attach themselves to the substrate. They grow up much like plants and capture their food out of the water column. Lots of sponges, we've seen some interesting fish, and then all kinds of different associated organisms that live on these corals and sponges. And we're heading up towards the rim of this little crater-like feature where we believe that the currents will be even more favorable to these suspension-feeding communities. And that's in part because of the depth, in part because we're out of the crater feature, but also because the shape of the rim accelerates currents over it, just the way the thick part of an airplane wing creates
also
a
looks similar
lift
to the,
in
one
the
that
air.
the student was studying.
And so that should be really favorable to suspension feeders. So we're curious to see, will we see a very high density of this community, although we're already seeing a spectacular community. This, to me, coming into view, it looks like the bamboo coral Ecnomysis. Les Watling and I described several years ago a new species, Ecnomysis dallioi, so far is the only species in that genus. And so we could guess that this is it, although the one that we sampled was in the western Atlantic. So don't know for sure. But it has a large anemone growing on its branches. And this is an interesting thing. Cindy Van Dover brought this up, I think, maybe last summer when we were having some discussions about things that we saw on corals. That anemone looks really thick and heavy, but it's really not. It's neutrally buoyant because it's mostly full of water and it's surrounded by water. So it's not really creating much strain on this coral at all. You can also see a lot of threads that are moving by to the right. That's a good indication of what the local current is right now. And those threads are the feeding tentacles of benthic ctenophores. In the water column, you may be familiar in shallow water with the sea gooseberry. That's another kind of ctenophore. These are specialized ones that get flattened. But just like the sea gooseberries, they have what are called retractable tentacles. That is, tentacles that have musculature in them, so they can be pulled into the body. And they're studded with these structures called coloblasts that are very sticky when they impact something. And so they're used to fish for zooplankton. And if something comes in contact with that tentacle, it can get stuck to it, and then they'll use the muscle to pull it back and get it into the mouth. And there, actually, you can see some on the branch there. They're kind of yellowy orange in color. Just on the backside, this curly branch in the front got in the way. But I can see a few individuals. Oh, thank you. Manuela Ramos, who's down at the Exploration Command Center down in Horta, said that that anemone is Actinocyphia aurelia.
One on the right with those strands.
That name aurelia refers to its sort of golden yellow color. There we go. We're getting a little bit of a look at these ctenophores that are on the coral. The coral itself is kind of more pink in color, has a white internal skeleton. So those blotches of yellow-orange you see, that's the ctenophores sitting on the coral. Very cool. And we've seen now lots of these, so they're obviously pretty common in this area. Okay. And we were able to get a sample of them, so hopefully we can get a name applied to those so that in the future we'll be able to say something about the number, the abundance of that particular species up here on the northern Mid-Atlantic Ridge north of the Azores.
Is lasers as well.
Yes.
Today we're about 127 kilometers north of the Azores. We've been getting closer and closer, and over the next 2 days we have 2 more dives as we move towards the islands. We'll be really close to the islands. We'll be up on the plateau. We're still not on the Azores plateau or on the Mid-Atlantic Ridge, so those dives may have a very different character.
Yeah, and it's one of the things I'm excited about seeing in terms of the change in the geology. So Scott was saying we're on the Mid-Atlantic Ridge, which is a mid-ocean ridge, or one of the forms of divergent plate boundaries in plate tectonics. It's one of the places that the plates that form the outer, where we are, we're about 25 kilometers west of the Mid-Atlantic Ridge on a normal fault. So that's a place where the Earth has been faulted or sheared in half, and we have an uplift on one side and a downdrop on the other, and we're kind of on the upward end of that normal fault. We're on the North American plate right soft, now. Now, if we went to the other side of the Mid-Atlantic Ridge,
all the way up
due east, we would be on the Eurasian plate.
South for me as well.
And as we travel south towards the Azores, we're going to get in this really geologically interesting area, because we're going to have a place where the Mid-Atlantic Ridge that we're on now, the segment that we're on now, and then another section of the Mid-Atlantic Ridge that's coming more or less east by south or west by southwest intersect, and then from there a second mid-ocean ridge known as the Terracera Ridge moves out due east. And so we're on this, what we call a ridge intersection, and that's where 3 of these tectonic plates are separated. So right now we're separating the North American plate and the Eurasian plate, but when we get down towards this ridge triple junction, we're also going to be separated away from the African plate. So really interesting in terms of general tectonics. Now, to confuse all of that, we also have the Azores hotspot. So we have this process of hotspot volcanism, or volcanism that's occurring within the middle of a plate. So a form of volcanism that we do not associate with standard plate tectonic processes. And that kind of confuses everything else that's going on. And so as we're traveling south, most of the Azores archipelago is along that Terracera ridge. And it's kind of on straddling the end of the Eurasian plate and the African plate. And the two northwesternmost islands, the islands of Corvo and the islands of Flores, they're still on the North American plate. So we're kind of, we're moving through the ocean, and we're coming up on the Azores Plateau, which is a large bathymetric swell, or a bathymetric high point. It's an increase in the mean depth, or the increase in the mean elevation, a reduction in the mean depth of the surrounding ocean floor that's formed by this hotspot volcanism. And then as we come closer, we're going to get to this ridge triple junction. So it's really, really interesting and exciting from a geology and geophysics standpoint. And, one of the things that's will be interesting to me, not being a biologist, will be, does this Azores Plateau kind of act as a barrier? Are the benthic communities that we've been seeing different? How is the overall geology and changes in the tectonic setting also contributing to the changes in biology?
It's a great scene here.
Partial.
We just got one of our colleagues on shore said, this is what I call a seascape. It's a really beautiful scene. We're looking at black coral just in front on the left side there. That's Bathypathes pseudo-alternata. And then in the background there is that Swiftia that we saw before. What else do we have here? I think there's two Swiftias and some bamboo corals, two of the upright colonies of Chrysogorgia. Oh, there's that purple plexaurid again. So this one actually has a few side branches. I do think that's similar to the one that we collected
a little less than 2 hours.
In the earlier part of this week. You see a cup coral. There's a little Storopathes black coral underneath that bamboo coral on the left. There's a Thuarella, maybe grasshoppi here in front. So let's see, that's one. Oh, and there's a Dendrobrachia at the back. 1, 2, 3, 4, 5, 6, 7, 8, 9. Maybe another Dendrobrachia.
Slow tilt, right?
A couple of hydrocorals, 10. Oh, and the Swifty, 11. So right here in this view right in front of us, 11 different species, plus the cup coral, 12 different species, plus the Lophelia, 13 different species. So talk about diversity. Instead of just seeing a rock that's all filled with the same thing, which may be a high abundance, that would be a low diversity. It's not a very speciose or biodiverse landscape or seascape. In this case, we have pretty good diversity.
Back on.
I didn't even mention the sponges.
We might want to get a small move, and then we might want to start heading more south.
Understood. You'll get another 15.
I was thinking 15 sounds good.
Understood.
15 going.
110.
110. 110.
Oh, there you are.
Get you there at 3.5.
Understood. We lost the follow of that downwelling as soon as we came over the top.
And so Scott and I, we introduced ourselves, but this work that's out here is a multi-group effort by a lot of different partners and collaborators. Scott is, as you said, is from the University of Lafayette.
Louisiana
at Lafayette.
Yeah.
I am from the U.S. Geological Survey. We have, sitting on my left, our science data manager, Arvind Shantaram, who's from
NCEI.
NCEI and the Northern Gulf Institute.
What's NCEI? What does that stand for?
National Center for Environmental Information.
There you go, right? Environmental information. We have, of course, the the NOAA ship crew, all our NOAA Corps officers and ship's crew and stewards, our union members and our contractors. And then we, of course, have this great partnership with GFOE, the Global Foundation for Ocean Exploration. And they bring an amazing team out with them that includes, it's a huge party of people, right? We have video engineers, we've got ROV pilots, we've got videographers, we've got network wizards. We have a whole group of people. And so in this back row, you've heard from Arvind, myself, and Scott, and then we have Anna, who you heard from earlier a little bit, who did a birthday shout out to her sister. And then in our front row, we have our three amazing ROV pilot, co-pilot, and navigator, and then our video engineer on the far right. And so maybe we can get an introduction to our ROV team when they are ready. They're just finishing a ship's move, but I'm sure they would love to introduce themselves and say hi. They're responsible for all this wonderful work we do.
Hi, I'm Jonathan Allen, and I'm running Navigator for right now.
You can take more video.
Sitting pilot, we have Chris Wright. Sitting video, we have Roland Bryan. Myself is Lars Murphy, sitting co-pilot. And behind us, we have Anna Sagatova, sitting Klippa.
Yeah, thank you. So, those ROV, the ROV team up there, the system we're using is a two-body system. It's got the main ROV that you're seeing that's taking all the footage, and that's the ROV Deep Discoverer, and that's connected to a tether to a second ROV, Sirius. And in order to move this entire system, we have to move first the ship, the NOAA ship, Okeanos Explorer, and hanging off the ship, Okeanos Explorer, over a winch is a cable that goes down to the bottom of the ocean. Right now it goes down to about 1,100 meters below sea level, and attached to the end of that cable is Sirius. So our ROV navigator communicates with the ship and helps move the ship which will move Sirius. We have a co-pilot who helps direct Sirius, and then connected to Sirius is the Deep Discoverer, and our ROV pilot moves Deep Discoverer within a radius, the length of the cable that's connected to Sirius. So it's a really intricate dance between these three very complicated pieces of machinery.
Wait, sediment?
Yep.
Understood.
It's a snap on it or something as we go by. They've sort of looked at them each time we've seen them.
You can take more there if you want.
May also be a rock. It's a rock.
Thank you, Anna. Pilot, if we could look after this upper right, there's a large white colony coming into frame. Yeah, up there. Thank you.
Now Scott's going to say, what you going to say?
I was going to say that we'd be remiss not to also point out that there's a whole host of people on shore who are helping us out. If you are enjoying the web pages, for example, that are associated with this cruise, you see that there's new content going up every day. So we have Emily Crum and her team who are keeping that updated. We have folks who are managing the video streams from shore. And I have to note, I have 5 students back in Lafayette who are working on annotating the video, and their fingers must be killing them today because there's an awful lot of stuff to annotate. And I've seen that there are folks, various interns in different line offices of NOAA who are also working on annotations and different aspects of this expedition, and then of course all the planning that went into the expedition. Our EC, Derek.
Yeah, our expert expedition coordinator, Derek Sowers, who's probably
got his fingers in every pie
in his room with his headset on, listening intently to what we say.
He doesn't listen to what we say.
Yeah, okay. Well, it depends. Depends if we say something right now and he comes running in.
Yeah. I'll bet he's in there banging his head on the desk.
Oh, yeah.
I wonder. What do we got here?
Out of joy. And then we of course have a whole mapping team who's also come out, and they're the ones who do all of our multi-beam sonar mapping. in order to create these really situationally needed dive products. So these are maps of the seafloor topography, the bathymetry. So the ROV knows both exactly what it can expect when it gets down to the bottom of the ocean, but also so Scott and I can interpret them and plan where this dive should take place. They are working there.
Take more video.
It's 3 in the morning and they'll be up working. It's a little scary sometimes, but they do an amazing job as well. And so that group is being led right now by
Shannon Hoy.
Shannon Hoy. I was going to say your name.
We all suddenly like.
I was going to say a NOAA expedition coordinator who's
sorry.
acting as a mapping lead right now, Shannon Hoy.
Your version was better.
I know. That's why sometimes I got to take a second or two to
You're right.
compile my thoughts.
You're right.
Yeah.
I usually have to take more.
tilt down
Oh,
again
actually,
also?
that's really helpful. This view at the very
Strands
top.
on the left
You can see
edge
the
of the
skeleton
screen.
pointing out. I think this might be the dendrobrachia
left.
again. Oh, but
See how flowing those are?
Tina Sacco think maybe it's something completely different. I
And
don't know.
we left somebody else out.
We left probably a lot of people.
I know, but we also left
Tina Sacco. Oh, all the shore.
all our shore-based scientists.
We didn't leave them out. Come on. We didn't leave you out, guys. Chris Ma, right now I'm just reading in the room. I don't know if I got disconnected and have lost a lot of names because I know there's more people in that chat room than the list shows, so I apologize. We're not going to name you all, but Chris Ma. Oh, stand by. So we're seeing some really long tentacle-like structures there. The only thing I can think is that these are the equivalent of sweeper tentacles. They've been described from various shallow water corals and at least one deep sea bamboo coral in the Pacific. And their response to, in shallow water, they're kind of a response to competition. So your neighbor's getting too close, you stick out some of these and try to fend them off. Internidarian warfare, I guess. So perhaps that's what I see. It's a little bit hard. It's, there's so many polyps in there. There's so much white. I can hardly see where they're extending from. But they do appear to be individual tentacles that are elongated, I think.
Only there. I have a little bit more if you'd like.
Yeah, bring it up full.
So, I mean, there. Oh, yeah, that's great. Thank you. That extra lighting helped. Yeah, you can definitely see that it's some individual polyps on the left side have really long tentacles. And so it's some kind of growth response to, I'm not sure what. And what you can also see is that those long tentacles don't have side branches. I thought sweeper tentacles either came from within the polyp. I guess those would be a concha. So yeah, somehow they're being modified. It's a mystery. Yeah, so in our chat room right now, this is basically a way that the scientists can engage each other and engage us on shore with various observations, feed us information, ask each other questions. We have Elena Hebert, that's one of the students who's in my lab in Lafayette. Chris Ma, who you heard from earlier at the Smithsonian. Daphne Cuvelier, I think she was one of the Azorian scientists, if I recall. She sent us a really nice photo mosaic this morning from the Moitira event.
Yeah, the Fomorian hydrothermal chimney.
Yeah, that was very cool. She took various images and stitched them together to make one overall view. James Aubrey, another student in my lab, he's working on Acanthogorgia, so he was really interested in that yellow coral that we just passed. Mary Bilan, I noticed yesterday, introduced herself from Italy. Tara Luke from Stockton College in New Jersey, I believe. Tara.
Yeah, I think we're still looking at
Asako Matsumoto, who's a coral biologist in Japan. Les Watling, who is presently at the University of Hawaii, but he lived and still lives in Maine. He was at the Darling Marine Center for a very long period of time. He spends his summers there, so I think he's checking in from the East Coast. I just see Megan McCullough has now joined the room. I believe Megan works on bryozoans, so she's seen a couple of good things today. And Megan, we did collect a beautiful specimen of a fan-like bryozoan this week. Who else do we have?
And one thing I love about the shore-based scientists who participate in the chat, some of these wonderful biologists, is they seem to either consciously or subconsciously, but as a collective group, have decided that as soon as you leave for lunch and I'm here alone, they ask for a sample as pretty much as soon as you walk out the door. And it gives me this quick 5 minutes of panic where I look around going, does anybody know what I'm looking at? And then go, do we take it? And then I kind of just stumble and kill time until you walk back in.
I'm going to have a talk with them because I specifically wrote in there when I left, don't gang up on Ashton.
I don't think it's on purpose.
Okay.
And they are very helpful with giving me words that I pronounce horribly.
Absolutely. Yeah, thank you. Tara points out that it's Stockton University, although when she first started working with Okeanos and chiming in the chat room, it was Stockton College, so I didn't totally make it up. Notice there's a purple fan with a lot of branches at the top, and we've been seeing, actually there's one at the base of the screen right now, these purple unbranched forms, and there's been a lot of discussion today about whether those are the same thing. We don't know. There's so much need for time-lapse observations in the deep sea for a lot of this stuff. You know, it's not like we can just pluck it up and bring it into the lab and watch it grow, and especially since they grow so slowly, we're talking about years-long studies.
Is anyone doing that? Is anyone putting down, like the equivalent of deep-sea game cameras that only take a frame a month or a frame a year?
Oh, sure. I mean, there's been drop cameras and camera tripods and sleds that have been deployed in many parts of the ocean.
But anything that's got like a 10 or 15 or 20-year time record?
I'm not certain, but I would bet, like Ken Smith at MBARI, Monterey Bay Research Aquarium Institute, has been doing this kind of thing for, more than 20 years. So it wouldn't surprise me. It doesn't seem like it's all that much more effort, especially the setup that they have over there to, put something out that's take a photo every, once a month? But I don't specifically have knowledge of that. I think this is a Bathypathes black coral, just a really tall one, this feather-like one that has this really cool chirostylid crab on the top. That name, chirostylid, this is a type of squat lobster. The squat lobsters in general can be divided up into two major groups, those that closely associate with sponges and corals and those that are basically moving around the seafloor. And the ones that associate with the coral have specialized back legs that allow them to hang on, and then they have those very long chelate legs with the claws at the end. And that's where the name Chirostylidae comes from. Basically needle arms.
Good copy now, thank you.
So that's pretty cool. Very spiny skeleton.
Understood, stand by.
See its little feeding appendages. Those are called maxillipeds, those two that are sort of curled up just under the eyes.
Take more if you got it.
These decapod crustaceans have fairly complicated mouth parts. So let's see if I can remember all this. They would have a mandible, which has a grinding surface and a cutting surface, and then they'd have two pairs of maxilla, which assist in manipulating the food. And then they would have these maxillipeds, these larger structures that can also manipulate food.
Do the claw.
And I don't know if these long claws are used in prey capture or are used in interactions with other individuals of the same species. Pilot, when we're finished with the video here, can we just get a look at the base of this colony, please?
Of course.
Thank you. And maybe turn on the lasers. And the lasers can come after if there's a beauty shot involved.
Understood.
That's a good shot there.
Yeah, and for our viewers who might be new, just so you can understand, what you're seeing right now, those two little red dots are lasers that are shot out at the ROV, and they're calibrated to be a set distance apart at 10 centimeters. And that allows us to get an idea of the scale of the things that we're looking at. You know, a
I
lot of
thought they
times
wanted them on.
objects in your mirror are larger than they appear, or objects in your ROV tend to be smaller than they appear. And so some of these things look really, really big, but then you put these lasers on them and you realize there might be 10, 20 centimeters across. The resolutions of these cameras are amazing, so we can zoom in to really, really, really small scale.
Yeah, it's like taking a dissecting microscope under the ocean. That black disc that you see is what we call the holdfast. So that's the base of this colony and where it's attaching to the rock. And you see that it's actually missing tissue. If you go just above those two snails, you can see where the tissue begins. And I don't know, but we could speculate from this what look like is going on here, that the snails may be feeding on the tissue of the black coral. But another hypothesis is that the tissue is already missing from there, and the snails are grazing on the bacterial film that is since developed on that skeleton. Thank you for that view. Pinkish colony. There's a pinkish colony somewhere here, hemicorallium or stylasterid. Let me look, Asako. I'm not seeing it yet. Oh, upper left by the sponge, just behind the sort of beige sponge. Right. To the left of that beige sponge on the upper right.
Understood.
Thank you. There's a question here. Is that a hemicorallium, which is the precious coral, or is that a stylasterid, which is a kind of hydrocoral? The precious corals are a different group of octocorals. There's an awful lot of coral terminology that gets thrown out here, so you need your list. It's actually an Okeanos bamboo coral, so it was neither of those two.
I was going to say, when in doubt, it's usually a bamboo coral.
There you go.
Or an octocoral.
So a bamboo coral is a type of octocoral.
Exactly, right? Yep. So you get
Sure. Well, I mean, so the serious part of what you're saying reflects that the bamboo corals, that's the Kurchatov, we're good on this, and whenever video's happy. The bamboo corals, Kurchatov, the chrysogorgiid corals, the golden corals, and the prenoid corals. Those are three different families, taxonomic families, of deep-sea corals that are the most speciose and diverse of the deep-sea corals. And so we encounter them very often. And one of the things that's really cool that we found with the genetic studies is that they all seem to arise from a common ancestor. And what that means is that there have been deep-sea corals for a very long period of time, and they have widely diversified into these many different taxa. And so that raises these interesting questions about, well, why? You know, what are they responding to that might lead to all these different species? And most people want to jump to an adaptation explanation. They're responding to something in the environment. But it might just be that they're isolated. The sheer distances in the deep sea are such that they may stop reproducing with one another. It's easy to see that, Atlantic versus Pacific, for example. And given enough time in isolation, you should evolve to be different just by random processes. So we don't have all the answers yet, but we have lots of interesting questions. $1,113 meters depth, so just about 10 meters left to go before we get to the rim. The top of this rim. Oh, I may have been looking at the wrong colony. Lots of the bird nest sponges, lots of the Okeanos bamboo corals. I think she's referring to those very bright white colonies that you see lined up on the rock back there. So yeah, there's a question about whether those are hydrocorals or whether those are precious corals. So maybe if we find some closer. There's some patches of Ophelia, again, starting to show up.
Sure.
That's the stony corals, kind of beige in color. Very highly branching, much bigger polyps than we see on most of the octocorals, except maybe things like Umbellula and Anthomastus.
Want to get another 15 meters in, co-pilot?
Yeah, that sounds good. So we're still heading
yeah, we're starting to cheat down
south
here.
Southeast.
Yeah,
Yep.
next point's
Last
down there.
move was at 115.
Or east-east-south, I should say. Copy that. I'm already at that heading, pilot.
Yep, sounds good. 15 meters.
I'll put that in. You said 10?
15.
15, copy.
And pilot, we still have that fog light on from the last close-up image. Do you?
No, I'm good. If video's good.
Yeah, video, did you copy that?
Yes.
Pilot, if we can get a look at the bright white colony just coming into view on the right side, lower part of the screen.
Understood.
Thank you.
Okay, I can pay out a little bit, pilot, but you're running out of leash.
Yeah, I think we're just going to be right here, down in front of me.
I was hoping as we got to the top of this feature, we'd actually see more pillow basalts and less of the fragments, and it seems like actually we see significantly more sediment. So keep your eyes out for pillows still. I still want to grab one.
Understood.
Pillows, check. That's a very interesting, large, fuzzy colony in the background. I think that's a massive Thuorella, and if it is, that's probably the largest one I've ever seen.
Yeah.
And by large, I don't necessarily mean in height. I mean the number of branching points in it. Asako, this is starting to look to me something like Enelopsamia or related stony coral genus.
I'd say probably put another 15 on that move if you're okay with that, pilot.
And come in there, but yeah, that sounds good, Copa.
Copy, will do.
Let's see, what have we got here? Yeah,
Tilt up.
It's probably Enelopsamia or related. I don't know why they always feel like they have to be facing away from us, but they are. But that is actually a hallmark of Enelopsamia, that all the polyps open up on one face. But I'm hesitating a little. I don't want to totally rule out hydrocoral because I'm not all that familiar with the hydrocoral, or not as familiar with the hydrocorals. But this has the feel of a scleractinian coral to me. And so, of course, as soon as I say that, Manuela says it's Crypthelia, which is a hydrocoral. Any way we can look on the other face, pilot? I realize that's a bit of an ask because you got to swivel around.
We was just waiting for a little tether here to.
Okay, cool. Thank you.
Look at the
Appreciate it.
white patch. All the way.
I will agree with Asako that we can all say it's not an octocoral.
That one in the background, did it?
Yeah, on the left. It's still on this rock. You know,
Gotcha, just to the left.
Yeah.
Yeah, we're going to do a shift change here in just a second. So.
Yeah, just seeing if the polyps may be more visible.
I'm rotating off. Let me see if I can come around a little bit to the right there.
Watch your toes too, pilot.
Understand.
Nice.
Quickly becoming a pirouette king. You can fly my drone any day.
Just a great scene here with all these elevated rock surfaces with that clearly show how the diversity changes with a little bit of elevation above the flat surface. And that has to do with current flow, sedimentation, settlement spots. All right, this should be a better angle.
All right, Roland, I'm going to take pressure on that. Tilt down.
It's the left ones we want to look at.
Yeah, try to get over there.
Try to bring up the...
there we go. That's okay. Great view. So what I'm looking at, you can see in some of these where it looks like the calyx, the part of the skeleton that the tentacles are arising from, is almost closed over. And it looks like there's kind of like a smiling mouth where the tentacles are arising. So that is absolutely a hydrocoral. That is not a stony coral. So this, I agree with Manuela, the genus Crypthilia. Good call, Manuela. All right. Good stuff.
All right.
So, yeah, I agree. This comment Joanna has said she always thought the hydrocorals were much smaller. I agree. That is quite a big colony for one of those. And, you know, that's why we have to look once in a while. It's the way we learn.
All right, I got some trivia for you.
Okay.
All right.
I love it.
There are 4 primary controls for sediment distribution in the North Atlantic.
Okay.
What are they?
So are we talking about on a broad scale? Like
on a broad scale. Broad scale of the North Atlantic, yeah.
So like productivity would be one, because that'll control the delivery of the sediment particles.
How much
so
do we have left?
So well, it's close. It's the nature and the location of sediment sources. So
okay, right. Okay, so that's good. I would imagine depth is another, because that's going to control dissolution. Or temperature, depth or temperature together.
Yeah, so
or temperature
tectonic
pressure,
history or age of the crust, which is directly related. Depth is directly related to the age of the crust.
Okay, that's
so
fair.
That's 2.
Let's see, what else would control it? What about the, I guess it's origin. I can't say like hydrothermal origin or influence or change.
We kind of said that in terms of...
that's the first one.
Yep.
What else? Something about the reworking?
It's one of the ones we kind of talked about in terms of when we see sediments over some of these areas and why we do or do not see sediments.
How fast the currents are and whether they can accumulate or not, I guess.
Yeah, so that's
waypoint 3.
Waypoint 3 is...
That's the processes delivering sediment to the deposition center. So, you know, the large-scale currents to a certain extent.
Okay, thank you.
And then there's one more, which is a little bit of a niche.
Thank you.
And I'll give you that one. Structural trends in the basement rock. So large-scale structural trends in the crust. So not necessarily the depth, but you can think about those fault features, right? So that helps control those bottom currents, which are also controlled by larger-scale oceanic currents. Yeah, so those are the 4. Yeah.
Good stuff.
Yeah. Try and keep you on your toes.
I like it. Yeah. I'm learning a lot about geology from a non-geologist.
Well,
you were very clear about that.
Yeah, I try. Dig through my book. If I see if I have any other interesting trivia I can stump you with later.
Perfect setup here, pilot.
Roger that. Sounds like we've acquired the top of the ridge, and we'll be making
Roland.
a right turn here shortly.
Just to go along with the comment that Joanna made earlier about that hydrocoral being one of the larger ones that we've seen. Again, so this is Thuarella. We looked at this closely earlier. It's a kind of prognoid.
Look like it.
I don't recall seeing them this large with this many branches, so that's kind of cool. I wonder what that bodes for. Just peeking up over this ridge that we're climbing. So, yeah, I see that Asako, well, I'll just type this in the chat room.
Yeah, according to the Bathy, we have like another 30, 40 meters to go.
Okay. Copy that. I think I heard we just completed a ship move a minute ago.
That's right, a 30-meter ship move.
Copy that. And what was the direction of that move, Copilot?
110.
110, copy.
That's Sirius heading.
Roger that.
I see you looking down at me, 5-0, so good delta. You can probably get another move in when I get out here in front of you.
Sounds good.
And once Nav gets settled. Yep.
All right, pilot, co-pilot.
Hey, Nav.
Nav.
Looks like we got roughly like 30 meters until you're like dead center here. General plan is to head south after this. So maybe another like 15-meter move at a 115. Bring it down a little bit, and then we can start doing cheating south.
Yeah, that sounds good to me, Nav.
Copy that. You ready for that move now?
I think we're in a good configuration for that. Yeah.
Copy that.
So I think one of the differences that we're seeing compared to earlier, one of the reasons we're seeing a bit more sediment and a lot more flat areas is that we're coming up to the top of this crest, this lip of this crater-like feature. So we were on steeper terrain before, harder for the sediments to accumulate, basically more rock outcrop. So the nature of things up here is fairly different. The nature of the seafloor, the nature of the attachment points. Several more of these Crypthelia hydrocorals. That large white colony that we had a little bit of debate on about what it might be. Dendrobrachia or possibly some kind of plexaurid. Several Chrysogorgia colonies, Acanella, that magnificent large barrel sponge over there, the Pheronema carpenteri, the bird's nest sponge. I think I've seen somewhere they've also called it something like old man's beard sponge. Yeah, the large fan Asako, I think that was the one we were looking at a little bit earlier. We said was probably a plexaurid that had the purplish tissue with the yellow polyps. I said reminded me of a Paramerissia. Looks like maybe a synaphobranchid eel in the distance off to the left.
Hey, watch lead pilot.
Go ahead, pilot.
I understand we're trying to acquire the top of this ridge and reach waypoint 3, is it?
That is correct, yep. That's the one just before we turn, right?
Waypoint 2 is what we're close to right now.
Oh,
and we're about to make a turn south.
Right. So we don't necessarily have to get to waypoint 3, but we want to head in that direction once we're on top. Thank you.
If there's anything you'd like to see, just let us know.
Copy.
Yeah, and I'll be curious because a lot of this upper surface, I had hoped to see some in-place basalts that might have not been affected by this talus field, right? That might have been the origin of some of this talus field. And
sorry, is that a wagon wheel underneath this little?
It almost looks like it.
Yeah.
But I think that that post the post-deposition faulting. So the faulting of this volcano after the volcanic products were originally deposited or in place has kind of acted to flatten this upper surface. And that flattened upper surface has probably allowed for more sediment accumulation at the top of it. So we're probably, we were going to an area originally on the flat interior, a little sedimented. We headed up the slope where there was lots of rubble. No, I didn't see any in-place basalts that looked well defined. And then we got up to the top again, and it's flat. And our basalts that formed all those fragments are probably under a meter or a few meters of sediment right here.
And imagine, certainly at least the data we've collected so far, there's no way of knowing the time frame of the faulting relative to the origin of this crater-like feature itself.
Yeah, you can look at the faults that are developing on the active portion of the Mid-Atlantic Ridge and kind of infer them there. A lot of these faults develop pretty early on, pretty close to the ridge axis.
Is that right?
Okay. So I imagine this faulted plain is at least 2 million years old.
Got it. Okay. So closer to the time of the birth of the crater rather than after it's sort of moved to this position away from the rift axis.
And so, that would be kind of a conundrum if this little volcanic cone or peak was a little bit further to the west and hadn't been bisected, then you wouldn't really have an age control of when it was formed. But the fact that we can clearly see it bisected means it was formed before faulting, which means it was probably formed near the ridge axis, and it's probably 2+ million years old. That would be my first interpretation of it. And that's only because we can see that little sliver of the volcano on the downdraw portion.
Get an overview of our sample space
to the fantastically beautiful maps that
where we stand on that.
have been produced from the multi-beam work that's been going on at night.
Sample sheet.
Wonderful.
Currently have one
pilot, as you're moving by to the left of where the laser should be, there's a multi-branching fan. Get a look at that.
Roger that.
Thank you. And maybe you can turn the lasers on.
Lasers on.
Thank you. There we go.
This guy coming into center screen?
Yep, right above the lasers now.
Copy that.
Thank you.
You have 2 suction jars, starboard outer,
Copy
and starboard forward rock box.
Thank you. Appreciate that. And you can come on in, video. Let's go for a little zoom here.
Oh.
Copy, thanks, video.
That's a black coral. I think that's a black coral, Tina Sacco.
Can you get any closer to it?
Sure. So we'll just hold this for a couple of minutes to see what our colleagues on shore say. That I would have sworn that was going to be an octocoral as we were zooming in, but
I'm going to go in for a slightly closer look. Watch lead.
Yeah, great. Thank you. That is not one I'm familiar with. I'm kind of wondering, as we're getting ready to get another look at this coral, if that closer pseudobroid urchin back there is propped up and eating something. Chrysopathes. How cool. Okay, so this is the first representative of the genus Chrysopathes that we're seeing. The suggestion is that it's a species Microcantha. Chrysopathes microcantha. So it is indeed a black coral. Quite a different morphology from all the other black corals that we've been seeing. This one is clearly a very planar fan, very regular branching. It's totally disguised as a sea fan, Gorgonian, but when we got close, I could see the spines of the black coral skeleton, and then the polyps were just all wrong. They're very smooth tentacles.
There we are.
And, I mean, the polyps are tiny, but you can really see the spines. And remember, I said that black corals have another common name, that is the thorny corals. And on this one, you can see those thorns are very prominent. Usually, they're so small, they're only creating little bumps under the tissue.
30%, co-pilot.
Go ahead, video.
There we go.
Can we bring up the extra lights about 30%?
Yeah, good stuff.
Thank you.
And I think what's hung up on it there, on the just right of center, I think that's a radiolarian. I've often wondered, I've been seeing, or it's a radiolarian or a foram.
Is your stable cup out?
I've been wondering, I've seen these things hung up on corals before, and I've said actually on one of the early cruises, boy, that looks like a molt of an ophiuroid, but ophiuroids don't molt. And now that I've done another midwater dive and I've seen several of those in the midwater, I understand that those are midwater organisms that are getting caught up by the
pretty
coral.
close to stationary, if not
Thank you for
already.
that, pilot.
It's been 5 minutes since the ship moved.
Copy that.
It's been hitting you pretty quick where you're only at 1100.
Let's hold that for a few seconds.
Copy that.
Is there a large whitish fan right around here? I'm getting some questions
at a
from
sample
shoreside.
Copy video. Nice shot.
Large. Well, they must be talking about the ones in the distance, because I don't see any others. Oh, yeah, sorry. Just a quick look at this urchin that is to the right of the lasers.
Yeah, copy that video.
I guess it's just behind that coral. Maybe that's what's going on. I just wanted to see if it was feeding on it. Oh, there's some of the fins.
Right here?
Yep.
Come on in. Snap some.
Hey, you see how it's propped up? It sure looks to me like it's feeding there. It's got its spines kind of wrapped around the base of this coral. At the base there, that's an old scleractinian coral. And then growing off of it is that purple plexor coral. All the polyps I see have been retracted, and I wonder if that's because it's being disturbed by this urchin that's down there. So probably urchins are grazers in general. I'll bet it's grazing
Floating right now, video,
on
so.
bacteria.
Is it grazing on that purple?
No, I think it's grazing on the stuff underneath the purple. That's my suspicion. And then just to its lower left, I saw another one. This very small white colonies. That was a hydrocoral. And so if you turn right, pilot, we're getting asked about the bright white colony.
Roger
I think that's another one of these stylastron hydrocorals. Yep, just came into view on the far right side.
Copy that. Let me pick up a little.
And then, folks, I think we're going to move.
All right, come on in, video.
Yes, indeed. So this is the Crypthelia we were just looking at a little bit downslope, the hydrocoral.
Stand by.
And one thing I didn't say before, what looked like a whole series of tentacles sticking out, and I think I even used that term, I believe are actually individual zooids, individual polyps. And if I recall, they're called dactylozoids, and they're the ones that would sting the prey. But I don't think that they are the feeding zooids. I'm not 100% certain with this particular type of hydrocoral, but that is that is the situation for other stylastrids.
Roger that.
We have a bamboo coral here on the right side. Acanella, probably Acanella arbuscula. That's the common species in the North Atlantic.
Video's clear.
Copy that. Full wide at your discretion.
Oh, I even see another little rock pen behind that Acanella and to the right. They're very small, the rock pens here.
Very beautiful landscape here.
Yep. It's great stuff.
Lifting off.
So, just as a warning to shore, I'm pretty sure that we have less than an hour left in this dive. Maybe, on the order of 45 minutes or so. So.
Well, actually, we have 1 hour and 6 minutes.
Copy that, Nav. Thank you very much. Yep. 1 hour and 6 minutes. So let's use our requests for zooms and
last move is
collections
115,
judiciously.
Correct?
That's correct.
We're very close to the
and
upper part of this rim.
To make a
you can see
right
it's
turn
flattened
yet,
out a
or do we need
lot,
to push
so
forward
we're
a
kind of
little
on the
I
inner
think you're
lip.
getting there. You have this one feature kind of out in front of you, roughly like 20 meters away. Maybe it might be good to just kind of push forward, see that, and then maybe swing over.
Sounds good. I like that plan.
Yeah, what do you think about ranging out real quick, pilot?
Let's go for it.
Okay. Nav, would you mind doing that?
On center?
Yes, please.
Give you a little altitude here. Oh, yeah.
There's 50.
Rock field there.
That might be a good time to make the turn south.
I'm okay with that, co-pilot. You ready?
Yeah, sure.
Let's do it.
I'm not sure if I'm imagining it, but I think I see a pretty large colony out there in the distance.
180.
Atop a rather large rock.
Okay, I'm rotating up a little bit, pilot.
Copy that.
And do you want to start heading over and I'll follow you around?
Yeah, I'm okay with that. Watch lead, you're okay with us turning south at this juncture?
I guess so. I mean, if there's still any elevation to go, I would like to get right up to the crest, but we can at least go, let's see, what's our heading right now is east, so southeast, let's say. Is that fair?
Southeast.
So we're sort of cutting across this saddle.
Yeah, that puts us somewhere in this vicinity here.
I'm just trying to interpret that from this 3D perspective that I'm looking at.
Mm-hmm.
Yeah.
little ridge feature there in the
Yeah.
distance.
Yeah, let's scoot over to that 15-meter move.
Yeah.
135.
Sounds good, and then we can turn south from there if we want.
Copy that. I'll put it in.
I guess I would say whatever you think is the quickest way to the
This
local
is
high.
ROV nav. We have a move if you're ready. Range 15 meters, bearing 135 degrees, speed 0.2 knots, please.
Just going to turn to the right and survey a little.
Sounds good, pilot. I'll kind of wait till you get a little bit right of center, and then
Good copy, Bridge. Thank you.
Change my heading.
Roger that.
I don't have that much to go. I only have 15 degrees, 20 degrees.
All right, pilots, that move is in 135.
135.
Thanks, Nev.
It's like a little drop stone over there, Ashton.
Yeah, that one lone boulder.
Yep.
You know what we call that?
A drop stone?
Not in place.
Not in place, definitely. That even I would know.
I still think, the one rock I really wanted to collect on this, and I didn't, was that long line piece of granite right in the mid-Atlantic.
That had something tied to it, didn't it?
It did. It had a whole bunch of stuff tied to it.
Yeah.
But I just, it would be a great centerpiece in on a coffee table. And people go like, where'd you get that beautiful piece of granite? And you go, Mid-Atlantic Ridge. And then they either just nod because they don't know what you're talking about, or just look at you strangely.
Shake their head.
Yeah, like,
So we're just waiting for Sirius to catch up here a little bit. Watch leads.
Okay, we can take a zoom in on the sponge then, if you'd like to look at something.
Copy that.
There's some discussion in the chat room about how some echinoderms just don't know when to say when. So Chris notes that there are some sea stars that'll continue to eat and grow until they kill themselves.
For a soft landing here video, and then we can go in for zoom.
that sounds like me at sea.
Yep.
I'm
Some
going to
truth
go
to
ahead
that.
and change my heading.
Try to get in
135
closer.
pilot.
Try to get it closer.
Yeah, I'll get
to
it between the skids for you video.
Here's another one of these sideroid urchins, which sort of started the conversation,
And
and I
also
didn't notice it, but I think the
paying
comment
your
was made that
HV2
there were a
down
bunch
is a
of
good
spines
cue.
without a test,
that.
which suggested that something ate the urchin. So there was some discussion about, well, what might be eating the urchin down here? Or did it just die of old age or overeating?
Okay. I don't want to stir up too much silt here, so.
So this is Pharonema
See how
carpenteri,
this works here.
the bird's nest sponge. You can see it's got all
Come
these
on in,
siliceous spicules extending from the base that go into the sediment and allow it to be sort of anchored in position. So what's this urchin doing? It's got a little crab on one of its spines.
Go ahead, Bridge. Good copy. Thank you.
That's a Chrysogorgia
moves
colony
complete.
on the
Pulled down.
back side of it.
Copy nav, thank you.
And it certainly doesn't have its mouth oppressed to the surface. It has to. The mouth has to be a lot further down in order to actually feed on anything. Just kind of hesitating, wondering, making sure it's not upside down. I saw one of those yesterday, meaning the mouth is on the left side. I think it actually might be.
Max zoom.
Copy that.
Get some
I'm no
cluster
echinoderm biologist, but
vibration.
I think it's upside down. What else am I seeing? Is this a little decorator crab, perhaps? Looks like it's got various things stuck to its back. Can't tell if it's spongy.
Okay. Full screen.
Roger that. Stand by. I'm losing my footing here a little bit.
It's good enough.
Copy that. I got it steady here. I'll float away. Nice shot.
Couple of very large fans here on the right side.
Turn to 135 here.
Okay, I'm already at 135.
And I don't know if you noticed, but they were both oriented in the same direction,
feature there to our
which
right.
gives us a little bit of indication about the dominant current flow during the lifetime of that coral as it grew. And that would have been probably from the, let's see, upper left to the lower right.
So for Sirios, go ahead.
Just 3 more of these bird's nest sponges, several Okeanos bamboo corals, Chrysogorgia, and I think I see a large bamboo coral looming in the distance. Swatley and I aren't quite sure what that species is, but we wonder if it's Kurchatov gray eye, which would be actually quite significant because that species is the type for the genus Kurchatov. And I think it may be similar to the situation with the Iridogorgia that Les mentioned earlier, that there isn't really good type material. A type specimen or a holotype specimen is the specimen that the scientist who originally describes the species uses as the basis for that species. And it gets housed in a museum with a label, and so that in the future, if there's any debate about what is the species I'm looking at, you always reference that original specimen. And so if the type is damaged or lost, or the description of that original type isn't sufficient, it needs to be redone.
We finished with that move now?
Yeah, that move finished at 16:55.
16:55.
So about 3 minutes ago. Ready for another?
I think so. I think we need to determine if we want to keep on this heading or go south.
Did you make it to that little ledge?
The ledge was actually not quite at 135. It was a little north of there.
Copy.
So.
I mean, I feel like in the sonar it looks flat.
Yeah.
I'd say
you may want to head south from here.
Copy that.
I concur.
All right. I'll call on a move 180.
180.
That'll bring us right towards waypoint 3,
Roger
15
that.
meters. Bridge, this is ROV nav. Got another move if you're ready. Range 1.
So pilot, yeah, just to stress, what's important is not waypoint 3, but is being on the maximum part of this lip to make sure we're exposed to the flow that's coming from the valley on the other side.
Roger that. Watch
Thank you.
You can go for snap.
Your copy, bridge.
And let's see. Yeah, this is this morid, this codling we've seen now a few times. That really long fin in the front. Lepidion. I forget the species name. That's the genus name.
Lasers.
Lasers on.
Video is clear.
Copy. So our bathymetry indicates the local high should be south of here, right? Directly south?
Yeah, so we've reached the local high of this ridge feature. And now we'll head down here towards the local high of the whole feature.
Yeah, roger that. That's what Watchlead wants us to achieve if we can.
Yep.
And I'm still at 130. I'll wait till you get over towards the right side of the screen.
Roger
and I'll
that.
start changing my heading.
I personally am impressed at how many of these Pharonema sponges there are and Acanella in the flat parts here. It's more than a decent density. Lots more of these tall bamboo coral whips. Some black corals, hydrocorals, Lophelia, Thuarella. And it's interesting, you know, there are elements of this dive that I'm seeing that remind me of dive number 5. What did we call that dive again?
Was that Zenith?
Thank you.
Yep.
Zenith.
What's that, Pilot?
I was just letting you know I'm
that
pushing
there's
forward.
a fairly high
Just get
abundance
around
and
the little feature
diversity,
here on the
but
bottom.
it's not a total replication of that dive. We're actually seeing a number of
Alright, I'll
different
start
species
swinging
that we
around
didn't
to
see that
180.
day.
Copy that.
Some overlap, but plenty of differences.
You can take the fish if you want, Lydia. I'm sure they've seen plenty of these.
I see. I went to get a drink of water, and you did not find me my basalt, Scott.
It's not for lack of looking.
No. Look to the horizons, my friend.
I keep doing so.
He's right under us.
Go ahead, Bridge. Good copy, thank you. Pilots, that move is complete.
And so if you are just joining
Copy
us,
enough, thanks.
we are at a underwater volcano. That's probably, if I had to put a reasonable estimate on the age, it's probably about 2 million years old. It was probably formed around the same time as the general oceanic crust was being erupted at the Mid-Atlantic Ridge.
Roger that.
And we're currently on about 2 million year old seafloor, so that's the age of my estimate for the volcano. And the reason I would say that is because if we look in the multi-beam bathymetry that we mapped last night, the eastern edge of this volcano has been cut in half. It's been sliced
goes slightly
right on
southeast
one
from
end.
here.
Kind of about 20% of it is just cut off.
Maybe the direction we're
And
next
that
moving.
cut, that slice, is actually a fault. And it's a normal fault where everything to our east has been down dropped. We call that a normal fault. So it's slid down. And the remainder of that volcano we can actually see in the bathymetry on the seafloor topography on that bottom of that fault. And so that faulting usually occurs near the axis of the ridge. This area was at the axis of the ridge about 2 to 2.4 million years ago. So that makes me think this volcano is very old. Along with that, there's a lot of sedimentation. We see a lot of this white marine snow that's settled on the ocean floor. And then we also see a lot of this black, gnarly, knobbly crust. And I think those are concretions that's cementing together of things like pteropod shells, marine organism tests and shells. And so that tells me that this area has been sat here for a long time, and it's allowed all these shells and calcium carbonate to accumulate. Now, in a general context, we're about 20 kilometers away from the Mid-Atlantic Ridge, which is on our eastern side. And we're about 127 kilometers north of the Azores Islands. We've been diving on the Mid-Atlantic Ridge for about a week and a half now. This is our 8th dive. Today we're staying a little bit more shallow. Yesterday we were pretty deep. We got down to about 3,300 meters below sea level. Today we're diving down to 1,100 meters.
1702.
1702, copy.
And or
So,
excuse me, to about
kinda
1,200 meters, and we've come up to about 1,100. We're at about
Downhill
1107
to our
right
If you
now.
want another southeast move, co-pilot, how would you feel about that? Those have been 10-meter moves now?
15,
15.
1-5.
Okay. It does.
Yeah. We can get another southeast move.
Yeah, I think so.
135.
135, yeah, 15 meters.
Cover
If
that.
would,
You
thank
good with that, co-pilot? I'll call it in now. Bridge, this is ROV nav.
I'm going to work my way.
Got another move if you're ready.
Up.
Range 15
Pilot, watch
meters,
two.
bearing
Go watch
135
Are you
degrees,
able
speed
to tell
0.2
what way the
knots.
currents are moving here right now? Is it,
Sure.
I'm just curious if it's coming across that lip. I think that was
Good
maybe
copy,
an
thank
emergent,
you.
but anyway, sorry.
Holding stationary for a moment, and we'll try
Thanks.
to gauge him. Yeah. Judging on, based on our movement, it looks like I'm being pushed back, and you can see the marine snow going left to right a little bit.
Yep. So kind of out of the east-southeast.
Yeah.
Okay.
Affirmative.
So that's pretty good. So that's coming from the deep part of that fault.
Yep.
So coming out of the fault and over the lip. Mike says that was a larvation out of its house that drifted by. Thank you, Mike.
Yeah, east-southeast current it looks like. Very flat in this area. Let's see if we can find some more slope.
How about while we're in this area with all this stuff, we trip a Niskin?
Yeah, we can do that. Watch lead.
Thank you.
Stand by. I'll work with the front row. Co-pilot nav. You guys
Go ahead,
ready for a Niskin? I think watch lead 2 would like us to collect one here.
Copy that.
We have 2 left, so this will be our second to last. At your discretion, co-pilot.
Okay, spinning HPU, resetting Niskin, and all stations firing Niskin in 3, 2, 1. Looks like a good fire.
Oh, yeah, actually we have 2 more after this one. My apologies.
Okay, we're doing all the Niskins?
No, just they wanted one right here in this area.
Okay. So
Thank
I just
you, pilot.
fired one.
Copy that.
Yeah.
So we have 2 more after this one now.
Oh, 2 more left.
Yes, affirmative.
Okay.
No, I got a thumbs up from her.
Yeah, that was why I called out to Survey 2. I had Survey 2 latched.
Go ahead, Bridge. Thank you much. Pilot's move is complete.
Copy that. Thanks, Nav.
Copy Nav.
How's the blue view look here, co-pilot?
We have a listing of the black corals from Tinasako that we've seen during this dive. So 2 species of Bathypathes, 2 Antipatherine whips unnamed, a Parantipathes, a Trissopathes, a Chrysopathes, Antipathes viminalis, and Leopathes expansa, and a Phanopathidae.
Copy that.
So at least 10 different species of black coral that we've seen so far on our trip up this crater-like feature. I think, is there, I can't tell if it's one of these rockfish or a sea star, the pink thing on the rock to the left of where the lasers would be.
Sirius is still at 180. I can swing over.
Roger that. Did you want to zoom on something, watch lead?
Yeah, so there's a pink thing just under the lasers. I can't tell if it's a fish or a sea star.
Yeah, let's go for a zoom here. Thanks, video.
Sea star.
Sea star, yeah.
Yep. Looks like one that's been recently feeding. It's kind of bulged up in the center there.
Stand by.
That's a Storopathes black coral in the foreground, so I think we can add that to the list of black corals, so maybe now 11 species.
Did you want a closer look at any of these, watch lead? We're getting some shadows above us, some fish swimming.
I don't think so.
Copy that. Watch
I'm
this.
still trying to get to that high before we end the day.
Copy that.
Thanks.
Yep. Thanks, video.
Oh, yeah, you're right. What camera was that in?
So we're looking for as much slope
Must have
as we
gone
can
underneath
find
us
here.
then? Must have gone
How do you guys
underneath
feel?
us as we were zoomed in. Yeah.
Fish over to your left.
Fish on left. Copy.
Chimera.
So while we were... Oh, there it is. There's the chimera.
Go for zoom.
Nice.
What's your altitude?
Roger that.
Very nice.
Coming down.
Go a little up.
Laser's clear.
There you can see the lasers on it, so it's probably about 25 centimeters long.
I'm going to sit down here. Actually, I'll keep going.
Okay, I'm rotating down a little bit.
Roger that.
That's 55.
Copy that.
Video's clear.
Copy that. Pushing against a little current here. I think I'm pointed into it.
It's a good shot on Sirius.
Maybe he'll lead us to a higher slope. Looks like he's smiling. He's gonna go under us. Did you want any more imagery of this watch leader?
Nope,
Can we press
that's
on?
good.
Copy
Thank
that.
you.
Pushing out.
So we're at 11:06.
Just having a little look in the chat room, and I see that
maybe
there's
do one
already
more move
469
and see if we get
annotations
any-
made
if the
today.
depth
It's a
changes.
busy day.
Yeah, sounds good to me. Another southeast move, or
It's like a little
Yeah,
plexaurid
looks
here,
like we
probably
just
a
went 11:05.
paramerciad. Has its
but
own
you're a
little rock
bit off.
as its home, and then two large
that.
bamboo corals in the
15
background.
meters, 135. Bridge is RV now.
Sirius is at
Got another move
135
if you're ready.
already.
Roger that.
Range 15 meters, bearing 135 degrees, speed 0.2 knots, please. Good copy, Bridge. Thank you.
Okay, that's back at 45.
It may be warmer
Copy
on the
that, thank
seafloor today, but it's distinctly cooler in the control room.
Are you wearing a short sleeve shirt and shorts though?
I am, but I
Yeah.
have been doing that throughout the expedition. Today's the first day I'm cold. See some more stylastrid coral down there, the hydrocoral. And I think this is the dendrobrachia, this fan that's on the left side. The tall stuff is the bamboo corals that we suggest are maybe Keratoisis grayi. Wow.
Ask and you shall
That
receive.
is amazing. So I have the dive is playing in my room, and my roommate was in there and heard me and brought me my sweater. That's awesome. Thank you, John.
Wait, let me see if that works. I could go for a hot chocolate and a piece of pie.
Sadly, as you know, there's no feeding or drinking in the control room.
I'm waiting, Derek. Well, I'm a little sad that we didn't see any more basalt. I would have really liked to grab one for our colleague at the University of Lisbon. You know, I could have grabbed one of those pieces of talus on the lower slope, but I was really hoping out I would see something in place like those beautiful samples we grabbed the other day. And, you know, we talked...
Such a rookie. We've learned so much that, you know, when you see something early...
I know, but...
Yep. I'll get it later, and then it's like, I never see it again.
I know, but, you know, I was just trying to be respectful of all the ground you were trying to cover today. So
that's very good. Oh. I know the trade-off well.
Yeah.
Yeah. Like that bamboo coral, if this was my expedition, would have been in the box long ago.
Yeah.
Yeah.
Well, and I think that's something important to reflect. This is not a geologically focused expedition. It's not a biologically focused expedition. It's a multidisciplinary expedition. And Scott and I are not the principal investigators. We're not leading the scientific investigation here. We're acting as collaborators and kind of linchpins for everyone on shore to communicate what the scientific community thinks is the most important in terms of benefit to our discovery.
Yeah, it's really well said. And all the specimens that we're collecting, instead of going into personal research collections, are going into institutions where any scientist can bore them, given the right credentials and explanation for what they're doing, and returning the material later.
Roger. So we're on to something.
It's a true community effort.
I think we're probably not going to gain much more. Well, at least we wouldn't think we would, based on the bat feed.
Copy. You want to make it south?
Maybe south on the next move.
Yeah.
Understood.
And if Copilot's ready, I think we can put that in.
Okay. Yeah, I'm standing by.
Copy that. 15 meters due south.
Thanks, Nav.
Calling it a nav. Bridge is RB nav. We have another move. Range 15 meters, bearing 180 degrees, speed 0.2 knots, please. Good copy, Bridge. Thank you. Pilot set move is underway. Heading south.
Thank you.
I'll let you get a little over to the right, pilot, and then I'll change Sirius' heading.
Roger that.
I just had a name of Vazella for one of the sponges that we just recently passed. I'll have to look that up so I have the right search image. No.
Okay, pilot, I'm rotating over.
Copy that. Read my mind.
It's kind of interesting how the character of the habitat and the community has changed a little bit. Several of the species have continued from what we saw down below, but we've lost some others. But what's most obvious probably to everybody watching is how most of the fauna are aggregated on these rock highs, and you get a pretty different sort of situation when you look out on the sediments.
180.
Copy that.
some of these
Looks like
species
it
are adapted
starts
to live in the
slope up a
sediment, and
little.
others aren't. And so, if you see them, like
Yeah,
a
here
couple
I'll-
of these would look to be hydrocorals to me, they must surely be on little rock
Doesn't look
outcrops
like
that
any
we're just
big
barely
obstructions,
seeing
more
peak
or
above
less
the
the same
sediment.
bottom as we're seeing.
How much bottom time do we have left, Nav?
Same.
Interesting little patch here. Whole lot of Chrysogorgia colonies. That's the lighter pink ones. One Acanella bamboo. And the Thuarella just out of frame, and probably the Dendrobrachia, the white fan on the far right side. And then all these little whiter fans, all these smaller white fans, I believe are the hydrocoral, the Crypthelia. I've been getting some requests to collect one of those. We're just really trying to get to the highest point before we take any time to collect again. And I understand how difficult that is for everybody. Very interesting abundance here. Copy that. Yeah, so we've been in kind of a little saddle. You know, we came up the slope from the inside of the crater, and then we reached the rim, and where we reached it is a saddle between two high points, one to our north and one to our south, and we turned and we're moving towards the south to basically the last possible high point that we could get on this dive. It's interesting, you know, if this were the beginning of the dive, I'd have so much to say about everything that we're seeing here in terms of abundance and diversity, but I'm kind of exhausted after a day of talking about all these things. But it isn't to diminish what we're seeing here.
But it's amazing wherever we get one of these little patches of kind of this
outcropping.
Yeah, these concreted calcareous. They tend to be very, very rich, and then these more or less deserts of the visible epifauna.
I seem to recall we had this conversation, I think it was on dive 1, where we were surprised that when we got to the top, we just saw all of the coral rubble and very little live growth, and it was below the rim that we saw all the live growth. And, you know, at this particular second, there's a lot less of the live and the exposed substrate than there was a little bit further down the slope.
Yeah, those were skeletons of what kind of coral?
Of Guccisamia.
Guccisamia.
Yeah, which there was some question from our colleagues at the Azores, the University of Azores, about whether that would also be replicated at the top of this feature. They weren't sure. This is a little bit deeper, and so far I have not seen a single one of those. No, I want this.
Well, look at this little garden right here.
Sorry, it's got to tune up that. Okay. Let's see. Yeah, so another garden of, I believe, a whole bunch of Chrysogorgia in the foreground. A couple of large sideroid urchins moving through that garden. I see a purple plexaurid fan. It's kind of, we're edge on that fan, so it's a little hard to see, but that's on the left side of all these Chrysogorgia colonies. Then I'm seeing a couple of Parantipathes black coral whips. Further back, climbing up, several more Chrysogorgia, and then I think the Thuarella primnoid. And maybe we'll get a zoom of this. I saw Sako asking about that earlier. Just to the left of the lasers right now, there's something. Oh, no, that's okay. It's okay. It was waving in front of a sponge and kind of confused me. So, pilot, above the lasers, there's kind of a white brushy coral. I'd like to get a look at that, please. There's a stony coral, either madrepora, maybe it's Selenosmilia, that's what they were saying the other day. Just to the right of the lasers now. So we're having a look at this. It's the one that's kind of in the background. There's two of them together. They're waving top of the, yeah. So this is the primnoid, and earlier today we had some nice imagery of a species in this genus, and there's some question in the chat about whether this is a second species or whether it's the same one, and you know, it's hard to say about the way they branch. Yeah, keep in mind, sure, that, you know, we've already asked the ship to move. We're moving pretty quickly so we can achieve this local high. So the ROV pilot doesn't have a lot of time to take a close-up before he's going to get pulled away. That's the one, yeah, in the background there in the center. Yep, that's the one. There we go. And these polyps are too small to get really good images of, but we can get some clues by the pattern that these small branchlets are rising from the main axis. Ultimately, for some of the species characteristics, you would simply have to have it under a microscope to be able to know. Okay, that's good, thank you. Thank you, Mary Bilan says that sclerotinian is Madrepora, genus Madrepora. This looks like it steps up.
Mm-hmm. Now, so those look like they might be the heavily encrusted and kind of sedimented pillow lobes that we saw, yesterday.
There's one cracked over there.
Yeah. So you can imagine trying to grab a sample of something like this. You're really, you're probably just going to end up grabbing a sample of that heavy crust, and not necessarily that nice, dense crystal rock with that glassy rind that we got yesterday.
Pilot, as we get close to this next rock, on the left side of it, there's kind of a loby, upright, platey sponge in the background there. Yeah, if we can get a look at that. Joanna earlier was asking about sort of a flabellate sponge, and this is the closest I've seen since that time. The one to the left of that. Yep, that's the one. There's an interesting kind of, this is for the folks on shore, there's an interesting sort of planar, or much more planar Acanella on the top of that rock. It's actually, yeah, no, this is good. The one with the urchin on it, yeah, that'll be a good view of the urchin and see what it's feeding on. So it's actually much more like a thin plate / amphitheater than I thought it was. I thought it was going to be a thicker thing. So I'm not sure this is what she meant, but it's still a good view. Boy, look at the thickness of the spines on that urchin. Well, it must be an old one. And it's interesting, yesterday we also saw several occasions of this kind of enveloping umbrella-like sponge over a Madrepora colony, and here we're seeing something very similar. And I'm wondering if the sponge larva is settling on the calcium carbonate skeleton of that coral, and that's why it looks like they have an association. That's okay. To me, this looks similar to something that we sampled earlier in the week. A little bit hard to tell because I'm not a sponge taxonomist, so I don't know at this gross scale what are the characteristics that are being looked at. I will note that on this urchin, we have a crab, just like we did on the last one. I think it's the same kind of crab, that sort of decorator crab. It's kind of interesting. And again, we have zoanthids growing on the spine. We have a brittle star. So they're all in a little mobile home. Okay, that's good for me. The purple sea fan there on the left is a Plexauridae. Very diverse family of corals in both shallow water and deep water. And unlike what I had described earlier for the bamboo corals, primnoids, and chrysogorgiids, the center of radiation, the center of diversity or evolutionary diversification for the Plexauridae is not in deep water. It's in shallow water. And so there are some representatives that have moved into the deep water, but they haven't speciated to the degree that those other three families have. The black corals are another one that have really radiated in deep water. Most of the diversity is in the deep sea. Huh, looks like a whole field of little hydrocorals up here. Oh, there's a little iridogorgia. I haven't seen iridogorgia in quite some time. I made a collection of one earlier, but there's one on the vertical rock face just on the far left. Oh, look at that. Looking in the distance. Note all the fans are kind of oriented in the same direction. And that's so that they get the width of the fan to capture the maximum amount of current. You don't want to be edge on to the current. You want to be face on to the current. And you can see here that it's occurring in different species. So it's a really nice indication. And right now our current is coming across from the upper left to the lower right. And that would mean it's coming from the deeper side of this crater feature on the fault side and coming across the upper rim and kind of down into the crater. Beautiful, I think, euplectellid glass sponge. Well, the community just keeps on coming. I just realized that I have not seen any entry in the chat room in quite some time, and I just typed something and nothing happened.
I think I just saw a message that it says it seems Scott doesn't notice he's kicked out from the chat.
But I'm not though. I can see it. This is weird. That is very strange. I've never had a situation. Yeah, I don't understand what's going on.
It gave me that earlier too.
Huh. No, that's how I knew. I saw there was no navigation input, and then when I just tried to reload it, I've got a "your connection is not private" message. So I don't know what's happened here, but you are right, Onshore. I am out of the chat. Do you still have it live?
Yeah.
Okay. So maybe I should, I don't know what people are saying or asking. Copy that. If it is, before we leave bottom, we'll fire one more Niskin number 4. Thank you. So folks on shore, we're almost out of time here on the bottom. We're at, actually, look at that. We surpassed 1,100. We're at 1,097 meters. And so this nice little garden that we see up here will be our final move. Yeah, I'm getting some reports from shore. There's some folks also having some problems.
Should we just have both manipulators take a big giant hug of everything they see? Grab some rocks, some corals, some sponges.
I'm trying to...
Joanna is asking if we can collect the rock holding the yellow sponge.
That huge rock here?
I think she might be joking.
Okay, good.
I tried.
So, yeah. I also tried logging in through Pigeon. That didn't work either. Well, I mean, we could take a clipping of the...
Asako's asking about the hydrocoral.
Where? I don't see it. So do we see a white fan anywhere right in front of us?
Was it on that rock?
What rock?
Yeah.
The rock with the white sponge? It's got to be one big enough that you can grab. I mean, those things are really brittle. They'll shatter.
I think it might have been on the big rock.
Okay. There might be...
Oh, he says in front of us. He says right in front of us. Next to the Bathypathes.
The Bathypathes is back to the right.
Maybe there's a...
Yeah, there's a delay.
There's a delay. To the right.
Yeah. Yeah, it's pretty small. I don't know.
Oh, that tiny little white thing?
Yep.
Oh, yeah.
I mean, no, it's to the right of the lasers by that reddish feather. The thing is, they're so brittle, they're shatterers. So I think you've got to have something big enough that you can actually get in the claw to retain some of it.
Yep.
What two color morphs is she referring to?
I wasn't sure if she was referring to the sponge. Oh, she wanted a piece of the yellow sponge. It is the yellow morph of Hertwiggia.
Yeah.
Yeah.
Yeah. I mean, everybody wants everything here at the end. I understand. Yeah. I do too.
Is that a little bit of the yellow sponge on the upper left?
Yeah.
She asked about this earlier as well.
Yeah.
Yeah.
And so it wasn't the yellow morph we got earlier. I guess it was the white morph that we got earlier in the expedition. Yeah. I mean, what do you think? Do you think you can grab that yellow thing in enough time? Then let's go for it. If you don't, then, we're done. I understand. Yep. Yep.
John is asking if we could zoom in to make sure.
Sure. I'm going to suggest, I'll let you guys make the decision. You know your tools, but that's a pretty soft sponge, so maybe you could just suction it off of there. If you think it's easier to grab, I leave that to your discretion.
Yeah, and Joanna said we got the white one earlier, the white morph.
Yeah, I'm just trying to make a suggestion that's whatever's fastest, given the bottom time.
Oh, no, I was just saying your question about the color.
Yeah.
Oh, yeah.
I'm talking about
Sorry.
Yeah, I believe that is the Hertwiggia. So I believe that is the target. Understood. It's actually sitting on a much larger sponge. Yeah, I mean, I can't get in anywhere. I can't get in on pigeon. I'm getting reports from shore. It's the same thing. Yeah. That was me ignoring.
No worries. We appreciate the valiant attempt.
Yep.
It was,
And just a reminder for tripping Niskin before we lift off bottom. Thank you.
Yeah. We've seen those a few times. I sure. Hopefully we'll see them again. Yeah, and Joanna thanks you as well. And I'm sure when she's on next year's, you'll have lots more attempts for her. Every sponge.
All right, folks on shore, so that's the end of the bottom portion of our dive. It was pretty successful. Just before everybody checks out, just want to let you know that we will have a dive planning call about tomorrow's dive, and let's say give us at least 7 or 8 minutes, so let's say 5 after the hour, so we can stretch a little bit, and we just got a little bit of paperwork to do on board. But otherwise, thank you all for joining us. I apologize, I still can't get back in the chat room, so I don't know who's there, but I think this was a pretty successful dive. We went into this crater at, let me get my notes here, 1,235 meters, and we climbed the slope of the crater towards the upper rim, and we weren't sure what exactly to expect. Sediment-filled. There was some suggestion that based on other studies of some craters in the area that there might not be any animals at all, but those were at a shallower depth. Well, that certainly wasn't the case here. We actually found a marvelous diversity of corals and sponges and fish and many different associates all throughout the entire dive. The characteristics changed as we got to the flatter upper rim. Fewer numbers of species, but still a very good abundance of species. And then we also saw some really large fans, and we made several collections that will keep us busy. and have made 4 water samples so far for eDNA. We'll make 1 more in the deep scattering layer as we go up, and then we collected 1 rock that had several sponges on it, as well as what I think was a tunicate. And of course, I will spend some time tonight sampling that. So thanks everybody. Thanks everybody who made observations in the chat that helped us out. And as I say, 5 after the hour, we'll talk about tomorrow's dive. The rest of you, we will see you tomorrow in the deep sea.